Good morning, everyone, and thank you for joining us. I'd like to call this meeting of the Anesthesiology and Respiratory Therapy Devices panel to order. I'm Dr. Stephen Nathan, the chairperson of this panel. I am the medical director of the Advanced Lung Disease and Lung Transplant Program at NOV Fairfax Hospital, which is in Falls Church, Virginia. A note for the record that the voting members present constitute a quorum as required by 21 CFR Part 14. I would also like to add that the panel members participating in today's meeting have received training in FDA device law and regulations. For today's agenda, the panel will discuss ongoing concerns that pulse oximeters may be less accurate in individuals with darker skin pigmentation. The committee will also discuss factors that may affect pulse oximeter accuracy and performance, as well as the available evidence about the accuracy of pulse oximeters, recommendations for patients and healthcare providers, and the amount and type of data that should be provided by manufacturers to assess pulse oximeter accuracy and to guide other regulatory actions as needed. I wanted to mention that FDA has received 25 comments that were submitted to the docket as of October 31st, 2022. These documents are available to the public at the following website, www.regulations.gov.docket/FDA-2022-N with a cap -2110. And these have been sent to the panelists and FDA for review. Before we begin, I'd like to remind the public and panelists that this is a non-voting meeting and ask our distinguished committee members and FDA participants attending virtually to introduce themselves. Committee members, please turn on your video monitors if you have not done so already, and unmute your line so that we can hear you when you speak. When I call your name, please state your area of expertise, your position, and affiliation. Let's start off with Dr. Cassiera, please.
Good morning, everyone. I'm Hugh Cassieri. I'm critical care pulmonary. My specialty is I'm the medical director for respiratory therapy services at a large transplant center. I work for Northwell Health, and I'm very excited to be joining the meeting.
Thank you, Dr. Cassieri. Sorry, I butchered your name a little bit there. I'll try and get them right as I go. Next is Dr. Kirsch.
Good morning. I'm Jeff Kirsch. I am the professor of anesthesiology and pain management at the University of Washington. I'm the chief of anesthesiology at Harborview Medical Center.
Thank you, Dr. Kirsch. Next is Mr. Branson.
Hi, this is Rich Branson. I'm a respiratory therapist. I'm a professor in the Division of Trauma and Critical Care at the University of Cincinnati, where I've been the director of clinical research for 30 years. I'm also the editor-in-chief of Respiratory Care.
Thank you, Mr. Branson. I'm not sure if Dr. Yarmus is on. Dr. Yarmus, if you're on, if you could please introduce yourself. All right.
Yeah, hi. Hey, everybody. Lonnie Yarmus. I'm the clinical chief of the Division of Pulmonary Critical Care at Johns Hopkins Medicine.
Thank you, Dr. Yarmus. Next, we have Dr. Wilson.
Morning, Bill Wilson. I'm an cardiac anesthesiologist and critical care physician. I was the CMO at the University of California, Irvine Medical Center, and I am now the CMO at Massimo and in charge of clinical research.
Thank you. Next, we have Mr. O'Brien.
Hi, I'm Joe O'Brien, and I am President and Chief Executive Officer of the National Scoliosis Foundation. I am also a patient with 21 surgeries.
Thank you, Mr. O'Brien. Next is Ms. Edwards.
Hi, I'm Beverly Edwards. I'm the consumer representative on the panel. I teach at the University of Memphis in Tennessee.
Thank you. Dr. Hudson is next.
Yes, good morning. I am Arlene Hudson. I'm a cardiac anesthesiologist, and I am chair of the Department of Anesthesiology at the Uniformed Services F.A. Baer School of Medicine.
Thank you, Dr. Hudson. Next, please, Dr. Loeb.
Good morning, everyone. My name's Robert Loeb. I go by Butch, but Robert's fine for this meeting. And I am Professor Emeritus at University of Florida, now living in San Diego area.
Thank you. Next, please, Dr. Klein.
Good morning, everyone. I'm Andrea Klein. I am a pediatric acute care nurse practitioner and nurse practitioner director at University of Michigan Health in Ann Arbor, Michigan.
Thank you very much. Next, please, Dr. Hennessy.
Good morning, everybody. My name is Sean Hennessy. I'm a pharmacist epidemiologist at the University of Pennsylvania, where I lead the Division of Epidemiology.
Thank you very much. And next, please, Dr. Connor.
Hello, I'm Jason Connor. I'm a biostatistical consultant, owner of Consulate Stat, also assistant professor of medical education at University of Central Florida. I usually don't introduce myself further back, but I also, before becoming a PhD biostatistician, did PhD work in biomedical optics, specifically involving using lasers for diagnostics. So that seems relevant today.
Thank you, Dr. Connor. Next, please, Dr. Katz.
Hi, I'm Dr. Elliot Katz from Boston Children's Hospital. I'm a pediatric pulmonologist and sleep physician at Harvard Medical School.
Thank you. Next, please, Dr. Collip.
Hi, good morning. I'm Nancy Collip. I'm a professor of medicine and neurology at Emory University. My expertise is in sleep medicine. I run the Emory Sleep Center.
Thank you, Dr. Collip. Next, please, Dr. Lynch.
Hi, I'm Jen Lynch. I am a pediatric cardiac anesthesiologist at the Children's Hospital of Philadelphia. I also have a PhD in physics and do research in the field of biomedical optics.
Thank you very much. Dr. Alam.
Good morning. My name is Murad Alam. I'm a dermatologist and clinical researcher at Northwestern University, where I am a professor and vice chair of the Department of Dermatology.
Thank you very much. Next, please, Dr. O'Connor.
Hi, I'm Michael O'Connor. I'm a professor of anesthesiology and critical care at the University of Chicago. I'm a practicing anesthesiologist and intensivist.
Thank you very much. Dr. Lee. Dr. James Lee. I'm not sure if Dr. Lee is.
Hi, James Lee, Division Director for Sleep Disorder, Breathing, Respiratory, and Anesthesia Devices here at FDA.
Thank you very much. Dr. Loya Berrios.
Hi, my name is Nilsa Loya Berrios. I'm an epidemiologist and acting associate director in the Office of Health Technology 1 at CDRH.
And last but by no means least, Dr. Eidelman, please.
Good morning, everyone. My name is Malvina Edelman. I'm director of the Office of Health Technology 1, which oversees all of the anesthesia devices. Thank you all for joining us today, and we truly look forward to hearing your thoughts and opinions on this very important matter.
Thank you, everyone, for introducing yourselves. As folks can tell from the introductions, a very well-rounded panel that we have today, so thank you very much. Next, Dr. Akinola Owajopi, the designated federal officer for today's anesthesiology devices panel, will make some introductory remarks.
Hello, good morning, everyone. I'm Dr. Awajope. I'm the DFO, the designated federal officer for this advisory meeting. I will now read a conflict of interest statement to everyone. The Food and Drug Administration, FDA, is convening today's meeting of the Anesthesiology Devices Panel of the Medical Devices Advisory Committee under the authority of the Federal Advisory Committee Act, FACA, of 1972, with the exception of industry representative. All members and consultants of the panel are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulation. The following information on the status of these panels compliance with the federal ethics and conflict of interest laws covered by, but not limited to, those found at 18 U.S.C. Section 208 are being provided to the participants in today's meeting and to the public. FDA has determined that members and consultants of this panel are in compliance with the federal ethics and conflict of interest laws under 18 U.S.C. Section 208. Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have a financial conflict when it is determined that the agency's need for the particular individual's services outweigh his or her potential financial conflict of interest. Related to the discussion of today's meeting, members and consultants of this panel who are special government employees or regular federal employees have been screened for potential conflict of interest of their own as well those imputed to them, including those of their spouses or minor children, and for purpose of 18 U.S.C. Section 208, their employers. These interests may include investment, consulting, expert witness testimony, contract, grants, credits, teaching, speaking, writing, patents, royalties, and primary employment. For today's agenda, the panel will discuss the ongoing concern that pulse oximeters may be less accurate in individuals with darker skin pigmentations. The advisory panel will discuss factors that may affect the pulse oximeter accuracy and performance. The available evidence about the accuracy of the pulse oximeter, recommendations for patient and healthcare providers, the amount and the type of data that should be provided by manufacturers to assess the pulse oximeter accuracy and to guide other regulatory action as needed. Based on the agendas today's meeting and all financial interests reported by panel members and consultants, a conflict of interest waiver has been issued in accordance with 18 U.S.C. Section 208, Part B, 3 to Dr. Stephen Nathan. Dr. Nathan's waivers addresses his personal financial interest in healthcare sector, mutual fund that contain underlying asset shares in potentially affected and competing firms. The aggregate values of his holding in the fund is between 200 to 300,000. The waiver allowed these individuals to participate fully in the panel deliberation. FDA's reason for issuing the waivers are described in the waivers documents, which are posted on the FDA website. Copies of this waiver may also be obtained by submitting a written request to the Agency Division of Freedom of Information, and the address is 5630 Fisher Lane, Rockville, Maryland. Dr. Williams Wilson is serving as industry representative, acting on the behalf of all related industry. He is employed by Massimo Corporation. For the record, agency notes that Dr. Christopher Halmon, who is an invited guest speaker with us today, has acknowledged his employer's interest in the form of a research grant that is federally funded. Mr. Paul Bochelder, who is also an invited guest speaker with us today, has acknowledged his employer's interest with numerous affected competing firms in the form of research contract. Dr. Philip Beckler, another invited guest speaker with us today, has acknowledged his employer's interest with multiple affected competing firms in the form of research studies. We would like to remind members consultants that if the discussion involve any other product or firm not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participant need to exclude themselves from such involvement, and the exclusion will be noted for record. FDA encourage all participants to advise the panel of any financial relationship they may have with any firm at issues. I will now read the temporal borrowed memo. For the duration of the Anesthesiology and Respiratory Therapy Devices panel meeting on November 1st, 2022, Sean Hennessy, Pharmacy D, PhD, has been appointed to serve as a temporary non-voting member. And for the record, Dr. Hennessy served as a consultant to the Drug Safety and Risk Management Advisory Committee at the Center for Drug Evaluation and Research, CEDAR. This individual is a special government employee who has undergone the customary conflict of interest review and have been reviewed materials to be considered to this meeting. The appointment was authorized by Russell Fortney, the Director Advisory Committee, Oversight and Management Staff, on October 24th, 2022. A copy of this statement will be available for review and will be included as a part of the official transcript. Before I turn the meeting back to Dr. Nathan, I would like to make a few general announcements in order to help the transcribers identify who is speaking. Please be sure to identify yourself each and every time that you speak. The press contact for today's meeting is Shawana Nelson. Thank you very much. Thank you, all the panelists. I'll hand it back to Dr. Nathan again.
Thank you very much. We're going to now move on to some FDA opening remarks. And I'd like to now invite Dr. Jeff Shuren, Center Director for the Center for Devices and Radiological Health, as well as Rear Admiral Ricardi Arahu, Associate Commissioner for Minority Health, to provide introductory remarks, starting with Dr. Shuren.
Well, thank you. Good morning, and thank you to our panel members and other participants in today's meeting. Pulse oximeters are non-invasive tools used to estimate blood oxygen saturation. Although pulse oximetry presents benefits for patients, the limitations and risks associated with inaccurate pulse oximeter readings must be well understood. During the COVID-19 pandemic, the use of pulse oximeters significantly increased for both clinical and at-home settings. In 2020, FDA became aware of a post-market signal regarding pulse oximeter performance. In a December 2020 letter to the editor in the New England Journal of Medicine, the authors of a retrospective cohort study found that patients who self-identified as Blacks were almost 3 times more likely to have hypoxemia not detected by pulse oximetry when compared to white patient counterparts. These results suggest that Black patients are at increased risk for failing to detect occult hypoxemia when pulse oximetry is used, or at least some pulse oximetry devices are used to triage patients and adjust supplemental oxygen levels. Numerous subsequent publications have also raised concerns that there is a potential bias in pulse oximetry performance due to skin pigmentation, potentially putting patients with darker skin pigmentation at risk of delays in treatment and worse patient outcomes. Therefore, we are holding today's panel meeting for the following purposes and objectives, specifically to discuss the impact of skin pigmentation on clinical performance of pulse oximetry technology, which could be indicative of racial disparities in the performance of these devices. Discuss and make recommendations regarding the design, conduct, and reporting of data for studies assessing the accuracy of pulse oximeters and potential bias due to skin pigmentation. Promote transparency on this important public health issue and on the agency's activities to date, and provide a public forum for the many stakeholders impacted by this issue to express their views: patients, healthcare providers, professional societies, researchers, and industry. Our agenda for today's meeting includes the following: an overview of the current regulatory framework for pulse oximeters, including relevant standards, guidance documents, and pre-market study requirements; a summary of the currently available real-world evidence regarding the potential bias in pulse oximetry due to skin pigmentation; invited speaker sessions where the perspectives from adult and pediatric patients, researchers, 10 professional societies, industry, and numerous authors of real-world evidence papers will be shared; an open public hearing; and a panel discussion of FDA's questions, as well as the panel's interpretation of the currently available real-world evidence on this issue, recommendations regarding tools to assess skin pigmentation for future studies, recommendations regarding expectations of pulse oximetry accuracy across various clinical settings, arterial oxygen saturation ranges, and patient subpopulations of varying skin pigmentation. Also, whether ARMS is the best metric to assess device accuracy, and recommendations regarding device labeling to convey the potential inaccuracies due to skin pigmentation, as well as the content of labeling for lay users who may use pulse oximeters at home. FDA has taken several actions to inform today's discussion and to address this issue. They include conducting a comprehensive literature review, reviewing data from medical device reports, conducting a market assessment of pulse oximeter devices, performing a systematic review of desaturation studies submitted in pulse oximeter 510Ks since 2000, issuing a safety communication in February 2021 to inform patients and healthcare providers regarding the limitations and risks of inaccuracy for pulse oximeters and to consider these when using such devices to make diagnostic and treatment decisions. And finally, awarding grants to the USCF Stanford CERSI to conduct 2 real-world prospective studies that will assess the performance of pulse oximeters in the adult and pediatric patient populations. The issue of racial disparities in pulse oximetry is of great public health importance, and it further highlights the need to appropriately assure that all medical devices are safe and effective in all the populations in which they're intended to be used, which is one of the goals of CDRH's 2022 to 2025 strategic priority to advance health equity. We have invited Rear Admiral Charday Araujo, Associate Commissioner for Minority Health in the Office of Commissioner, and the Director of the FDA's Office of Minority Health and Health Equity, to join us today for discussion. She'll deliver some welcoming remarks. So let me now turn it over to Rear Admiral. Welcome.
Thank you, Dr. Shuren, and good morning, everyone. I am glad to be able to join you for this advisory committee meeting on such an important topic. The ongoing effort to better understand and evaluate the relationship between race, ethnicity, skin pigmentation, and oximeter accuracy is crucial and related to the priority across FDA, including CDRH and the Office of Minority Health and Health Equity, to advance health equity and address disparities among racial and ethnic minority populations. And of course, the FDA's Office of Minority Health and Health Equity engages broadly across our agency, as well as with external stakeholders, and we have a dedicated mission to protect and promote the health of racial and ethnic minority and other diverse populations through research and communication that addresses health disparities and advances health equity. As we have all witnessed over the past 2.5+ years, the COVID-19 pandemic brought health equity to the forefront of our national dialogue and highlighted racial inequities and disparities that have long adversely impacted diverse communities. So throughout the FDA, information on race and ethnicity has and continues to be integral to our understanding of health issues affecting the US population and support of improving population health outcomes. Central to our work at FDA is also to support the consistent collection and reporting of race and ethnicity data, because we know that variations associated with race and ethnicity have been correlated with risks for certain diseases and conditions and responses to regulated products. Understanding these factors remains important for identifying and addressing disparities in health, which is vital to achieving health equity. And advancing health equity is a top priority for the FDA, including CDRH and the Office of Minority Health and Health Equity. The performance of medical devices needs to be well understood to mitigate any negative unintended consequences for patients in different populations. We are committed to the continued evaluation of the safety and effectiveness and availability of regulated products, including how medical devices perform across racial and ethnic groups. So today's discussion on the real-world performance of pulse oximeters and factors that impact their accuracy will inform FDA as we consider the regulation of these medical devices. So with that, again, I'm glad to be able to join you today, and I look forward to this important discussion. Thank you.
Thank you very much, both Dr. Shuren and Rear Admiral Arahu. I would now like to invite the FDA review team to start their presentations. The first session will have presentations covering the regulation of pulse oximeters, pulse oximeter technology, standards for pulse oximeters, and the real-world performance of pulse oximeters. The second FDA session will cover various topics related to clinical study design, methods, and analyses for studies evaluating pulse oximeters as well. I would like to remind public observers at this meeting that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel chair, that be me. FDA, you may now begin your presentation. Thank you.
Good morning. I'm here to present regarding the agency's regulations of pulse oximeters, which includes reviewing the technology, its limitations on accuracy, and how the agency regulates the device type. My name is James Lee, and I am the Division Director here at OPEC OHT1, Division for Sleep Disorder Breathing, Respiratory, and Anesthesia Devices. My co-author today is Mr. Neil Patel, our Senior Reviewer in the Anesthesia Devices team. Pulse oximeters are widely used in various situations and clinical settings. In particular, and in recent years, there has been an increased utilization and public familiarization with pulse oximeters inclusive of the COVID pandemic. In general, the FDA considers the environment of use and intended populations when evaluating the safety and effectiveness of devices like pulse oximeters. Factors for consideration in our review include the fields of use and populations like healthcare providers and facilities, and also patients who might require indirect measurements of blood saturation or SpO2. It is important to keep in mind that these measurements of oxygenation levels in the blood or ratios of oxygen-bound hemoglobin are surrogate measurements of physiological parameters, meaning that they are not direct measurements of oxygenation levels, but indirect measurements of oxygenation via light wave absorption of the tissue. True oxygen levels in the hemoglobin are from SaO2 measurements obtained via arterial puncture, which is considered the gold standard for assessment of blood oxygenation saturation levels. Blood gases, or SaO2, while the most accurate in assessment of oxygenation levels, involves an invasive procedure. SpO2 is an estimate, therefore has an error, and the measured values are expressed as a percentage with a labeled error range. As mentioned on the prior slide, SpO2 is estimated as a percentage of oxygenated hemoglobin over the sum of oxygenated and deoxygenated hemoglobin. An important factor is that oxygenated and deoxygenated hemoglobin have different absorption spectra, which allows the use of optical techniques that utilize two or more different wavelengths of light to measure differences in absorption of the tissues within the targeted field. The lower figure shows two typical wavelengths of light used to measure SpO2. We have red light at 660 nanometers and infrared light at 940 nanometers. The total absorption is relatively low, allowing enough light to pass through the light-emitting LEDs to the photodetector sitting opposite, as shown in the figure above. Oxygenated hemoglobin, defined as O2HB in the lower figure, absorbs or attenuates the amount of infrared light more than compared to the red light. The opposite is true for deoxygenated or reduced hemoglobin, which absorbs more red light and allows relatively more infrared light to pass. A second basic principle of pulse oximetry is the presence of a pulsatile arterial signal, which allows changes in light absorption to be measured. The upper figure to the right shows the changing absorbance of light during an arterial pulse. The portion that changes is often referred to as the AC portion of the signal. The portion that remains constant due to the presence of residual arterial blood, venous blood, and other tissues is referred to as the DC portion of the signal. With a ratio of the AC to DC signal, both red and infrared light, a ratio of ratios referred to as the R value, which again is the ratio of AC to DC for the red signal over the ratio of AC-DC for the infrared signal, can be calculated. With the calculated R value, you can estimate the SpO2 value using empirical calibration data as shown in the lower figure. Regulation of prescription pulse oximeters are based on a moderate device risk level, meaning these devices are reviewed under the FDA CDRH 510K program and cleared under the basis of substantial equivalence. As described in our 2013 FDA guidance document for pulse oximeters, these devices undergo clinical testing, bench testing, and other standardized tests like biocompatibility to review the device's relative safety and effectiveness against predicates with the same intended use. These devices are considered for spot checking and trending tools and not for diagnosis of a disease. A cornerstone of the FDA guidance document and review is the use of internationally recognized standard for the evaluation of pulse oximeters. This standard establishes the basic safety and testing needed and defines protocols and tests that are utilized in the evaluation of a pulse oximeter's defined performance. Specifics on these tests and framework of the standards will be covered by our next presentation. These pulse oximeters are labeled for trending or spot checking for oxygen saturation levels of patients who are hospitalized, in doctor's office, and also for prescribed use in homes by patients under the care of a physician. Regarding what is considered to be over-the-counter, regulation of these devices are under enforcement discretion and fall under product codes like PGJ or OCH. These are specifically for general wellness, which apply to sports and aviation use, but are not intended for medical purposes. For this reason, they do not undergo FDA pre-market review and can be sold directly to customers and patients. Please see our reference here at the link for our general wellness guidance on low-risk devices. The FDA reviews medical technology like oximetry under established regulations under the Code of Federal Regulations or CFR. Here we would like to provide the established definitions of these regulations. Furthermore, the FDA buckets or groups within these regulations by product codes, which allow for a more granular definition of products and allow for detailed assignment according to both established regulations and sorting devices by product code. What should be noted is that oximetry devices utilize both visible and invisible wavelengths of light and rely on absorption by tissues where the remaining signal can be transmitted to an opposing sensor. Innate configurations like this, while effective in picking up physiological signals, have limitations due to the technology and configuration of the sensor system. Highlighted here are several confounding factors that may contribute to either inaccuracies of the signal or increase the variability in relation to ground truth or absolute oxygen saturation in hemoglobin. One of the main considerations is the level of skin pigmentation, which is a major part of our discussions today. In addition, there are patient dispositions that may arise or challenge the accuracy of the SpO2 signal due to the ratio of oxygenated and deoxygenated hemoglobin, which does not include considerations for other types of hemoglobin. Therefore, disorders of the hemoglobin molecule or anemia may cause a reduction in pulse oximeter accuracy. In addition, other factors like intravascular dyes, tattoos, nail polish, and ambient light may interfere with the sensor and contribute to lower levels of accuracy. Regarding MDRs, please note that this information was included in our executive summary. We conducted a keyword search using SKIN for adverse events submitted to our medical device reporting or MDR system, dating back some 22 years. Reports of death were reviewed for inaccurate SpO2 readings as a factor, of which there were 99 such reports. But a detailed review found that there was a lack of sufficient information to evaluate an association between these adverse events that were reported and inaccurate readings. Please note that this was not included in our executive summary. However, we are including this slide here to provide a full context of information for consideration. The chart on the right shows the total number of MDRs spiked after the 2013 guidance was published and have trended downward. An additional search was conducted with keywords such as inaccurate, inaccuracy, and incorrect to see if those helped in informing a potential source of the problem. Continuing with the agency's analysis, we took a granular look at the information contained in the individual reports. Again, this was not included in the executive summary. A further detailed review found that there was a lack of sufficient information to evaluate the association between adverse events and inaccurate readings. We found that the top 3 reported health effects relate to no consequence to the patient, and the top 3 reported device problems related to inaccurate measurements by pulse oximeter devices. Importantly, although MDRs are a valuable source of information, this passive surveillance system has limitations. The incidence, prevalence, or cause of an event typically cannot be determined from this reporting system alone due to underreporting of events, inaccuracy in reports, lack of verification that the device caused a reported event, and lack of information about the details, such as frequency of device use. Because of these limitations, MDRs comprise only one of the FDA's several important post-market surveillance system data sources. These reports, along with data from other sources, can contribute important information to a medical device's benefit risk assessment. Reports like these help the agency look at a class of devices and consider in totality of post-market performance. In total, while oximetry has provided an excellent tool in evaluating non-invasively important vital signs like oxygenation levels, our conversation will continue with our next speaker, Dr. Sandy Weininger from our Office of Science and Engineering Laboratory, who will discuss how the FDA uses international standards to address issues of effectiveness and basic safety. Thank you.
Welcome, distinguished panel members, and thank you for participating in this important activity to understand the performance of pulse oximeters. I am Sandy Weininger, an electrical biomedical engineer in CDRH's Office of Science and Engineering Labs, where I perform regulatory science research on the safety of interoperable systems and consults for the safety of electromedical devices. I am the current chair of the ISO-IEC Pulse Oximeter Committee and have been involved in oximeter standards and performance assessment of pulse oximeters since the early 1990s. Pulse oximeters were introduced in the early 80s, and clinical societies very rapidly adopted them to support safe care. It was quickly recognized that engineering safety needed to be addressed as well. As technology improved over the years, devices have evolved in response to clinical observations of underperformance, including darkly pigmented and lightly pigmented individuals, motion tolerance, and low perfusion. Let's first look generally at how safety is supported by standards, then specifically at how the pulse oximeter standard addresses these issues. What does it mean to be safe? It means that the relevant hazardous situations have been identified and risk control measures built into the device. This happens most effectively when one has a structured design and development process in place and then makes good decisions within that process. ISO 13485 establishes the process for design and development of medical devices. It works in concert with ISO 14971, which provides the process for identifying and assigning risk to hazardous situations. These standards establish the how-to for design, but not the how good should the design be. The how good comes from IEC 60601, the general standard for the safety of electromedical devices, which provides answers in terms of safety requirements, test methods, and acceptance criteria. ISO 80601-2-61, our pulse oximeter standard, inherits these processes and requirements and customizes them for pulse oximeters. In general, international standards are intended to promote a minimum level of safety and performance that is accepted by regulators around the world. The standard is developed with the intent of harmonizing with these local requirements, as has been done with FDA's guidance document. Let's look at what the standard covers. The scope of the standard is for devices that provide an estimate of functional arterial hemoglobin saturation in all use environments. The standard provides requirements, test methods, and acceptance criteria for engineering basic safety and essential performance, but does not address what performance is necessary for a particular type of clinical application. In addition to the safety requirements, the standard provides significant supplemental and educational material to help properly understand and interpret the results from an oximeter. The standard recognizes and addresses sources of error and attempts to address these to facilitate testing and characterization of the device. One difference from the FDA guidance document is the standard does not specify the type of use, spot testing versus continuous monitoring. The standard uses the construct of whether the device has a physiologic alarm signal, as this was determined to be the primary difference in clinical use. There's no need for an alarm when spot checking. FDA has a process to recognize standards for use in regulatory activities. While FDA encourages the use of recognized standards, it may not recognize the full extent of the standard. This is the case for pulse oximeters. FDA does not recognize the clause that sets a limit on accuracy. In the US, the FDA guidance document takes precedence. In the FDA guidance document, accuracy limit is set to 3% for transmittance oximeters, where in the standard, the limit is 4%. These were found to be the worst performers at the time that the limit was set. There are times when health delivery organizations, clinicians, or consumers may want to go with a device with different performance or features. Technology continues to advance, enabling better performing devices. The standard is updated to reflect these developments. Let's look into the standard more deeply. The primary measure of performance of a pulse oximeter is defined to be accuracy. The standard defines accuracy as the root mean squared difference between the estimated value of saturation, as measured by the pulse oximeter, and the reference value as measured by a well-performing co-oximeter. This metric, ARMS, is presented as a single statistic to communicate the average bias and precision across the operating range 70 to 100. Additional subranges can be specified with a minimum width of 15% to prevent any false indication of performance. Accuracy at higher ranges is typically much better and should not be used to represent performance at lower saturation values. An alternative currently being proposed is to add disclosure for the bias and precision for each decade. The downside is this is more information to comprehend and understand. The standard requires disclosure of performance curves, Bland-Altman plots, to communicate the actual performance over the operating range. The standard has focused on conveying clear and clinically useful information regarding pulse oximeter performance. The fixed declared range is one of many constraints designed to provide a standardized reporting characteristic that enables comparison across devices and supports a determination of substantial equivalency. An oximeter must achieve an ARMS less than 4% to be in compliance with the standard. The standard recognizes that there is an inherent uncertainty in the estimate of SaO2 by pulse oximeters, some of which you're hearing about today. These contribute to the calculated ARMS. The standard defines the formula. Note that it looks like a standard deviation, and in fact it behaves like one. As a result, if we knew the true value of saturation, and of course we do not without an arterial stick, then there's an expected range of values estimated by the pulse oximeter. For example, if the measured value from a co-oximeter is 92%, a pulse ox could be expected to read below 86 or above 98 in 5 of 100 patients. Given the number of times oximeters are used per day, a significant number of estimates from a properly operating pulse oximeter can be expected to appear to be inaccurate. How does one collect the data from a measurement that has a significant variability? The standard defines a protocol for controlling the variability in collection of verification data. It relies on healthy subjects that can safely tolerate repeated desaturations, can tolerate an arterial blood draw, can follow instructions to perform the maneuvers, are capable of providing informed consent, and be physically located in a laboratory with the necessary testing equipment. Methods require a stable desaturation plateau to perform simultaneous measurements of SpO2 and SaO2. This is in contrast to the convenience samples that are collected during real-world studies. The use of laboratory desaturation studies enables stable measurement conditions for simultaneous samples to be obtained. The goal is to minimize variability in testing so that device performance can be compared and a maximum performance can be communicated to the clinician. It is known that real-world performance is likely to be degraded when conditions deviate from the idealized testing environment in the lab. The protocol recommends parameters to be included in the study. The standard recommends including ranges of age, gender, finger size, and skin color, and requires the population demographics used to verify the performance of devices be disclosed. In the US, these inclusion criteria are narrowed by recommending at least 10 subjects with a minimum of 2 or 15% with darkly pigmented skin. Additional factors affect performance. The standard goes to great lengths to document external factors that can adversely affect pulse oximetry accuracy. And these are detailed in the standard for consideration during pulse oximetry laboratory testing. These factors include ambient light, low motion, low perfusion, electromagnetic interference, certain dyes, and dysfunctional hemoglobins. Device fidelity is another issue that needs to be understood in order to collect high-quality data. Device and user settings and algorithmic complexity can influence the fidelity of the estimate of the depth of the desaturation and duration of the event. The pictogram shows that the nadir of saturation is underestimated and the onset is delayed. The causes of these errors may be inherent in the device design, in user settings, and from external factors, such as inflation of a blood pressure cuff upstream from the oximeter probe on the same arm. Capturing context, for example, sensor location and bandage pressure, may be challenging in real-world studies. The panel is asked to consider the influence of labeling with respect to pigmentation and lay use. The standard recognizes that there is increasing use by laypersons and is trying to standardize language directed at this population regarding best practices for using and interpreting the data from oximeters. The committee awaits the panel's deliberation regarding pigmentation and lay users as input to updating requirements in the standard. In conclusion, the standard provides definitions and requirements that address hazardous situations found in pulse oximeters and establishes test methods, acceptance criteria, and rationale to verify risk control measures are in place and effective and acceptable. The standard harmonizes with FDA's guidance document to support the regulation of pulse oximeters and assure reasonable safety. Thank you all, and especially distinguished panel members.
Good morning. My name is Allison O'Neill, and I'm the Safety Signal Coordinator for OHT1 in CDRH. I will be presenting a systematic literature review of the real-world performance of pulse oximeters. First, I will begin by explaining a bit about the history of the safety signal and the purpose of the lit review. The scientific community has been aware for some time that certain patient factors may impact the accuracy of pulse oximeters, and one of these factors may include skin pigmentation. In 2013, FDA published a guidance document which makes recommendations for pulse ox manufacturers, specifically that each pre-market study should have subjects with a range of skin pigmentations. Between 2013 and 2020, there was some limited literature on this subject. In 2020, the COVID-19 pandemic brought increased awareness of the importance of pulse oximetry both in the hospital and home settings. In December 2020, Shoding et al. Published an article which reported that Black ICU patients had approximately 3 times the rate of occult hypoxemia compared to white patients. This article concluded, quote, that reliance on pulse oximetry to triage patients and adjust supplemental oxygen levels may place Black patients at increased risk for hypoxemia, end quote. The article received media attention and spurred additional interest in related research studies. The FDA wanted to provide the public with information on the interpretation and limitations of pulse oximetry. In February 2021, the FDA released a safety communication with recommendations for providers and patients. At that time, the FDA committed to the continued evaluation of the safety and effectiveness of pulse oximeters, including a review of relevant literature. Today, I will briefly present a high-level overview of our systematic review of relevant literature published since 2013. The panel will be asked to incorporate this information in the discussion of the clinical evidence from the scientific literature about the accuracy of pulse oximetry among patients with darker skin pigmentation. We conducted a search of the publicly available PubMed database to identify published articles relevant to the real-world performance of pulse oximeters as it relates to skin pigmentation. 162 publications were identified from the initial search, and 6 additional publications were identified by cross-reference and additional searching after the initial search date. These 168 publications were reviewed for possible inclusion. We excluded articles published before the 2013 FDA guidance document, which outlined more stringent recommendations for accuracy. We also excluded publications that were not relevant to the topic or did not include any clinical data, such as non-systematic reviews and editorials. After exclusions, 28 publications were selected for inclusion in the review. I will now briefly describe the overall body of literature, but in the interest of time, I will not cover each individual study. For more detail, please see Appendix A of the executive summary. Of 28 selected articles, there were 3 systematic literature reviews and 3 lab studies. There were also 22 publications that used real-world data from hospital patients, usually inpatients from the ICU or surgical unit. Of the 22, 7 were considered cross-sectional studies that prospectively collected data, and 15 were considered retrospective studies that relied on electronic health record data. Most studies were conducted using U.S. patients, although there were some from Europe, Asia, Africa, and Australia. The retrospective studies all relied on self-reported race and/or ethnicity, whereas 5 of the cross-sectional studies were able to prospectively collect pigmentation data via either the Fitzpatrick scale or, in one case, the Munsell system. Many used the endpoint of bias between paired measurements, or occult hypoxemia with varying definitions. Others used an endpoint such as less than or equal to 94% SpO2 to qualify for treatment. These variable definitions are listed by study in the appendix. 3 systematic reviews pertaining to the review topic were published in 2022. There was some overlap in the articles that were included in each review. 2 of the 3 reviews concluded that there were overestimations in people with darker skin pigmentation based on the evidence reviewed. The systematic review by Cabanas et al. Found that 8 of 11 studies at low risk for bias found inaccuracies due to skin pigmentation. The meta-analysis by Shi et al. Reported about a 1% bias in both people with high level of skin pigmentation and people described as Black or African American, based on moderate to low certainty evidence. The 3rd review was focused on poor perfusion, but it did also comment on skin pigmentation. The authors noted that only 1 of 22 studies in their review controlled for skin pigmentation, and none strictly followed FDA recommendations for including at least 15% subjects with dark skin pigmentation. Systematic reviews typically represent a higher level of evidence than one study alone. Of course, it should be noted that lit reviews inherently have the same limitations as the studies that are included in the review. Also, all three of these reviews contain articles going back to the 1970s and 80s, which is a larger time frame than considered for the FDA's review, which included articles published since 2013. Regarding the 7 cross-sectional studies, each study reported either the bias between SpO2 and SaO2 or an odds ratio for occult hypoxemia. 5 of 7 papers reported a positive bias or statistically significant odds ratio for subjects with darker skin pigmentation or African American race compared to the reference group. The largest study, Henry 2022, collected approximately 130,000 simultaneous paired measurements of SaO2 and SpO2 in more than 26,000 ICU or surgical patients at 3 medical centers in the U.S., and they reported that Black patients were 1.65 times more likely to experience occult hypoxemia compared to white patients. The odds ratios for Asian or American Indian patients were not statistically significant. Additionally, occult hypoxemia was significantly associated with in-hospital mortality. Of note, one study also reported that Black race was also associated with hyperoxemia. The details of the cross-sectional studies that were identified by the review are listed in Table 12-1 in the executive summary. The 15 retrospective studies were more varied in design and chosen endpoints. Overall, most studies reported a statistically significant association between Black race and either occult hypoxemia or SpO2 bias. Additionally, some individual studies also reported an association with certain clinical endpoints, including treatment probability, organ dysfunction, and in-hospital mortality. It's important to note that real-world retrospective studies have certain limitations and nuanced differences in study design. I will discuss this more in depth on an upcoming slide. The largest study was published by Wong et al. In 2022 with electronic health record data for more than 79,000 patients admitted to the ICU. Paired measurements occurred within 5 minutes of each other. This paper reported a higher incidence of occult hypoxemia in Black and Hispanic patients compared to white. Occult hypoxemia was associated with greater organ dysfunction and higher in-hospital mortality. Please refer to Table 12-2 in the executive summary for a table of results by study. You will hear presentations later today by some of the authors of these studies, including Drs. Schoding, Wong, Fawzi, and Gottlieb. The Lit Search identified 3 laboratory studies using healthy volunteers. The first was a desaturation study performed in Korea to compare pulse ox sensors with or without optical crosstalk. The authors reported a measurement error in African American subjects compared to Caucasian or Asian in the sensor that did not prevent crosstalk. The second was a case series performed in the U.S. designed to compare photoacoustic imaging versus a pulse oximeter. They reported no significant differences in SpO2 by skin type. The third study was a desaturation study performed in the U.S. with 491 subjects classified as dark, medium, or light skin pigmentation. There was a small positive bias in the dark pigmentation group. For more details on each of the lab studies, please see Table 12-3 at the end of the executive summary. Now I will discuss the important limitations that should be considered when assessing the published literature and especially real-world data. First, study variables were defined differently by study. There was no standardized cutoff for what SpO2 and SaO2 levels merit classification as occult hypoxemia. This may be defined differently by site based on clinical need. Skin pigmentation data may be captured by different tools, although race/ethnicity was often used as a proxy. Secondly, pulse ox accuracy often appears worse in real-world studies than in desaturation studies. Using real-world data means that the paired measurements of SaO2 and SpO2 are usually not simultaneous, which may lead to larger disagreements due to normal fluctuations in SaO2 and treatment effects on SaO2. Also, researchers must often rely on self-reported race ethnicity, as skin pigmentation data such as Fitzpatrick scores are typically not collected for hospital patients. And there may be some residual confounding from variables not captured. Thirdly, the real-world population is more heterogeneous than a controlled lab study, typically including very sick patients from an ICU population rather than healthy volunteers. The prevalence of hypoxemia may vary between such groups, impacting the positive and negative predictive values and confounding comparisons of the occult hypoxemia rate. Fourth, heterogeneity of technology used. Pulse ox accuracy will most likely vary depending on which device or product is used, and this data was not provided in every article. It can be assumed that brands, models, and the use of reprocessed sensors varied across hospitals and even within the same hospital. Additionally, technology has advanced over time, and thus pulse ox accuracy may have changed over time. Finally, there is often inherent publication bias in any literature review, which means that results showing statistically significant associations are more likely to be submitted and accepted for publication compared to results that do not show significant associations. Overall, despite the limitations noted, there appears to be mounting real-world evidence from studies capturing tens of thousands of patients. The pulse oximeter accuracy may vary by self-reported race and/or skin pigmentation. However, there's still a need for prospective studies that utilize standardized measurement of skin pigmentation, capture simultaneous measurement of SaO2 and SpO2 paired data, and systematically collect data on important confounders in order to have more robust evidence about the impact of skin pigmentation on real-world pulse oximetry. This has been a high-level overview of the recent relevant literature. Later today, there will be presentations from a number of the authors of real-world evidence studies relevant to the topic. At the end of the meeting, the panel will be asked to discuss the questions shown on the slide. Please discuss the clinical evidence from the scientific literature about the accuracy of pulse oximetry among patients with darker skin pigmentation. In your deliberations, consider the strengths and limitations of the studies, including study design, outcome definitions, and potential confounding factors that can impact interpretation of the evidence. Specifically, please address the following. A. Does the currently available clinical evidence demonstrate disparate performance in patients with darker skin pigmentation? If so, do you believe such disparate performance may lead to increased risks? Please include prescription use and over-the-counter pulse oximeters when used for medical purposes in your deliberations. B. Do you believe the reported disparate performance or increased risks may be explained by factors other than darker skin pigmentation, such as perfusion index or motion artifacts?
Hello, my name is Mary Zhang, and I'm an epidemiologist in the Office of Clinical Evidence and Analysis within CDRH. Today, I will be presenting an overview of the desaturation studies used in cleared 510(k) submissions for pulse oximeters. Given concerns about the potential impact of skin pigmentation on pulse oximeter accuracy, the FDA wanted to better understand how skin pigmentation information is captured and reported in cleared 510(k) submissions. Thus, a study was conducted to provide an initial examination of skin pigmentation information in cleared 510(k)s for prescription use pulse oximeters. The study objectives were first, to examine pre-market clinical study characteristics and reporting of skin pigmentation classification, and second, to assess reporting of factors in device labeling that may potentially impact device accuracy, such as skin pigmentation. On this slide, the process used to ascertain the final sample of examined 510(k) submissions is illustrated in the flowchart. Out of a pool of 420 510(k) submissions received between January 1st of 2000 and December 31st of 2020, an approximate 10% sample, or 44 submissions, with equal numbers of pre- and post-guidance submissions, was identified through an iterative process of random selection and review for eligibility by subject matter experts. This 10% was selected to provide an initial examination of 510(k) submissions with the post hoc assessment to ensure representation from manufacturers with the largest market share. The 22 pre-guidance submissions represented 7% of all pre-guidance 510Ks in the original pool and 44% of pre-guidance submissions, while the 22 post-guidance submissions represented 20% of post-guidance 510Ks and 65% of sampled post-guidance submissions. Submissions were excluded if clinical data were not required, the device was not a pulse oximeter, the device was for pediatric use only, or the data was not relevant. Pulse oximeters that did not require clinical data included 510Ks with well-defined manufacturer modifications of their own marketed devices, in other words, special 510K submissions, and submissions leveraging predicate device data for modifications that did not affect the oximetry technology or performance, an example of which would be addition of respiratory rate software. Next are the data elements that were extracted for the 510K assessments. For Objective 1, examining the clinical desaturation studies, extracted data elements are listed to the right, including the number of subjects, number of paired oxygen measures per subject, availability of patient line level data, inclusion criteria, reporting of participant characteristics, use of Bland-Altman plots for analysis, and skin pigmentation assessment and categories. For Objective 2, device labeling for submissions were reviewed to extract listed factors that were indicated as potentially impacting device accuracy, such as skin pigmentation. Descriptive analysis was performed on the data elements to examine their distributions overall and also by pre- and post-guidance. Key differences will be highlighted in the subsequent slides. The overall clinical study sample sizes found in the cleared 510K submissions are presented in the table. Desaturation studies in the pre- and post-guidance submissions had a median of 12 study subjects ranging from 6 to 33, and a median of 24 paired SaO2 and SpO2 measurements ranging from 13 to 35 per subject. The 2013 FDA guidance recommended providing a line listing of data. As shown in the figure to the right, reporting of patient line level data increased from 54.5% of pre-guidance submissions to 95.5% of post-guidance per the 2013 guidance recommendations. To characterize the clinical study population, common inclusion criteria for all the sample desaturation studies are shown in the left-hand figure. All pre- and post-guidance desaturation studies listed being healthy as an inclusion criterion for subject participation. 43.2% of studies specified in their inclusion criteria that participants must be non-smokers, and 50% indicated that participants must not have specific medical conditions for eligibility, such as hypertension, cardiovascular disease, and respiratory disease. The second figure shows the percentage of sampled studies reporting various participant characteristics. In general, demographics were commonly reported, with age, sex, and race ethnicity being provided in more than 72% of studies, while nationality and biometric measurements, specifically weight, height, and finger size, were reported in less than 28%. Regarding analyses, a large increase was observed, as shown in the figure to the far right, in the number of submissions including Bland-Altman plots per the 2013 guidance. Specifically, 36.4% of pre-guidance and 100% of post-guidance submissions used Bland-Altman plots. Clinical studies used inconsistent methods for assessing and categorizing skin pigmentation, with many of these classifications lacking clear and standardized definitions. As shown in the table, categorization of skin pigmentation varied, including singular assessments indicating only subjects with dark skin pigmentation, use of 2 to 5 skin pigmentation categories ranging from light to dark, and application of scales, specifically the von Luschen and Fitzpatrick scales. Although the skin pigmentation categories remain heterogeneous and poorly defined post-guidance, skin pigmentation reporting post-guidance improved, with 95.5% of submissions specifying skin pigmentation categories as compared to 50% of pre-guidance. Shifting our focus to device labeling for Objective 2, this slide illustrates factors listed in the labeling by the sponsor that may impact device accuracy. The top 3 listed factors across all examined labels were use of intravascular dyes, excessive light, and the presence of electromagnetic or electrosurgical sources. The 2013 FDA guidance indicates that labeling should include all applicable safety information, warnings, cautions, and notes. Subsequently, Inclusion of the factors in blue shading increased in the post-guidance as compared to the pre-guidance submissions. When examining inclusion of skin pigmentation, 18.2% of device labeling for all submissions included dark skin pigmentation as a potential factor that may impact device accuracy. The percentage of labels with dark skin pigmentation increased from 9% among pre-guidance labels to 27% of post-guidance. In summary, for Objective 1, information on pulse oximeter clinical studies that was recommended in FDA guidance were provided in a larger proportion of post-guidance submissions, including greater specification of skin pigmentation classifications, availability of patient line-level data, and use of Bland-Altman plots. In addition, a wide variety of skin pigmentation categories was observed, many of which did not include clear or standardized definitions. For Objective 2, examining device labeling, increases in the reporting of factors that may impact accuracy were observed from pre to post guidance, including dark skin pigmentation. However, the potential impact of dark skin pigmentation on device accuracy was not provided in 73% of post-guidance 510K submissions. This finding identifies a potential area for consideration regarding required contents for device labeling. The panel will later be asked to deliberate and make recommendations on standardizing, categorizing, and reporting skin pigmentation, as well as potential modifications and recommendations to labeling for prescription use pulse oximeters to better communicate their limitations to clinicians and patients. Thank you, everyone, for your time and attention.
Good morning. I'm Kumadini Hendricks, anesthesiologist and medical officer at CDRH. My presentation is on desaturation studies required for market pulse oximetry submission. I will discuss when pre-market desaturation studies are required, how they are expected to be conducted, data submissions for FDA review, and additional convenient sample verification for neonatal use. Finally, I will discuss limitations of pre-market desaturation study. FDA guidance recommends pre-market desaturation testing to be conducted per ISO 80601-2-61. As per the 2013 FDA pulse oximeters pre-market notification submissions guidance, in vivo pre-market desaturation testing for SpO2 accuracy is recommended under laboratory conditions for all new pulse oximeters, as well as for all prior cleared pulse oximeters with significant electro-optical sensor modifications and/or SpO2 algorithm modifications. The general purpose of in vivo pre-market desaturation studies is to verify the SpO2 accuracy of pulse oximeter device performance in comparison to the gold standard measurements of blood SaO2 by a co-oximeter over the specified SaO2 range of 70 to 100%. Therefore, typical labeling is for general indication for non-invasive measurement of blood oxygen saturation. If a manufacturer wishes to seek a specific clinical indication for use of a pulse oximeter, for example, to screen for or diagnose a disease or condition, then additional clinical safety and effectiveness data is requested to be submitted. Per Annex EE of the referenced ISO standards, the pre-market desaturation study should include sufficient number of subjects to attain statistical significance necessary to demonstrate a specified SpO2 accuracy. A minimum of 200 pooled data pairs, that's SpO2 and SaO2, distributed evenly over the tested range of 70 to 100% from a minimum of 10 healthy volunteers is recommended. As per FDA guidance, the study should have subjects that range in age, gender, and skin pigmentation. FDA guidance recommends at least 2 subjects or 15% of subjects, whichever is greater, to be darkly pigmented. Per ISO standards, carboxyhemoglobin should be less than 3%, methemoglobin be less than 2%, and total hemoglobin greater than 10 grams per deciliter. Per ISO standards, allowable testing conditions for pre-market desaturation studies include application of warming techniques to improve circulation and pulse amplitude at the pulse oximeter probe site, covering of pulse oximeter probes with opaque material to prevent optical interference, and the addition of carbon dioxide to inspired gas mixture to maintain normal carbia and to prevent respiratory alkalosis secondary to hyperventilation caused by hypoxia. A catheter is placed within the artery on all test subjects prior to desaturation. A cleared pulse oximeter is placed as reference to detect stable plateaus. The fraction of inspired oxygen is varied in a stepwise manner so to achieve a series of targeted steady-state saturation periods. When the reference pulse oximetry stabilizes for 30 seconds or more, arterial blood is sampled for comparison of simultaneous data pairs of pre-market device SpO2 as well as SaO2. Per FDA guidance, all the following data as it pertains to pre-market desaturation testing is requested to be submitted for FDA review. Pertinent test apparatus used, inclusion-exclusion criteria, number of samples taken per subject, specific conditions of testing such as motion, low pulse amplitude, laboratory conditions, type and frequency of motion testing if applicable, criteria and methods for determining stability of reference arterial blood oxygen saturation at the pulse oximeter sensor site, desaturation profile including target plateaus and ranges, as well as formulae used for determination of root mean square difference, that is ARMS. Additionally, individual data pairs as well as pooled data pairs are requested to be plotted in a modified Bland-Altman plot to show arterial saturation, that is SaO2, on the x-axis as it is considered ground truth. These plots, as on the right side of the slide, have SaO2 on the x-axis and the difference between SpO2 and SaO2 on the y-axis. FDA guidance also requests population mean bias, that is mu0, between subject variance, that is sigma mu squared, within subject variance, that is sigma squared, as well as upper 95% and lower 95% limits of agreement. FDA asks for discussion and rationale for any points excluded from the analysis. As per FDA guidance, all noticeable outliers are requested to have a discussion of the state of health, subject characteristics, test setup, test procedure, and any other factors that may have affected these data points. Also, a discussion of how the outliers do not raise safety and performance concerns regarding the accuracy of the device is requested as well. Devices intended for use with neonates are requested to conduct pre-market desaturation testing with neonatal sensors on adult subjects. Again, any new or significantly modified sensor intended for use in neonates are recommended with such pre-market desaturation testing on adult subjects. Additionally, convenience arterial blood gas sampling drawn in neonates for clinical decision-making in the real world is requested to verify form, fit, and function. The pulse oximeter probe is placed in the same circulatory stream as the sampled artery per ISO standards. FDA requests justification of sample size, subjects, and analysis technique. Pre-market desaturation studies have limitations that may impact real-world performance. Subjects are healthy and selected from a pool of limited volunteers. Sample size is typically not large, limited to 10 to 20 subjects. Skin pigmentation is generally qualified subjectively as light, medium, or dark, and not specific to any particular anatomical site, such as dorsum of hand, forehead, or ventral aspect of forearm. Additionally, currently pediatric indications are verified only with desaturation studies on smaller adult volunteers. Form, fit, and function remain unverified in non-neonatal pediatric populations. Conditions are ideal and remain quite different from actual clinical use. Hands are warmed up prior to pre-market desaturation studies for optimal performance. Sensors are shielded to avoid electromagnetic interference. Data pairs, that's SaO2 and SpO2, are obtained simultaneously after a stable plateau of 30 seconds or more, verified by another cleared reference pulse oximeter. Subjects undergo normocarbic hypoxic conditions and therefore experience less adverse effects from respiratory alkalosis. Subjects are desaturated down to tolerable limits only, and thus lower deciles, like 70 to 80%, may be acquired from a few individuals who can tolerate lower inspired oxygen levels. Accuracy of pulse oximeter is derived from pooled data pairs from all subjects within the entire tested range, typically 70 to 100%. Currently, clinically relevant thresholds such as 94%, 88%, or ranges such as 86 to 94% are not analyzed further. Importantly, the study is not powered to determine significant difference between cohorts, such as age, sex, or specific pigmentation values. Given the limitations and the purpose of pre-market desaturation testing, it is important to consider the following questions. Number one, currently, pulse oximeters are not diagnostic medical devices. Instead, they are verified to be estimates of SaO2 within 2 ARMS for 95% of the data pairs. In this function, they are tools, and the practice of interpretation and verification of SpO2 is relegated to the clinician or the learned intermediary. Is the current indication adequate for clinical decision making? Are there clinically relevant ranges or thresholds where a greater degree of certainty is required? And what is the needed accuracy for these critical values and ranges? Number two, can current pre-market desaturation studies be improved upon to provide clinically relevant pulse oximeter performance for all populations in the clinical setting? Thank you for your attention.
Good morning. I'm Gene Pinello, a mathematical statistician at CDRH. My presentation is on statistical considerations in the evaluation of pulse oximeters. I will first discuss performance metrics. For pulse oximeter studies, common performance metrics include mean bias, the mean of the difference between the pulse oximeter measurement SpO2 and the reference standard measurement SaO2 of arterial oxygen saturation. Precision, the standard deviation of the difference, SpO2 minus SaO2. Mean absolute deviation, and accuracy root mean square. These metrics are averages across all paired measurements of SaO2, SpO2 from all subjects. If a metric is not constant across all values of SaO2, for example, if SpO2 bias varies with SaO2 value, then a difference in the metric between two groups could be difficult to interpret if the groups differ in SaO2 distribution. SpO2 bias may be adjusted for SaO2 distribution using analysis of covariance, or ANCOVA. In ANCOVA, SpO2 is regressed on skin color group effect and SaO2. The skin color effects are differences in the SpO2 bias between the skin color groups at the same SaO2 value. ANCOVA facilitates comparing like with like, can be used to adjust for other covariates besides SaO2, for example, age, sex, perfusion index, finger size, etc., and adjust group comparisons for regression to the mean. I would like to elaborate on regression towards the mean. Regression to the mean is a statistical phenomenon that occurs between two random variables when variation occurs between or within the variables. A classic example of regression to the mean is tall parents tending to have shorter children and short parents tending to have taller children, and vice versa. Another classic example is comparing two methods of measurement. For example, a low SaO2 value of oxygen saturation will tend to be accompanied by a higher SpO2 value of oxygen saturation, even when the two methods are unbiased for each other. Regression to the mean in two methods of measurement occurs because of variation between the measurements made by the methods, measurement imprecision, that is variability in the repeated measurements made by a method, or natural biological variation if paired measurements are not simultaneous. Unless the two measurements are perfectly correlated, they will exhibit regression to the mean. To illustrate, a data set in which SpO2 was unbiased for any value of SaO2 was constructed by permuting the labels of SaO2 and SpO2 in the real-world data from Ebmeyer et al. 2018. The quantile-quantile plot on the left lies on the line of identity, indicating that SpO2 is indeed unbiased for all values of SaO2. However, the regression fit of SpO2 on SaO2, the right panel, indicates non-negligible regression to the mean in that low values of SaO2, for example 88%, tends to be accompanied by higher values of SpO2, the expected value of about 90.5%. And conversely, high values of SaO2, for example 99%, tend to be accompanied by lower values of SpO2, expected value of about 98%. I would now like to discuss box plots of SaO2 at each SpO2 value stratified by skin color. In many publications of real-world studies, box plots of the SaO2 median, interquartile range, and outliers by the SpO2 value are shown side by side for each skin color group. In the box plots reported by Shoding et al. 2020, the left panel, for each SpO2 value, the SaO2 values tend to be lower for Black patients than white patients. In the paper, occult hypoxemia was defined as SaO2 less than 88%, when SpO2 is between 92 to 96%. The red zone indicates occult hypoxemia, which occurred more frequently in Black patients than white patients. In contrast, in the controlled desaturation study by Barker and Wilson, the right panel, for every SpO2 value, the difference in the box plot between Black patients and white patients was much smaller than that observed in the Shoding et al. Real-world study, and occult hypoxemia almost never occurred. The protocol was consistent with ISO 80601-2-61, the pulse oximetry standard, which specifies that a third of the SaO2 values will be in each of the decades 70 to 79, 80 to 89, and 90 to 100%. In contrast, in the real world, SaO2 distribution is uncontrolled. Statistically, the distribution of SaO2 given SpO2, in other words, the box plot, depends on the marginal distribution of SaO2. The lower the marginal distribution of SaO2, the lower the distribution of SaO2 given SpO2, all else being equal. The box plot comparisons of two groups do not consider that the marginal distribution of SaO2 could be lower stochastically for one group than another group. If the groups differ in SaO2 marginal distribution, then differences between groups in the box plot may be difficult to interpret. In the real-world study by Ebmeyer, 2018, the SaO2 distribution for Fitzpatrick 5 and 6 subjects is shifted to the left with a longer left tail compared to the SaO2 distributions for Fitzpatrick 3-4 and Fitzpatrick 1-2 patients, suggesting that the box plot comparisons between the groups, which is not shown, may be distorted because they have different SaO2 distributions. If box plot comparisons are desired, then a suggestion is to standardize the groups to the same SaO2 distribution before drawing the box plot. Two methods for doing this are described here, both based on weighting of the observations. In many publications of real-world studies, authors have focused on the diagnostic accuracy of pulse oximetry to detect hypoxemia. Consider the real-world data in Vasoulis et al. 2022 for Black infants and white infants in the NICU. Hypoxemia is defined as SaO2 less than 85%. That's the first column in each table. The pulse oximeter is defined to be test positive for hypoxemia if SpO2 is less than 90%, the first row in each table, and otherwise test negative, the second row. The occult hypoxemia rate is defined as the proportion of test negative subjects with hypoxemia. The numerator and denominator are shown in light blue. The occult hypoxemia rate was numerically greater in Black infants than white infants, 11.8% to 10.2%. However, pulse oximeter sensitivity to detect hypoxemia was numerically slightly greater for Black infants than white infants, 39.7 to 38.2%. And pulse oximeter specificity to detect non-hypoxemia was also numerically slightly greater for Black infants than white infants, 81 to 78%. Note that the prevalence of hypoxemia was greater in Black infants than white infants, 15.3 to 12.5%. On the next slide, I show that hypoxemia prevalence influences the occult hypoxemia rate, which could explain the difference in the latter between the skin color groups. Based on the Vasoulis data, this plot shows that as hypoxemia prevalence increases, so does the occult hypoxemia rate. Thus, a difference between groups in occult hypoxemia rate cannot be interpreted without additional information on the hypoxemia prevalence in each group. Unfortunately, in many real-world studies with a focus on occult hypoxemia, the first row of data in the contingency tables is not reported, precluding the calculation of sensitivity, specificity, and hypoxemia prevalence. In summary, non-randomized comparisons of groups on any metric may be difficult to interpret without adjustment for potential confounders. Non-randomized comparisons of groups on occult hypoxemia rate are difficult to interpret because of confounding by hypoxemia prevalence. And in general, a pulse oximeter study may be difficult to interpret when the paired measurements of SaO2 and SpO2 are not simultaneous, data were excluded from analysis or not reported at all, or limitations exist in study design, conduct, analysis, or reporting. Thank you.
Good morning. I'm Josh Pfeffer, a biomedical research engineer in CDRH. The focus of this presentation is the assessment of skin pigmentation in pulse oximetry studies with subjective and objective methods. Recent clinical studies have provided evidence of racial disparities in pulse oximetry, which have significant implications for public health. While the root cause of any errors correlated with race in pulse oximetry has not been established, many consider skin pigmentation to be the most significant factor. This presentation focuses on issues that the panel will discuss today, including the acquisition, reporting, and standardization of data on skin pigmentation for the evaluation of pulse oximeter performance. Pulse oximeters operate based on the principles of tissue optics. As light enters tissue, it can be scattered by components such as cells and absorbed by constituents such as hemoglobin, melanin, and water. However, substantial amounts of light will be transmitted through the finger or reflected from other tissues due to the fact that these devices operate at red and near-infrared wavelengths, such as 660 and 940 nanometers, where light attenuation is relatively low overall. Epidermal melanin is more highly absorbing than even blood at these wavelengths and can reduce detected reflectance at 660 nanometers by up to 40% per pass through the epidermis. Detected signals are processed to mitigate the effect of melanin absorption, but may not be fully successful. Anatomic variations in melanin are also significant, with sites such as the fingernail and palmar finger containing relatively low levels compared to sites such as the forehead and the skin proximal to the fingernail. This presentation provides a summary of skin pigmentation assessment methods, which we have reviewed, including subjective and objective techniques. Starting with some of the less rigorous subjective approaches that are more commonly used in pulse oximetry studies, there is racial ethnic self-identification, skin color descriptors, the well-known Fitzpatrick approaches, as well as more standard color scales. Then there are more rigorous objective optical methods based on spectroscopy and colorimetry, as well as gold standard validation approaches based on skin biopsy to quantify melanin content. Perhaps the most common approach for retrospective pulse oximetry studies is to implement self-identification of racial ethnic categories. While relatively easy and inexpensive, there are a number of disadvantages to this method. For example, it is subjective and qualitative, providing little direct pigmentation information. Also, many people have mixed ethnicities, and pigmentation levels can vary significantly within an ethnic group and over time. Researchers have also noted that conflation of race with pigmentation may produce misleading results. The use of skin color descriptors is another common approach, and one which has been implemented in many controlled pulse oximetry studies. This method uses terms such as light, medium, and dark with no further definition of the category. Unfortunately, such an approach is highly subjective and qualitative, as it lacks any standardized definitions or scale, and there can be large variations in skin pigmentation within each category. In highly pigmented skin, color differences are often overlooked by visual assessment. Additionally, this type of categorization is not highly repeatable or reproducible given reader variability. One of the most well-known approaches is the Fitzpatrick Skin Phototype Scale. This method was established 35 years ago to assess the propensity of subjects with light skin to incur sunburn based on a questionnaire. This 4-tier scale was later extended to 6 to include subjects with brown and black skin. Over the years, many have adopted this approach to evaluate skin color. Yet no standardized color charts or definitions for each type have been established. The original methodology tends to show weak correlation with pigmentation and produces results that do not account for variations in melanin content due to tanning. Physicians typically assign individuals of color to phototypes 4 through 6 based on ethnicity, which is unreliable. Although skin pigmentation is a continuum, the color-based FSP method uses relatively coarse bins and categories are not well standardized. In studies where FSP was compared directly with more objective methods, correlations were highly variable. One type of subjective approach that may be more effective involves the use of skin color scales. These methods have been more commonly used in regulatory submissions for pulse oximeters than in published studies, and include the Massey-Martin and von Luschen scales, as well as the Munsell color chart. While theoretically standardized, access to well-validated color charts is a common limitation. Also, as with other subjective methods, inter-observer errors can be a significant problem. Recently, there has been increasing interest in implementing objective optical approaches to evaluate skin pigmentation for pulse oximetry. These melanometers, based on visible to near-infrared reflectance, have been studied extensively in the scientific literature. Numerous commercial devices implementing spectroscopy or colorimetry have been marketed in the US and abroad. These devices can provide data on specific tissue sites, such as the palmar finger, which contains very little melanin. Melanometers implement a range of acquisition and processing approaches and generate different metrics, some of which are not well standardized. One common colorimetry output is the individual typology angle, or ITA, which has been correlated to a range of skin colors. Overall, melanometers have shown good inter-device agreement, indicating that they appear to measure the same biological constituent. The final approach I'd like to cover today involves acquisition and analysis of a biopsy of the skin in order to rigorously quantify epidermal melanin content. This often involves optical transmittance or high-performance liquid chromatography. Since this is an invasive method, its role is to provide a gold standard reference for melanin content in the validation of a non-invasive assessment approach. The literature indicates that objective devices provide a relatively high level of correlation with gold standard measurements of melanin. Several recent publications on pulse oximetry have included statements related to the rigor of pigmentation assessment approaches. Authors of retrospective clinical studies have noted that subjective methods represent a limiting factor. They are only a surrogate for true skin pigmentation or melanin content measurements, and they have categories that are too blunt or broad. One recent article even recommended that researchers stop the use of subjective skin tone scales altogether. To summarize the findings of our literature review, subjective methods for evaluating skin pigmentation are more commonly used, inexpensive, and relatively easy to implement. However, they are less accurate and repeatable than objective methods. There are a variety of subjective methods with different levels of capability in differentiating skin tones. Some of these approaches may be sufficient for evaluating large differences in pigmentation. Objective methods represent an emerging alternative with greater scientific rigor and accuracy, including strong correlation to gold standard measurements of epidermal melanin. They may be most useful for site-specific measurements, such as on the palmar finger, but may not be necessary for all types of studies. Objective approaches are typically more expensive to implement and are currently not a standard approach in dermatology. This afternoon, the panel will be asked the following question. There are several tools to assess skin pigmentation, including, but not limited to, colorimetry, spectrophotometry, melanosome volume fraction, and skin color scales. Please provide recommendations for studies evaluating pulse oximeters for the following. A. Standardization of skin pigmentation assessment, and B. Categorization and reporting of skin pigmentation data. Thank you.
Well, just want to commend the FDA for the very thorough job that they did in terms of the literature reviews and the various evaluations and providing the panel with a lot of information, a lot to digest there for all of us. Just to remind the panel, we're going to have at least 2 hours, or at the most 2 hours, to discuss all of this and try and unpack it and answer the FDA's questions later on. But I'm going to open it up now to the panel for questions to our presenters. Let's try not to get into too much of a discussion. The questions should just be geared to informing our discussion, which will come later on this afternoon. And you can do that by raising your hand. And I see Hugh. Sorry, I'm referring to you by your first name because that's all I can see. Hugh, you want to go ahead and with your question, please?
Yeah, no problem. Just real quick, just a question about the desaturation studies, just to be a little more specific. There's a requirement to have at least 10% of participants have a dark pigmentation. Now, those 10% participants, they can be put into any category during the desaturation study. Is that correct? In other words, you could have 2 pigmented study subjects who are in the category that are receiving normal oxia, and you really don't have any data on their desaturation, or is that an incorrect interpretation?
Yeah, hello, my name is Malvina Edelman, and I'm going to ask Dr. Hendricks to address your question, please.
Sure. I'll be happy to answer your question. Thank you so much for your question. So even though let's say there are 10 subjects that are being desaturated, there are many that cannot undergo desaturation to a lower threshold. And so the lower deciles could be a few subjects. When FDA reviews the clinical study, We look at through the lens of gender, as well as age, as well as pigmentation, to see if there's any representation of data pairs there. It's the same number of data pairs, it just could come from a fewer subjects. So typically it's in that range that we have less individuals, but the same number of data pairs.
So just to follow up, so you can require that they have individuals that are highly pigmented, but they may not fall into the category where they're going to desaturate less than 88%.
That's correct.
Okay, so and there's no specific requirement right now by the FDA to have them do that.
What we do is we individually review all the individual blend alternate data pairs, as well as the table, and we inspect it case by case. And we do ask for more information and justification if that's the case.
All right, great, thank you.
Sure.
Dr. Alam, why don't you go ahead with your question, please?
Thank you. My question was regarding the use of various devices for rating the pigmentation. I understand we'll have an opportunity to address FDA specific questions downstream, but I was just trying to understand what FDA was concerned about when they're talking about melanometers and not using a subjective method like the Fitzpatrick rating scale. Are they speaking about what is optimal in the context of a research study, or what would be considered something that would be routinely used for patients to assess whether or not a particular pulse oximeter was right for them, or how to acquire an appropriate device?
So Dr. Elm, this is Malvina Edelman again. We presented this comprehensive review so that all panel members are aware of the different methodologies currently available, some of which are utilized in our clinical studies, which are used for pre-market assessment of pulse oximeters, which fall under regulated medical devices. Now, we would like for you to be aware of all of this information when you utilize it in deliberation of the questions which were specifically going to pose for you in the afternoon.
Okay, thank you.
Dr. Loeb.
I was particularly educated by the statistical presentation. I think that it was excellent. It really points to, in my mind, statistical flaws in many of the retrospective studies. And I just think there were methods presented, statistical methods that will really help tweeze apart potential influence of other confounding factors such as skin color. I guess that's all I have to say at this moment, but I really hope that some of that is taken into consideration in coming up with better metrics than what are used currently.
Thank you, Dr. Loeb. Dr. Connor.
There we are. Yeah, my question, I think it's for Dr. Pinello. I want to make sure I understand part of his presentation. So Gene, you were, describing the way, NOV or ANCOVA is used. And I wondered if it's doing more harm than good here. And like I said, I just want to make sure I understand it. That typically we would use, another explanatory variable like race, or maybe like baseline severity of a disease, when we're comparing, say, a treatment and a control, because some of the variability in the outcome is explained by baseline severity, or maybe, explained by race in some circumstances, things like that. But in a case like this, by putting in race on the right side of that regression, are we actually ignoring or sort of masking the role that race or skin color has in the outcome? Meaning, did we miss, the entirety of public health, I suppose, a global we, did we miss some of this because we allowed race to be on the right side of that equation? And going forward, should we not put it? Because then differences that we see in race will come out that like, oh, there's measurement error here that we were allowing to, be accounted for somewhere else.
Yeah, thanks, Dr. Connor, for the question. So I'm presenting the ANCOVA as just an example of what you could do. The concern, I guess, is that if you just look at the mean bias across all SaO2 values, and the bias varies depending on the SaO2 value, that mean bias comparison that isn't adjusted for anything may be misleading or just, maybe confounded by SaO2. It could be confounded by other factors. So I'm suggesting ANCOVA is one possible analysis.
Okay, yeah, and I was just trying to get at whether allowing race to be on the right side was maybe masking some of that variability that, rightfully belongs to the device if the device isn't, working right across that. Okay, yeah, just trying to understand. Thank you for that.
Thank you. Dr. Hennessy.
Thank you. I have a question for the engineers. It's sort of a theoretical question. I'm wondering whether the potential error that we're seeing in people with dark skin pigmentation is a function of the calibration of the devices, and if it's theoretically possible for a device to both measure skin pigmentation and then use that skin pigmentation reading to calibrate the results of the SpO2.
Perhaps Dr. Lee wants to.
Hi, it's James Lee, Division Director here at OHT1. Dr. Hennessy, thank you. That is one of the ways that a good algorithm, well-trained algorithm, to piece out the data set and refinement in what the information is basically deconvoluting. It is also an issue that we brought up in our presentation, a physics issue, right? So the darker pigments, darker color schemes will absorb more light and disallow more absorption by the other side, by that sensor. So it's a little bit of both. A refinement in the algorithm can certainly improve accuracies. And if you look at the publications that we have, when we clear 510Ks, we do declare different accuracies, but that obviously is both pooled and, as Dr. Hendricks said, we do publish Blant-Altman studies as well regarding kind of like the different subpopulations.
Thank you.
And before we leave it, I believe Dr. Pfeiffer wanted to add something.
Sure. Thank you. Yeah, I think you hit on a couple of interesting points. I'm not quite sure it's really been determined whether any effect of bias due to pigmentation can really be calibrated out through use of more patients with darker skin pigmentation, or whether there's a need for actual change in the instrumentation or design of these devices. But certainly these are things that we're interested in looking into. So there have been a variety of different mechanisms that have been proposed and potential solutions. And I should mention that there are devices like bilirubinometers that are known to be impacted by skin pigmentation, and they actually incorporate additional wavelengths in order to estimate the skin pigmentation level, and then kind of deconvolve the optical signals for that, the effect of that pigmentation. So there's kind of a precedent for that type of approach of adding wavelengths to improve the measurements and robustness to pigmentation.
Great, thanks. This is fascinating.
Thank you. Dr. O'Connor.
So I guess I've got a comment and questions for two different people on the panel. So first, And I don't know if even my questions are necessarily appropriate for this point. I mean, it's pretty clear to me that this problem will require an additional standard that would be the addition of multiple other LEDs, in fact, to deal with a variety of issues. So let me give you an example. African Americans, 1 in 10 of them has G6PD deficiency. And of course, in the hospital, they're often treated with medications that can produce methemoglobinemia with patients who have an undiagnosed G6PD deficiency. And that could explain a lot of what we're seeing in both the paired and retrospective studies in the clinical context. And so that's one where the FDA's expectation that you actually not study people with methemoglobinemia, and frankly, that you not study people with anemia, both of those, I think, have perhaps lent themselves towards some of the problems that we're talking about today. And so my first question, actually for James Lee, is several of the comments suggested that there are problems with quality control in probe production and calibration, and that interestingly, none of the discussion thus far from the FDA and its experts has suggested that there could be a problem with probe performance. That is to say, they're not really on frequency. They're not really at 660 and 990. And then my second question will be for Gina Pinello, which is, and so if you have that kind of variation, how does that degrade your statistical analysis?
Hi, thank you, Dr. Connor. A good quality system, a good manufacturing process is critical factor in device safety. And as we mentioned in our presentation, these are moderate risk devices which we regulate through our 510K program. And that being said, obviously any device manufacturer is required to have a quality system, manufacturing process, QA, and any corrections needed, they need to live up to that part of the CFR. We don't do an inspection prior to clearing a 510K, but certainly inspections and looking at a company's quality system in a very robust way is part of the 510K program. Companies do get inspected, their documentations are gone over, and it is a requirement. When a company perceives something and how they incorporate good QA and good control over the entire device lifecycle. That's one of the reasons why a big factor in us being now TPLC, where we're looking at a life cycle of device from cradle to grave. And so we're taking a holistic approach, and we will certainly take comments today from the panel to heart and evaluate both pre-market and post-market activities. I yield to Dr. Pinello.
Yes, thanks for the question. I guess statistically, if the probe is not on frequency, that might introduce bias. It might introduce measurement imprecision in the pulse oximeter.
And I guess I'm going to ask the regulators one last question, and that is, it seems to me that the best way to evaluate devices would be in use. That is to say, rather than a small number of volunteers who are selected to be healthy and without problems, we should actually go and perhaps do the equivalent of a phase 3 trial where we study them in patients at the bedside with, very deliberately timed simultaneous measurement of a saturation and a blood gas.
I was actually going to make the same point. It's just doing healthy volunteers. It's like a phase 1 study, but there's no phase 2 or phase 3 study in terms of seeing how these devices perform in patients who really need them, where we make these critical decisions. I think in the interest of time, we're going to cut it off after Dr. Hennessy's question, but Dr. Collip, why don't you go ahead with your question, please?
Okay, thank you. That presentations were awesome. I learned a lot. As I mentioned, I'm a sleep specialist, and in sleep medicine, the pulse oximeter is like the core signal for diagnosing most sleep disorder breathing disorders. And I guess my question is, because we rely so heavily on it both now with home sleep testing and even in lab, is there any evaluation or analysis of the ability of pulse oximeters to adjust rapidly with the trends, that can go up and down quite often during the night? Because I didn't hear anything about that in the evaluation of pulse oximeters. Thank you.
Hi, Dr. Culp, I could take that. Certainly, as we acknowledge, the saturation signal in a sleep study is critical, as you mentioned. Pulse oximeters generally are very responsive, and the way we use pulse oximeters in home sleep studies or sleep studies in general is that we measure it epoch by epoch, so that desaturation event coupled with a disordered breathing event and measuring that. And as you might know, the things that we look at when evaluating sleep study devices, either PSG or home sleep studies, is that a trained physician is designed to be the adjudicator and determines the accuracy. So you measure epoch by epoch, event by event, and it should be hand scored by a physician. But any comments today, certainly we will share with all the folks that use pulse oximeters, not just in anesthesia. Thank you.
And I believe Dr. Weinberg is going to add something. Go ahead.
Sandy Weininger, and just a brief comment, the clinicians can certainly set the smoothing rate in most medical grade oximeters, perhaps not in the finger clips, but in the medical grade, you can set it to respond as rapidly or as slowly as you want to smooth out those dips, depending on what your clinical application is.
Thank you. All right, I'll put myself in line, so I'll go next. I have a question and probably a comment. I saw right at the beginning there was a mention of reprocessed oximeters, which made me think that oximeters are really like any other technology, which tend to degrade over time. So is there a half-life on the accuracy of these pulse oxes? Because I'm not sure that they're replaced as often or checked as often as perhaps they should be. I'll let the FDA address that first, and then I might have one other comment after that.
Dr. Lee.
Hi, again, thank you, Dr. Nathan. Certainly, a labeled shelf life is something that we would review as a part of the 510K process when it comes in. So validated cleaning, reprocess instructions would have to show that the device does not deteriorate over time. And so a labeled life of the device is something that we would use in our review.
Okay, thank you. And just a comment for Dr. Pfeiffer with regards to skin pigmentation and evaluation of that. We typically put the pulse ox on the fingertip or sometimes on the forehead if we don't get a good signal on the fingertip. What's the general correlation in terms of pigmentation on the distal aspect of the finger versus other parts of the body? And in terms of evaluating pigmentation, is the global pigmentation taken into account or specific body areas?
I think that's kind of an open question about what's the best way to assess pigmentation. I think there's more of an interest recently in trying to go after the site-specific pigmentation level, and I think we're going to start seeing more studies that do that. And I think that's one of the benefits, certainly, of the objective approaches is that some of these devices are pretty good at providing that information, because as you referred to, I think the pigmentation level on the finger is typically much lower than on other locations like the forehead or ear. So one would expect that there would be more impact from pigmentation on those sites as opposed to the finger.
Dr. Nathan, we would appreciate greatly if the panel addresses this during their deliberation and lets us know if they have any recommendations on this specific topic, as we have pondered it extensively.
Okay, sounds good. We'll do that. I know we're running short of time. We're supposed to break at 10, and there'll be opportunity for other questions. So perhaps we'll just do one or two questions, and then we'll break, and there'll be opportunity probably in the afternoon to address any further concerns. So Dr. Connor, why don't you go ahead?
Yeah, Jason Connor. Yeah, a bit of a follow-up to Dr. Hennessy's. Like Dr. Pfeffer said, that bilirubin devices can measure, skin coloration. I know there's also hair laser removal ones that measure it, because some of those are actually not made to work for dark skin, because they look at skin versus hair and light and dark, and so that in darker skin, it can actually do damage. So the device itself says this is skin too dark that I'm made to work on. So devices are certainly capable of that. My question to FDA is, are any of the pulse oximeters currently on the market, do they attempt to measure skin pigmentation and account for that in their algorithm? Because we saw a lot of general stuff, but not about like specific devices that are approved. So I'm wondering if any actually try to do that these days.
Hi, it's James Lee.
Dr.
Thank you, Dr. Edelman. Not to my knowledge, but I think we'll probably have to follow up if I'm wrong later this afternoon. But generally speaking, their general use, we don't label them for a particular subgroup. They're considered general use tools.
Yeah, no, and it wasn't that I was implying there would be like one for light, medium, or dark skin, but rather would the device itself attempt to say, oh, this is someone with lighter skin, this is someone with darker skin, and maybe the internal algorithm was, slightly different. There's obviously computation going on inside the device. But yeah, I mean, what you're suggesting might be the simplest solution. But I was wondering if any actually measured it and accounted for it internally, not that there would be different ones. But
So not to the best of our knowledge. We will, however, verify and get back to you after lunch with the definitive answer.
If I can make one brief comment to follow up on, you mentioned that there are essentially melanometers that are used with laser therapy devices. And actually, I think those are the only ones that have gone through the FDA clearance process. Most of the melanometers on the market have not. They're not actual medical devices, but the ones that you mentioned, I believe, have.
Okay, great. Thank you.
All right, we'll take two more questions because all the questions are so good and the answers are shedding light on what we're going to have to ponder on. So Dr. Hennessy, your question, and the last one will be Dr. Bickler after that.
Great, thank you. Sean Hennessy, I'm wondering about standardized preclinical testing. So are there any simulation platforms that allow the prediction of the performance under different conditions, including different skin pigmentation patterns? Or is there any standard preclinical testing data that's submitted at all?
Dr. Lee?
If I understood your question, Dr. Hennessy, you were asking basically on the preclinical side, on the bench, how do they validate it? Phantoms certainly could be used as a part of product development, but we don't recommend them. We want the subjects to be studied in the clinical realm. Certainly, we use that for verifying the accuracy of the device. But certainly, companies, many different groups could, as a first cut, do different types of validation, particularly regarding sensitivity to spectra and light.
Thank you.
All right, Dr. Bickler, you'll be the last question of this session. Go ahead.
Okay, thank you. Phil Bickler from UCSF. In 2005, we called for a simple switch or provision on oximeters to correct for skin pigment. In subsequent years, we've realized that that is a very simplistic view of how to solve the problem. The issue of skin perfusion, sort of the pulsatile signal, is, we have actually found to be crucial in amplifying the bias caused by dark skin. I'll talk about that later, but I hope people don't think that fixing the pigment problem is as simple as selecting a button that indicates dark, medium, or light skin pigmentation.
Okay, thank you, Dr. Bickler, for that comment. And yeah, there'll be opportunity to address all of this later on. So great session, great questions, very engaging. We're going to take a 5-minute break now. I have it as 10 after 10, and we'll reconvene at 10:15. We're running a little bit behind, but no worries. There will be opportunity to play catch up a little bit later on. So we'll get together again at 10:15. Thank you, everyone. Good morning again, folks. It is now just after 10:15, and I would like to resume this panel meeting. We'll now proceed with the open public hearing portion of the meeting. Public attendees are given an opportunity to address the panel, excuse me, to present data, information, or views relevant to the meeting agenda. Dr. Awajapi will read the open public hearing disclosure process statement first.
Hello. Once again, this is Dr. Awajapi, the designated federal officer, DFO, for this advisory committee, Anesthesiology and Respiratory Therapy Devices. I will now read the conflict of interest statement regarding the open public hearing, OPH. Both the FDA, the Food and the Drug Administration, and the public believe in the transparency process for information gathering and decision making. To ensure such transparency, at the open public hearing section of the advisory committee meeting, FDA believed that it is important to understand the context of individual presentation. For this reason, FDA encourages you, the open public hearing speakers, at the beginning of your written or oral statement, to advise the committee of any financial relationship that you may have with any of the company or group that may be affected by the topic at this meeting. For example, this financial information may include a company's or group's payment of your travel, lodging, or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationship. If you choose not to address this issue of the financial relationship at the beginning of the statement, it will not preclude you from speaking. Thank you very much. I'll now hand it over back to Dr. Nathan. Thank you once again.
Thank you, Dr. Awajapi. The FDA has received 9 requests. Each speaker will be given 5 minutes to speak, and for the live speakers, just do keep an eye on the watch, please. We will hear 6 pre-recorded folks first in terms of the open public hearing presentations, and this will be followed by 3 live presentations. The first speaker is Veronica Hickson from the Uren Bevan University Health Board. Go ahead, thank you.
I'm Dr. Veronica Hickson, a practicing paediatrician in South Wales and director of the Electrode Company. I want to talk to you about a clinician's understanding. Many clinicians believe the system has the same accuracy level throughout the range of 70 to 100. This isn't the case due to magnification of errors. At lower SpO2 levels, the accuracy is less and it's much better at higher levels. In practice, these values are also impacted by the specification of the wavelengths in the LED in comparison to those used in the clinical trials. High reading bias, as seen here, can also be the case in high melanin. Clinicians want safe and effective medical devices that they can rely on. They want clear information on factors that impact SpO2 accuracy. They want to know the accuracy for the equipment they're using at high and lower levels. The pulse oximeter is used as a vital sign, and they want their equipment to be accurate for this. They want to be able to use pulse oximetry as recommended and advised in clinical to increase morbidity or mortality. It's often thought that pulse oximetry is a general guide. In practice, clinically, it's used with guidelines and protocols to dictate certain actions, and if they're not taken, then it could contribute to an adverse patient outcome. Also, the equipment should be complying with regulations, and if inaccurate, could also contribute to adverse patient outcomes. Pulse oximetry is used in many scenarios, from congenital screening in neonates through the NICU and up to and including COVID assessments and triaging patients. Vulnerable patients exist in various patient groups, including neonates, elderly people, veterans. They all require SpO2 measurements. Many professional groups, from paramedics such as ambulance staff and EMT, the nurses in the ER and on the wards, and the medics and doctors and family physicians, are using the results of pulse oximeters to guide clinical decisions. In some situations, yes, such as the operating theatre, the patient is monitored by a variety of different technologies, and pulse oximetry is one of the contributory information points. Users don't want confusing and misleading information. They want sensors that are fit for purpose and enable the pulse oximeter system to continue to work as intended and claimed. They want it recognised that pulse oximeters are capable of being and are used as diagnostic tools, and inaccuracy can be due to poorly compliant equipment and not that the patients are faulty. They don't want to be told it's statistically acceptable to be inaccurate. They want to maintain the accuracy of the system and not have additional bias introduced. The actual accuracy levels information could be more clearly provided by using discrete ranges. For example, 99 to 90% SpO2 of less than 1.5, 90 to 80% less than 2.5, and 80 to 70% perhaps less than or equal to 3.5. Information is available in manufacturers' previous bring down studies and could be used to inform new standard drafting against which they could then prove their current and continued compliance. The accuracy at extremes of the LED wavelength specification should be assessed and the data included in the files. Good manufacturing practice and quality management can therefore ensure compliance with the accuracy requirements that must be maintained in practice, in production, and not just for the initial regulatory data gathering and accuracy assessment. Additional biases introduced by a sensor of perhaps more than 3% at any SpO2 level should be considered as non-compliance and therefore not on the market or in use. I'm happy to take questions.
Hi, everyone. I'm Sam Ajezian. I'm the Chief Medical Officer for the Patient Monitoring Business at Medtronic. My medical practice was pediatric critical care for over 20 years, and I've used pulse oximetry in the care of my patients throughout my career and am well familiar with both its strengths and limitations. I want to thank our colleagues at the FDA for inviting us to share our perspectives on how to prioritize and improve health equity in pulse oximetry. This is the commitment you'll hear in my remarks today. Clinicians like me have relied on Medtronic's Nellcor pulse oximeters for over 40 years to obtain non-invasive estimates of blood oxygen levels to guide critical interventions. Medtronic stands behind the quality, safety, and performance of our devices, but we acknowledge that more can be done to enhance accuracy and to foster health equity. Specifically, we stand with the FDA, academics, clinicians, and patient advocates in an effort to further improve standards for pulse oximetry devices and reduce health disparities. When questions emerged about pulse oximetry accuracy, we went back and looked at our own data. We did a pooled analysis from previous validation trials with 88 patients and approximately 18,000 patient samples. We presented these data at the recent Evidence-Based Perioperative Medicine World Congress in July of this year. Nellcor pulse oximeters perform well overall, reporting values within 2% of directly measured oxygen levels. This is consistent with FDA requirements. When you dig more deeply, though, we see accuracy was slightly higher for patients with lighter skin pigmentation. While the overall impact on measured blood oxygen levels was not clinically significant, we see room for improvement, including further enhancing accuracy for patients with darker skin pigmentation. Pulse oximeter accuracy can be affected by melanin, a skin pigment that is present in higher levels in those with darker skin. Melanin concentrations, like skin pigment, vary widely within racial and ethnic groups. Melanin absorbs certain types of light and can affect the accuracy of pulse oximeters. Currently, manufacturers rely on a variety of subjective scales to assess the impact of skin pigmentation on device performance. In order to improve device accuracy for everyone, we must replace these subjective scales with a uniform, objective method of assessing skin pigmentation. Now, we do see a path forward. Medtronic is evaluating and sharing data, working with FDA and ISO, and making investments in our own devices and algorithms. At the same time, we believe health equity requires true collaboration across all stakeholders. Specifically, we need to work together to improve information sharing across all key audiences, to foster continued innovation in pulse oximetry, and to improve the methods we use to validate pulse oximeters, including standardization of how we assess skin pigmentation and increased diversity in skin pigments in clinical trials. We also need to work together to educate clinicians and patients around device capabilities, use patterns, and limitations. And we need to collaborate to develop reasonable regulatory standards that allow for innovation while at the same time promote safety, efficacy, and of course, the health equity of medical devices. As I consider our role in improving health equity, generally I think about my own experience caring for children in the pediatric ICU. Health equity was a priority for myself and all of our team members, and I see our mission at Medtronic to be fully aligned with my clinical experience. We know how our technologies affect patients and their physicians, and as such, the greatest commitment we can make is to consistently design and improve devices that positively influence health outcomes for all patients. Health equity is absolutely foundational to our mission at Medtronic and our collective successes. And as a company, we're committed to that goal. We thank you for this opportunity to share our data and our perspectives on this issue.
Hello, and thank you all for the opportunity to speak on this important topic today. My name is David Sturkin. I'm a hospital medicine physician at the University of Wisconsin, and I co-wrote a letter that the Hospital Medicine Reengineering Network sent to the FDA on this issue. I'm here today to briefly describe the recommendations in that letter. Number 1, require subgroup analyses by objective measures of skin pigmentation, race and ethnicity, and gender in the approval process. Currently, the FDA only requires at least 2 darkly pigmented subjects or 15% of your subject pool, whichever is larger. This means that a device that is accurate in the 85% of subjects with light skin pigmentation could still be approved even if it is very inaccurate for the 15% of subjects with dark skin pigmentation. To ensure accuracy for all groups, we believe subgroup analyses should be performed and that subgroups should be large enough to reach statistical and clinical significance for each group. This can be done in conjunction with a recommendation from the pulse oximeter manufacturer Masimo to include equal distributions of subjects with light and dark skin pigmentation. Since the relative contributions to these device inaccuracies from objective skin pigmentation versus from the social construct of race are not yet clear, as further explained in the letter, at this time we believe subgroup analyses should be performed both on the basis of skin pigmentation and separately on the basis of race, as well as on gender, given some evidence for gender bias as well. Number 2, prohibit the use of so-called race correction factors. Historically, when different subgroups of people have been found to have different results, it had been common practice to use so-called race correction factors in clinical calculations to supposedly correct for these differences. For many reasons, however, race correction factors are now considered to be very problematic and are being removed from some calculations, such as estimating glomerular filtration rate. Instead of using race correction factors to attempt to account for pulse oximeters' inaccuracies, the devices themselves need to change. Number 3, require pre-market data from both healthy and hospitalized patients for device approvals. The recent studies showing racial bias were in hospitalized patients. Although some studies have also shown racial bias in laboratory conditions, one manufacturer, Masimo, has recently claimed that their devices do not show racial bias in laboratory conditions. Notably, this claim has not been evaluated by the FDA, and it should be, and also Masimo did not perform similar analyses in hospitalized patients, despite their FDA submission packet indicating that they do have such data from hospitalized patients. Nonetheless, it is possible that racial bias is most prevalent in hospitalized patients. Therefore, pre-market testing should be required in hospitalized patients as well, because testing only in healthy subjects might miss some biases. Number 4, intensify the overall approval requirements for medical grade or prescription pulse oximeters and consider requirements for over-the-counter devices. Currently, the FDA only requires data from 10 subjects for approval of medical grade pulse oximeters. This sample size seems concerningly small for devices that will be used to make life or death decisions for literally millions of patients, and we believe it should be increased. Accuracy requirements could be intensified as well, as Massimo described in their comments. Over-the-counter devices, however, undergo no FDA review at all because they are supposedly not intended for medical purposes. It is quite clear, however, that many, if not most, users of over-the-counter pulse oximeters are in fact using them for medical purposes. So we believe some type of FDA review is now needed for these devices in order to protect consumers. We do, however, believe that the requirements for these devices should be kept separate from those for medical-grade devices so that the latter can have more stringent requirements. Number 5, have these new requirements apply retroactively to previously approved devices. Without applying these requirements to previously approved devices, manufacturers of racially biased pulse oximeters might choose to continue selling their current devices which have been grandfathered in, rather than try to get new devices approved, which would only serve to perpetuate this racial bias. Number 6, investigate other similar devices for the presence of racial bias. Other optical devices such as temporal thermometers have recently also been shown to demonstrate racial bias, likely due to the effects of skin pigmentation on their measurements. We believe the FDA should proactively investigate optical devices for racial biases, rather than waiting until independent research has found the bias for them, as has been the case with pulse oximeters. Lastly, although we recognize that the following 2 recommendations may not fall within the purview of the FDA specifically, we would like to encourage the FDA to advocate for them given the collaboration that often occurs between agencies. Number 7, create additional safeguards to detect and prevent biases in our other diagnostic tools. At the FDA level, this could include a dedicated committee that reviews all future devices for potential biases prior to their approval. Number 8, invest in independent research. The exact causes of the racial biases seen in these devices are not known. Skin pigmentation almost certainly contributes, but there may be other contributors as well, such as the health effects of systemic racism on individuals belonging to marginalized groups. Further research is needed to determine the mechanisms behind these biases and to find potential solutions for them. In particular, prospective studies in hospitalized patients are desperately needed, but would require significant funding, and it will be important for these studies to be independent of industry bias, so government funding is preferable. The Hospital Medicine Reengineering Network would be happy to help coordinate such studies among its more than 50 participating academic institutions. And with that, I would like to thank the FDA for its time, and I'd be happy to answer any questions that you may have.
Hello, everyone. My name is Grace Wickerson, and I am a Science Policy Fellow with the Federation of American Scientists. I'm delivering this statement on behalf of the Federation of American Scientists and our partners at the University of Maryland Medical System. Thank you so much for giving me a time to speak to the committee today. As we all know, pulse oximeters provide a rapid, non-invasive estimate of the body's ability to deliver oxygen to the tissues and are ubiquitous in medicine. This technology is a fantastic innovation and has eliminated the use of more painful measurements of blood oxygenation like the arterial blood gas. This technology relies on the absorption of light through the skin, and as an optical technology, anything that changes the amount of light received by the detector impacts this measurement. There's fingernail polish and tattoos, methemoglobinemia, carbon monoxide exposure, anemia, hypothermia, and the concentration of melanin within the skin of the patient. Collectively, current pulse oximeter models overestimate the oxygen levels in persons of color by an approximately 1 to 3%. This is because many models do not adjust for how high quantities of melanin lead to the absorption of more light within the skin before that light reaches the detector. Now, this bias may be perceived as small in magnitude, but within a statewide medical system, like that of our partners at the University of Maryland Medical System, these become large numbers of people. We estimate that about 1,012 African Americans with apparently normal pulse oximetry readings had hidden or occult hypoxia when they were discharged from the University of Maryland Medical System emergency departments over the last year. While this issue was identified as early as the 1990s, the problem of occult hypoxemia in darker skinned patients has come to the forefront with the COVID-19 pandemic. Oxygen saturation measurements were an important part of the management of patients with COVID-19, and occult hypoxemia was associated with delays in care and poor outcomes in patients with COVID-19. Recent retrospective studies have shown that the greater rates of undetected occult hypoxemia negatively impacted the care of African American patients with COVID-19, leading to those patients being less likely to receive supplemental oxygen on time and being at greater risk of death. The University of Maryland Medical System is doing what it can to address this problem. We are preparing our own investigation into the harms associated with a cold hypoxia in the patients treated in our medical system, and working to correlate patient outcomes with pulse oximeter models contracted at the time of measurement. University of Maryland Medical System is additionally reconsidering how clinicians are trained to consider and apply pulse oximetry readings. These interventions can provide care in the short term, but they do not solve the underlying problems encoded in the technology. We need improved standards for pulse oximeter devices so that clinicians can rely on the technology to provide accurate results across all populations and in the real-world clinical setting. Thus, we feel that action by the Food and Drug Administration is needed to address this pervasive technological bias. Immediate and long-term solutions should include requiring the publication of testing data for current pulse oximeters so that clinicians can make best judgment on which technologies are reliable for use in their patient populations, reconsidering which models of pulse oximeters can be used as predicates for 510(k) approval pathways, and only allowing devices that have been tested on a more diverse range of skin tones. Furthermore, requiring future models be tested in significant proportions of individuals of all skin tone types, in using objective metrics of skin pigmentation that reflect the broad range of skin pigmentation, such as skin tone scales like the Massey scale and the Monk skin tone scale, as well as colorimetric devices. Additionally, the FDA should consider device manufacturers ensure their devices work in critically ill patient populations where there are not stable SpO2 readings, embodying the real-world care environment where pulse oximetry would be used. We also think that there should be set a standard for greater accuracy in over-the-counter and wearable pulse oximeter devices, given the rapid growth of this market during the COVID-19 pandemic. And finally, in collaborating with funding agencies like the NIH and NSF, the FDA should seek to establish and fund research into low bias and bias-free tools and consider the use of accelerated approval pathways for technologies that have been proven to work effectively on diverse populations. With these, we believe we can make tremendous progress towards bias-free devices, and I thank you so much for my time.
I'm Jeff Matthews, Director of the Electrode Company. We specialize in pulse oximetry. Back in the 1980s, we invented adaptive noise cancellation in pulse oximetry. We sold this patent to Massimo. During the 1990s, we reverse engineered most of the OEM systems out there, and then went on to focus on sensor accuracy. Not all replacement sensors are as accurate as the manufacturers claim. There are often mismatches between wavelengths and the calibration data. Melanin can cause high bias in SATS readings. This is because the calibration data for most systems is appropriate for light skin and not for dark skin. Neither of these issues will be fixed until there's a better control of wavelengths during manufacture. During the 1990s, we hardly ever saw an inaccurate sensor. Then CE marking came in. It was as if this was a license to sell third-party sensors regardless of accuracy requirements. They seemed to work okay, so customers bought them unaware of any accuracy issues. OEMs had to drop their prices. Quality control on LED selection suffered. Sensor accuracy dropped. There are even third-party sensors from the Far East on the US market without 510(k) clearance. The number of inaccurate sensors on the market is increasing. There are new OEM sensors that are claimed to be rearward compatible in older systems, but they're not as accurate as claimed in older systems. There is no OEM that claims that every sensor is uniquely calibrated. This is false. Sensor accuracy is not as good as it used to be. Bad things have happened when manufacturers have been confronted with inaccurate sensors. Explanations have been contradictory. Investigations reported to customers do not make sense. Fundamental questions have been dodged. We need to know what the target wavelengths for a sensor are, and we need to know what they really are. This tells us how accurate a sensor is. Manufacturers should already have this information. It makes no sense to try and fix racial bias. Any adjustments will be swamped by manufacturing errors. Regulators have been led to believe that the only way of checking sensor accuracy is by read-down. This is false. Sensor accuracy can be checked with a spectrometer. We should not let commercial interests come before patient safety. We need to wind the clock back a bit and insist that sensors are at least as accurate as they used to be. Regulatory changes must be retrospective.
Good morning. We at MASO have been following with great interest the recent reports on skin pigment-related differences in pulse oximeter accuracy, and we commend the FDA for convening this panel on the subject. As a background, Masimo developed the very first read-through motion and low perfusion pulse oximetry using signal extraction technology, or SET, more than 30 years ago to address known pulse oximeter errors such as motion and low perfusion. SET also addresses skin pigmentation and other non-pulsating absorbers in the same way. We applaud the authors of the recently published studies for investigating this important topic of skin pigment, but we have the following concerns with the methodologies of some of these studies. Number one, most of the studies do not characterize the manufacturer or model of the pulse oximeter. In fact, many of them use several different pulse oximeters in the same data pool. Number two, the SaO2 or blood gas analysis co-oximeter data and the SpO2 pulse oximeter values in these studies are not actually simultaneous. Instead, the readings were often taken more than 10 minutes apart from one another. Number three, dyshemoglobins such as carboxyhemoglobin and methemoglobin were not measured or accounted for. Number four, artifacts such as low perfusion and patient motion are not reported. And finally, number five, severity of illness and other hemoglobinopathies such as sickle cell anemia are not reported or accounted for. Our data demonstrate that Masimo SET pulse oximeters are equally accurate for individuals of all skin tones in our volunteer human subject laboratory. An abstract of these data was presented at the Society for Technology and Anesthesiology, known as STA, annual meeting in January of this year, and the full manuscript is currently in press and will appear in the Journal of Clinical Monitoring and Computing, and I expect it to be published online literally any day now. Details of these published results were also submitted with our written comments for this meeting. As I said earlier, we began to address confounders such as low perfusion, motion artifact, and skin pigmentation decades ago when we first invented Masimo SET technology. We have also calibrated and validated our oximeters using almost equal numbers of dark-skinned and light-skinned individual volunteers. We support prospective clinical studies, patient studies, on this topic, and we are pursuing these now. But bear in mind, controlled desaturation studies with SpO2 values going down to 70% can be ethically done only in a laboratory using healthy volunteers, certainly not in patients. In recent years, Masimo has continued to improve our pulse oximeter accuracy, and our latest performance improvement coming in 2018 demonstrated an overall uncertainty of plus or minus 1.5% for our RD SET sensors, compared with the usual industry values reported as either 2 or even 3% uncertainty. In our recommendations to the FDA on the skin pigmentation issue, Masimo supports raising the standard on requirements for the percentage of dark-skinned subjects used in calibration and validation studies. We also believe it is important that the FDA regulates and applies similar oversight recommendations on all pulse oximeters, including those sold directly to consumers. The full summary of Massimo's recommendations to this FDA panel have been submitted as comments in the docket, and I thank you very much for your attention.
Thanks to our folks or our speakers for the pre-recordings. We now have 3 live presentations. As a reminder to our presenters, it's 5 minutes each as a maximum. And let's start off with Dr. Callender, please.
Good morning. Thank you for the opportunity to speak today on behalf of the National Center for Health Research. I am Dr. Elena Callender, an OBGYN with a Master's in Public Health, and I'm a senior fellow at a nonprofit think tank. Our center conducts, analyzes, and scrutinizes research on a range of health issues. We do not accept any funding from companies that make products that are the subject of our work, so we have no conflicts of interest. Thank you for convening this important meeting to discuss the critical issue of how skin pigmentation impacts the accuracy of pulse oximetry. The pulse oximeter is ubiquitous in medical care in the United States. Like blood pressure and respiratory rate, pulse ox is a vital sign used to make critical medical decisions. The accuracy of this device on all patients is of paramount importance. Although skin pigmentation was thoughtfully considered when this technology was first invented, pulse oximeters in use today are dramatically different from the original gold standard device. In early studies, researchers considered many patient-specific variables, including skin pigmentation, that would contribute to pulse oximeter measurements. To accommodate these differences, the original device was standardized for each patient and provided accurate measurements for every individual. Furthermore, early pulse oximeters use 8 wavelengths of light, which was considered enough to account for individual patient variations, but today's devices use only 2. There is now strong evidence that commonly used pulse oximeters are less reliable for patients with pigmented skin. The inaccuracies related to skin pigmentation vary based on the device used. While one device may be inaccurate only for pulse ox measurements under 90%, others may have significant inaccuracies at all ranges. In general, inaccuracies related to skin pigmentation increase as the level of oxygenation decreases. Clinically, this means sicker patients are less likely to get an accurate reading and are therefore less likely to get appropriate care. The FDA has designed a study to evaluate pulse oximeter performance in hospitalized adult patients of different skin pigmentation levels. This is an important step, but we urge the FDA to do more. Currently, the agency only requires pre-market testing to include at least 2 darkly pigmented individuals or 15% of the subject pool. This is woefully inadequate. We ask you to urge the FDA to require that manufacturers include individuals with a broad range of skin pigmentations in their studies. Moreover, subjective measurements of skin pigmentation are of little value because there is such wide variation in hues and so many contributing factors. Therefore, only objective tools for assessment of skin pigmentation should be used in studies of how it affects pulse oximetry measurements. The FDA's current guidelines do not require product labeling to address the impact of skin pigmentation on pulse ox measurements. While labeling is important to alert users to possible inaccuracies, it does not diminish the importance of developing devices that are effective for everyone. Medical devices must work effectively in all people, regardless of skin pigmentation. Products that are allowed on the market should not be accurate for only some races and ethnicities. The FDA should require more scrutiny to minimize bias in medical devices so they are accurate and reliable for everyone. Thank you.
Thank you very much for your comments, Dr. Callender. Let's move on to Dr. Abrams, please.
Good morning. Can you hear me okay? Sorry, can you hear me all right, please? Thank you. Hi, everyone. I'm Dr. Michael Abrams, Senior Health Researcher from Public Citizens Health Research Group. I have no financial conflicts of interest on this matter today. Public Citizens Health Research Group has a 50-year history of monitoring activities of the FDA, especially pertaining to the safety and effectiveness of medications in Class 2-3 medical devices. We believe this advisory committee topic, pulse oximeter accuracy given natural human variations in skin pigmentation levels, is one that exemplifies two ongoing challenges the FDA faces as the principal gatekeeper regarding the safety and effectiveness of medical devices. The first of these challenges is, of course, the issue of racial equity. The second, and one that I will emphasize in my remarks, are deficiencies of the 510K pathway for clearing medical devices. Concerning racial equity, we want to just briefly remind the committee and the FDA that lack of diversity in FDA-regulated clinical trials has been a longstanding problem that can lead to inferior outcomes for patients of color. The fact that, for example, as we've heard, persons of color have not received proper care related to COVID-19 infections because of inaccurate pulse oximeters is very simply unacceptable. Regarding the 510K process, we have long been concerned that evidentiary standards for safety and effectiveness are too lax for many medical devices, thus giving manufacturers a reckless fast-track pathway to marketing. In the case of pulse oximeters, we know that since the 1980s, hundreds of such devices or their components have been cleared for marketing. In its briefing materials, the FDA reviewed a subsample from a total of 420 510K applications and found that clinical data was not required for approximately 35% of such submissions between the years of 2000 and 2020. FDA further reported that the measurement of skin pigmentation level was methodologically limited, as we've heard, and variable across such device applications. Since at least 1991, researchers have observed that increasing levels of skin pigmentation correlate with overestimates by pulse oximeters of arterial oxygen saturation. The FDA's briefing packet for this meeting tabulates at least 15 such studies just since in 22, in fact, with thousands of subjects, including research that suggests treatments are withheld because of this apparent medical device error. Accordingly, we agree with the FDA that more research is needed on this issue and that better labeling may be useful, but we would go decidedly further than that. We believe that there is now well-supported urgency that the FDA should do the following at least: review all existing pulse oximeters for evidence that they accurately measure blood oxygen saturation levels in persons with increasing levels of pigmentation in their skin. Absent evidence, or with evidence of deficiencies in that regard, the manufacturer should be required to recall such devices until these problems can be corrected. And we note here that we are very concerned that the FDA's briefing packet included little about the history of pulse oximeter recalls in particular, and safety communications as well. Therefore, we suggest strongly this serious omission is inexcusable from the briefing packet and must be remedied immediately. And finally, the FDA needs to revise the 510K password standards to ensure that devices will only be marketed after they demonstrate reasonable safety and effectiveness in populations at least as diverse as the US population, and as necessary in populations enriched to make sure relevant minority subgroup issues are not neglected. We hope the committee will urge the FDA towards these much needed and long overdue regulatory reforms. Thank you very much.
Thank you very much, Dr. Abrams. Our last speaker for this session will be Rene Kwashi.
Thank you very much. Appreciate the opportunity to speak very briefly. I'm only going to take up a couple of minutes of your time. My name is Rene Kwashi. I'm the Vice President for Digital Health. The Consumer Technology Association, a trade association made up of 1,500 member companies, representing all facets of the tech industry. I have no financial conflicts of interest with regard to this issue. One of the things that we've been doing at CTA is really focusing on health equity. We were co-founders of the Health Equity and Access Leadership Coalition, or the HEAL Coalition, along with 35 other stakeholders in the space. And for us here at CTA, there are two general issues we believe need to be focused on. One is the technological and scientific attempts to rectify the existing problem, which we've been discussing this morning. But the other, which may be more complicated, is how do we help to prevent these types of issues from occurring in the future? So on the technological side of things, I know there are many researchers, scientists, and technologists working on the issue and starting to develop solutions to address the issue. I know, for example, the Professor Coombson up at Tufts is working on a solution using the same light available for current pulse oximeters, but also includes technology to measure a person's skin tone. Just today, I read an announcement from the company BioIntelliSense that has developed a patented alternative to traditional pulse oximetry that accurately measures blood oxygen levels across a full range from light to very dark skin pigmentation, as well as during movement and activity. So there are many people doing great work on this issue, but we may not be fully aware of all that's being done. So one of the things that we would be interested in at CTA is helping develop a repository of all the work that's being done across the globe, so there's more knowledge and coordination among stakeholders who build these solutions and use them in clinical situations. I think the larger problem I addressed in my intro, and perhaps beyond the remit of this particular panel, it's beyond pulse oximetry, is we believe that stakeholders across the ecosystem need to more intentionally think about equity as they develop health technology and medical device solutions. And there are many factors here, right? Inclusive design, by which I mean design that considers the needs and capabilities of the whole population to help decrease misalignment between the user and the design object, right? We understand that every design decision has the potential to include or exclude patients, but the concept of inclusive design emphasizes making informed decisions by better understanding user diversity. We think we need better recruitment of diverse populations in the testing of medical devices and health technology. I think the FDA is doing good work there. We need research teams, quite frankly, engineers, scientists, and others that are more diverse, because I think the diverse stakeholders bring diverse perspectives to the table. And I think the other thing is we need to be developing more quality and safety peer networks where technologists, researchers, clinicians, and patients can exchange information about the potential bias that may impact patient safety. The question for the agency is how to better incorporate all these factors in its decision-making in ways in which stakeholders can help. And just to finish up, other governments around the world are recognizing this. For example, the UK government earlier this year initiated an independent review on equity in all medical devices, seeking to identify and address bias. The review is looking at the device's entire life cycle, from analyzing the evaluation process all the way to marketing and implementation, that's going to lead to a set of recommendations. Now, that may be beyond the remit of this particular panel, but I think the agency may want to look at those kinds of reviews to ensure that we don't have this situation with other categories of medical devices. I thank you for your time.
Thank you very much, and thanks to all of our speakers for respecting the time limit. You're all well within that. And we now have 50 minutes to ask questions to the speakers. And as a reminder, the only ones who should be or can be asking questions are the panelists themselves. The speakers can't ask each other any questions. So let's open it up to the panelists and see if there are any questions that anyone might have of any of our current slate of speakers. Do not see any hands as yet. I'll give it another few seconds. See if anyone has any questions. Going once, going twice. All right. Oh, one question. All right. It just takes one question to get the show going. Dr. Loeb, go ahead. Oh, two questions. There we go. Go ahead.
There we go. I started a trend. Robert Loeb. I I do have questions about the wavelength inaccuracy. Specifically, how might the FDA be able to check wavelength accuracy of already released products? Or, I believe wavelength accuracy is an inherent problem with pulse oximeter technology. We've heard testimony that it's an increasing problem. It, I believe, could be something looked at by biomedical engineers in hospitals and medical centers, much more difficult to do in doctors' offices and by private individuals. But my question is, how could FDA, in a sense, police this if it's a problem?
Is there someone on the FDA who can try and address it? I guess it's not a question for our speakers. Is the FDA able to address that question in this current session?
So this is Dr. Edelman. I believe this session is dedicated to asking questions from the open public hearing.
Okay, well, why don't we save that later on? There might be opportunity to inquire of the FDA or recommend to the FDA later on. Dr. Alam, why don't you go ahead with your question?
Yes, I apologize. I don't recall the name of the speaker, but hopefully he's present and will identify himself. But one of the speakers stated their concerns that even though this is a moderate risk device, the 510K process only requires clinical information from 10 subjects in a study, or at least that was my understanding of what the speaker was saying. And the question for the speaker would be, if he believes this to be inadequate, does he have some suggestion regarding how the regulatory regime may be changed, and what sort of additional information or how many subjects may be required, or would he have concerns about this, in fact, going through a 510K process and want the process to be completely altered? Thank you.
I don't recall who the speaker was myself, but perhaps
it might have been a recorded speaker. That's certainly possible.
Yeah, they might not be with us. So yes. Unless someone wants to, unless any one of our speakers want to try and address that, I think, unfortunately, that one will go unanswered.
Thank you.
Okay, sure thing. All right, well, that puts us back on track. Not that we're too far off track. I now pronounce the first public open hearing session to be officially closed, and we will proceed with today's agenda. We'll now proceed with invited speakers' presentations. During this invited speakers session, we will be hearing from various perspectives from patients, industry, and researchers. In addition, the FDA has also invited a wide spectrum of professional societies representing clinicians who are most directly impacted by the issue of potential bias in pulse oximetry readings due to skin pigmentation. And all who accepted the invitation and completed the necessary paperwork have been given time to present during the session. We are pleased to be hearing from 8 of these professional societies today, and we will begin with Rekha Hagen's presentation, who will address the adult patient's perspective.
Hello, everyone. I'm Rekha Hagen, and I'm happy to be speaking with you about this important issue. I joined the Patient Family Advisory Council of the Hospital of the University of Pennsylvania last year on the recommendation of a neighbor and a friend who was a longtime member and who said they were a great group to work with. They are a great group with compelling individual stories who have had a long association with the hospital. I am one of the few people on the council without a medical background, nor have I had a serious disease or been a caregiver to someone with a serious disease. I bring the perspective of a layperson and community member who desperately avoids doctors and hospitals. Apologies to all you doctors out there. As you can see, I'm Asian Indian with a darker than average skin tone. My husband is white, and our 3 kids are often judged to be Hispanic or Middle Eastern. In other words, we are many shades of brown and white. Furthermore, we live in West Philadelphia, a diverse and dynamic community. So, the first time I had ever heard of a pulse oximeter was about 2 years ago. A friend from India asked if we had an extra one that we could loan to her daughter who was in college in Philadelphia. I had never heard of such a device and frankly thought it sounded too good to be true. But about a year and a half ago, I was standing in line at CVS and saw pulse oximeters arranged on a shelf at the end of the aisle. I bought one. I brought it home. All 5 of us tried it. It was almost like a toy and simple to use. It went into our medicine cabinet where it has been ever since. A few months after I had bought it, I heard that people were being reimbursed by insurance for purchasing one. So I felt more confident about the device, thinking it had been given the seal of approval. The first I heard of the oximeter not being accurate was from the council about 2 months ago. I was shocked to hear that this device could lead to false conclusions. As a layperson, I would compare it to using a thermometer or a weight scale, whereby we believe the number to be absolute and not swayed by other factors. In this time of COVID, it's very important to have an accurate reading because people are acting or not acting on this information. For example, if your thermometer says you have a temp of 105, you would treat it differently from a temperature of 101. I think of the pulse oximeter reading in the same way. And, frankly, if the reading was acceptable, I would not go to the hospital or seek help. Of course, this could be dangerous. My question would be, since we have many skin tones in our immediate family, who would we use this device on? As for current solutions for the FDA, perhaps you could have a skin tone color chart on the box, whereby you are advised not to use the product if you are darker than a certain skin tone. Or sell the oximeter behind the pharmacy counter so that the pharmacist can explain usage to the patient. And lastly, honestly, until I had heard about this issue from the Patient Family Advisory Council, I didn't know about the difference in pulse oximeter outcomes. As a frequent watcher and reader of the news, I'm sure that if I don't know, large sections of my community don't either. The FDA has time to fix this communication. They should start now. Thank you very much.
Good morning, good afternoon, members of the panel and distinguished guests. I'd like to start with a brief introduction of myself. My name is Jessica Kokolin. I'm a certified registered nurse anesthetist. I have a background in neonatal critical care nursing as well. Most importantly, I'm a mother to a child that had a significant congenital heart defect. Consequently, our day-to-day lives relied heavily on pulse oximetry. In speaking to you today, I raise concerns regarding pulse oximetry both as a practicing clinician as well as a mother whose child was critically ill. I declare that I have no interest or involvements with any of the pulse oximetry products mentioned hereafter. Since the invention of the pulse oximetry device in 1974, there has been a growing body of evidence that its technology may not accurately capture low hemoglobin saturation levels in individuals with darker skin pigmentation. To date, pulse oximeters in the United States are required by the Food and Drug Administration to be tested and certified to be no less than plus or minus 3% root mean square error at arterial saturation values. Most pulse oximetry technology involves a minimum of 2 wavelengths of light-emitting diodes that are differentially absorbed by oxygenated hemoglobin and deoxygenated hemoglobin. However, a vast majority of calibration and confirmation tests have been conducted on volunteer subjects of light skin pigmentation. At baseline, known issues exist, such that pulse oximetry will have decreased accuracy with body temperature, altitude, barometric pressure, nail polish or henna, motion artifact, pulse variations in tissue thickness, the age of the patient, and patient comorbidities. In the 2005 study by Bickler et al., at lower oxyhemoglobin saturations, greater differences in bias existed between light and dark-skinned subjects in comparing Nellcor, Novometrics, and Nonin products. In 2022, Cabanas et al. And Shi et al. Both conducted systematic reviews of the literature to find that dark-skin pigmentation influences the accuracy and performance of pulse oximeters. Overestimations by approximately 1% were found for people with high levels of skin pigmentation, meaning those of darker skin tone. The findings from these studies become particularly concerning when these overestimations place individuals at risk for occult hypoxemia, or when the arterial blood saturation is less than 88%, when the pulse oximetry persists with a normal reading of 92% or more. Pulse oximeter guides care in our most delicate populations. Decisions regarding the care of critically ill neonates balances the benefits of supplemental oxygen with the risks of retinopathy of prematurity, bronchopulmonary dysplasia, and even death. Pulse oximetry can also be used to detect infants with congenital heart disease. While the COVID-19 pandemic made pulse oximetry a seemingly household product, by the guidelines from the World Health Organization, home and hospital pulse oximetry are not created equal. The care of a child with a critical congenital heart disease necessitated the continuous use of a medical-grade pulse oximetry device at home. The reliance on the accuracy of this monitor literally meant life or death, life at home, or life in a hospital. The number on a pulse oximeter displays is not merely a number. It has implications for the next medical intervention a child receives, the timing of the next surgical procedure a child must endure, or even the loss a parent and family must suffer. Consequently, I urge you to reassess the standards and parameters to which our current pulse oximetry devices are tested and certified. Thank you again to this panel for allowing me to express my concerns regarding pulse oximetry and how their accuracy may affect individuals of darker skin pigmentation. Thank you for your time.
My name is Bob Kapotic, and I'm presenting on behalf of AdvaMed. I will emphasize the relationship between industry, clinical users, and associated device standards, both domestic and international, which guide manufacturers of pulse oximeters. Three relevant attributes and priorities of AdvaMed are listed. AdvaMed asked for this voluntary presentation owing to my clinical, industry, and standards development experience noted in the bullet points. As my current employer, Edwards Life Sciences, does not manufacture or market a pulse oximeter, I have no conflict of interest. The domestic and international regulations were detailed by others. Missing was the clinical perspective for understanding and use of a pulse oximeter, which I shall address. In the United States, this medical device requires a prescription for use on a patient. The pulse oximeter is a supplemental, non-diagnostic device for clinical assessment of patients. It provides an indirect and inherently imperfect estimate of arterial oxygenation and pulse rate. So if it's imprecise, why use a pulse oximeter? It's convenient, requires little user sophistication, and provides a continuous, non-invasive display of estimated arterial oxygenation and pulse rate. To improve understanding and use of a pulse oximeter, clinical use guidelines are available from various professional societies. These guidelines are published in peer-reviewed journals, commonly available online, and are frequently updated. You already heard some facets of user education and technology shortcomings that will soon be published to further improve user understanding and prompt manufacturer improvements. What are the assumptions that create the two required displayed values of a pulse oximeter, pulse rate and arterial oxygenation? Pulse rate is assumed from counts of detected light peaks, which are believed related to expansion and relaxation of arteries from contractions of the heart, or heart rate. The display of SpO2 is calculated from the amount of light lost in different wavelengths of light emitted into a pulsing volume of tissue, commonly across a fingertip. Only a small fraction of the light emitted into tissues is detected by the sensor, and yet it is that tiny amount of light which the SpO2 calculates. These are the common causes of pulse oximeter imprecision mentioned in the medical literature and clinical use guidelines. Others today have detailed these issues. International standards establish expectations of medical device manufacturers. Indeed, the title of each standard commonly includes the words essential performance and safety. The first pulse oximeter standard was released by ASTM in 1992. Dr. Goldman, Sandy Weininger, and I were involved then, but with a much smaller committee. With the transition to ISO, the committee of all volunteers includes international expert clinicians, key researchers, and device regulators, plus most major manufacturers. These standards require regular review and updating to meet additional clinical needs and advancements in technology. 5 of the major items already under development for the next version of the ISO standard are listed here. Subgroups of our committee are busy addressing those major items with the goal of the next version of the ISO standard being released next year. Thank you for your attention. Hopefully you have a better appreciation of this technology, which has benefited millions, and as with other aspects of healthcare, will improve as we understand ways to make things better.
Hello, I'm Paul Batchelder, Chief Clinical Officer of Clinimark. Today I will share pulse oximetry accuracy assessment from hypoxia trials in multiple systems. From controlled clinical testing, which evaluates the basic design and engineering of pulse oximetry. I'll first review patient data that stimulated increased investigation into the pulse oximetry in dark skin. Then describe conventional pulse oximetry statistics, and finally present data from controlled clinical lab testing meant to evaluate the basic design and engineering of pulse oximetry over the range of 70 to 100%. On the right is the plot of interest from the letter to New England Journal of Medicine. In the data presented, all pulse oximetry readings were from 89 to 96%, while in many instances, a significant number of co-auth reference readings were below 88%. The red rectangle highlights the occult hypoxia region. On the left is the data from the controlled clinical trials, plotted in the same box whisker plots. You will note that the red occult hypoxia rectangle has very few points in that region. Our goal is to help understand why there is such a difference between clinical lab and patient data. To provide an apples-to-apples comparison, I will utilize ARMS and regression plots. ARMS, accuracy root mean squared, combines bias with standard deviation into one number. Neither bias nor standard deviation can be very large in order to meet an ARMS of 3. Here is an example of an informative regression plot. You can see the 45-degree line of identity along which all accurate data clusters. It is easy to identify a significant consistent bias in one person here. This plot quickly identifies a very poorly performing device. This is a plot from a tight sensor placement study. All subjects were first tested with correctly placed sensors, with all data falling along the 45-degree line. Then the sensor was tightened on all subjects, similar to a sensor wrapped too tightly. You can see that all subjects with tight placement read inaccurately high, with black patients reading the highest. Now we'll look at data from sensors tested over 4,644 paired data sets using the same protocol with non-motion, carefully placed sensors in normal perfusion conditions. This data is from a fingerclip sensor with black and white data sets collected at the same time. The black data is on the left with white on the right. The difference in ARMS from black to white is ARMS difference of 0.31 and a bias difference of 0.51. This slide shows a forehead reflectance sensor in the top graphs and 8 different fingerclip sensors in the bottom graphs. On top, the forehead sensor showed a black to white ARMS difference of 0.5 and a bias difference of 0.32. The bottom plots of 8 different fingerclip sensors shows a black to white difference in ARMS of 0.8 and a bias of 0.73. Here we have inferred the data from the New England Journal of Medicine letter and then plotted those data in a regression format. In the left plot, you'll note that the dotted rectangle outlines numerous occult hypoxia readings from the patient inferred data and has no similarity to that of the control clinical laboratory plot on the right. It is known that the palmar surface of the hand in many black persons is much lighter than the rest of the body. In these two plots, we compared the ARMS and bias from black persons with a fingerclip sensor to the same black group with a forehead sensor. You'll note that there's little difference between the finger and forehead, with an ARMS difference of 0.12 and a bias difference of 0.18. Now, under carefully controlled clinical laboratory conditions, we found that when comparing to light skin over the range of 70 to 100%, the bias in dark skin is less than 1% SpO2. This indicates that the source of bias as reported in patients may be something other than the basic design and engineering of the pulse oximeter. Thank you.
Pulse oximeters determine oxygen saturation by shining a light through the skin at 2 wavelengths, 660 in the red region and 940 in the infrared region, and measuring the difference in light absorbance at these 2 wavelengths to estimate the arterial oxygen saturation. The ratio of absorbencies at these 2 wavelengths is calibrated empirically against direct measurements of arterial oxygen saturation obtained in a group of healthy people. The resulting calibration curve is then used to generate the pulse oximeter's estimate of arterial saturation. Shortly after the introduction of pulse oximetry, we observed that readings were less reliable in Black patients. Here you see the arterial oxygen saturation as AO2 plotted against the pulse oximeter's estimate of saturation, or SpO2, in white patients and in Black patients. Inaccurate readings were 2.5 times more common in Black patients than in white patients. We also observed that a pulse oximeter target of 92% ensured a safe arterial oxygenation, which we defined as a PO2 greater than 60, in 91.7% of white patients, but in only 50% of our Black patients. We surmise that the inaccuracy in Black patients is related to skin pigmentation, which may interfere with the absorption of light at the wavelengths used in pulse oximeters. Studies have shown that Black nail polish causes an increase in the difference in light absorbance between the red and the infrared wavelengths, leading pulse oximeter to register falsely low saturations. In addition, the calibration curve incorporated in the electronics of the instrument may be suboptimal for Black patients. It is possible that the calibration data in pulse oximeters are derived largely from white volunteers. So it's reasonable to expect that a calibration curve based on data obtained in Black volunteers would show better performance in Black patients than the present devices. The lesser accuracy of pulse oximetry in dark-skinned patients was brought to the fore during the COVID-19 pandemic. Studies conducted by Shoding in the US and Crooks in the UK once again documented that pulse oximetry was less accurate in Black patients than in white patients, as had been shown in the past. In the Crooks study, the differences between paired measurements of pulse oximetry and the true invasive arterial saturation in Black patients was plus 5.4%. Indicating that pulse oximetry tend to provide falsely high oxygen saturations. The reported 95% confidence limits, however, ranged from minus 25.9% to 36.8%, indicating that pulse oximetry also provide falsely low oxygen saturation in Black patients, as we highlighted in a recent letter to the European Respiratory Journal. Managing patients with unreliable measurements of oxygenation is hazardous, partly because dangerously low saturations are missed, but also because patients are erroneously judged to have lower oxygen saturation than is really the case, which results in unnecessary intubation. Clinical decisions based on faulty pulse oximeter readings most likely contributed to the severalfold greater number of deaths from COVID-19 in ethnic minority patients than in white patients. In the 32 years since we first reported the greater inaccuracy with pulse oximetry in Black patients, the physical structure of pulse oximeters have undergone tremendous change, but the inaccuracy in Black patients has not lessened. The mean difference between pulse oximetry and arterial oxygen saturations was 3.3% in our Black patients and 5.4% in the Black patient of Crooks et al. Although the two-study population differ in several respects, the data collected in 2020 to 2021 indicate that there has been no improvement in the accuracy of pulse oximeters in Black patients since 1990. In the interval, we are not aware of any manufacturer introducing adjusted algorithms into the software of pulse oximeters to resolve the inferior performance in dark-skinned patients.
Hi, I'm Dr. Eric Garman. I'm a pulmonary and critical care physician, and I'm speaking today on behalf of the American College of Chest Physicians on the clinical implications of flawed pulse oximetry based on skin tone. I have no financial disclosures related to this talk. So I think we'll jump right in to some of the clinical research that's been released in the last couple of years. Many of you may be aware of some of this research, but most of it was related to what's called hidden hypoxemia. So the pulse ox is reading a normal level on the screen, but indeed when they check it in the arterial blood gas, they may have a low level and sometimes a profoundly low level. And what's been found is that there is a disparate nature between the Black and white patients, a surrogate for skin tone, which is not a perfect one, but this is self-defined race. So anywhere between 15 and 30% in a very reliable way in multiple studies over the last 2 years, showing that much more Black patients are displaying a normal oxygen level on the pulse ox, but indeed actually have hypoxemia present. And diving a little deeper into this curve here from this study, if you look sort of towards the lower levels of normal on the pulse ox, so in this case 91% will take, over 50% of the Black patients here were hypoxemic and didn't know it, which potentially is having significant clinical implications both for treatment and qualifications for different levels of care, et cetera. This is a study from this year in COVID patients looking for eligibility for COVID-specific treatment, which a lot of times is based on pulse oximetry, noting that Black and Hispanic patients were roughly a quarter to 30% less likely to be offered COVID-specific treatment, presumably because they were reading higher normal oxygen levels than they actually had. Again, showing again the difference between hidden hypoxemia rates between whites and non-whites. This study took things a little bit further and looking at harder outcomes, showing that those with hidden hypoxemia, despite having similar levels of organ dysfunction and sickness ahead of time in that presentation, had excess mortality simply because of their hidden hypoxemia and worsened organ dysfunction scores in those with hidden hypoxemia. So significant clinical outcomes also seen just in that group with hidden hypoxemia. What is very striking to me also in the table from this study is showing the rates, again, at borderline levels of normal oxygen levels, how poorly pulse oximetry performed. And it was pretty much bad for every group. It was just worse with those with potentially with darker skin tones. So a quarter to even higher than that had normal oxygen levels on, or what we would consider okay oxygen levels in the hospital, but actually were hypoxemic and sometimes profoundly so. So that's the inpatient side of things, but what's going on in the outpatient setting could be even more profound and affect a lot more people. And pulse oximetry is tied into lots of things and guidelines for different criteria. And one particular one that we see a lot is the qualification for supplemental oxygen, qualification for supportive equipment or disability parking placards. Do we know how severe our patients' disease is and whether they're progressing or not? And can we trust that pulse oximeter reading that we're seeing? And one very significant whole other field of this is sleep apnea testing and other sleep study testing, where the pulse oximetry criteria are very much tied into the severity and the definition of whether the disease is present or not and how bad it is. So this is potentially a big issue for sleep medicine as well. So why is this potentially a big issue? Is that when you look at the laboratory versus the real world calibration and performance of these pulse oximeters, you see that they perform pretty well when it's done in a normal population that is artificially made hypoxemic. And the splay is not there in the data. Interestingly, the dark and light skin tone difference is preserved, but not dramatically so. But this is from the prior study showing and shown again in multiple other studies, how poorly performing the pulse oximetry is in those with chronic diseases, those with potential severe illness, with acute illness, with circulatory issues, and potentially a range of skin tones. So what I would advocate, and what I think we should advocate for, is the calibration cohort for pulse oximetry needs to be done in the population that it's going to be used in, not in an artificial normal population. So those diverse skin tones, diverse lung disease, levels of illness. So that's who we really are concerned about oxygen levels mostly in, and that's not the population potentially that all these devices are being tested in. And we wouldn't tolerate that for a medicine, so I'm not sure we should tolerate that for a device either. So where do we go from here? So first, I think we need to recognize that there's a problem and potentially a big problem, and that we need to test these devices and ask these device companies to show us who they're testing these devices in and making sure it's in the people that we're using the devices in, and that population is diverse, not only in an objective skin tone way, but also in a variety of disease process ways and inpatients, outpatients, et cetera. And in that way, we can start to trust the levels that we're seeing and hopefully more appropriately trust the accuracy of this device in taking care of our patients. Thank you so much for the opportunity to speak today.
From the Guthrie clinics. The question at hand is whether pulse oximetry is affected by skin pigmentation and color. For treatment in the ICU, as well as with anesthesia, the pulse oximeter is something that is very important to our treatment of patients. And the American College of Surgeons not only lives by the motto, but performs by the motto, to heal all with skill and trust. It is understood that there are several factors that can affect a pulse oximeter reading, and the most accurate way to obtain oxygen levels in blood is to actually do a blood gas sample. Things like ambient light, the size of a person's finger, the presence of nail polish can all affect pulse oximetry in patients, and these are well-known factors in the ICU. Because of this, more sensitive probes, like a probe on the ear, can help ascertain this risk. And in other issues in the ICU, again, just obtaining a blood sample to confirm oximetry is important. going forward, if we can improve the accuracy of pulse oximetry, which at present is only fairly accurate between 70 and 100% saturation, this would help this issue, especially since the studies have shown that the inaccuracies increase with the decrease in saturation. Thus, the sicker the patient, this may be affected. I would be happy to take any questions, but the American College of Surgeons is standing behind treating all patients equally and accounting for any disparities in equipment with blood testing and blood gases in the ICU. Thank you.
I'm Jesse Ehrenfeld, President-elect of the American Medical Association, a board-certified anesthesiologist and a routine end user of pulse oximetry. The AMA has long been aware of concerns regarding the accuracy of pulse oximetry in pigmented skin. Variations in the accuracy of pulse oximetry readings by patient race in critically ill patients has been reported as early as 1990, typically seen as an overestimate of oxygen levels in patients with darker skin pigmentation. Increased use and reliance on pulse oximetry broadly during the COVID-19 pandemic has highlighted the race-based inaccuracies of these devices. The Journal of the American Medical Association has reported an increased incidence of hidden hypoxemia in racially and ethnic minorities, specifically Black, Hispanic, and Asian patients. Failure to appropriately identify a patient with very low oxygen levels can result in patients not receiving supplemental oxygen and potentially life-saving medical treatment, leading to an increase in major organ dysfunction at 24 hours and increases in in-hospital mortality. Now, the British Medical Journal has reported Black patients having higher odds than white patients of having occult hypoxemia noted in arterial blood gas, but not detected by pulse oximetry. There is significant additional literature demonstrating significant issues of racial bias and inaccuracies of pulse oximetry measurement in those with darker skin pigmentation. Additionally, the FDA has noted its awareness that skin pigmentation can affect the accuracy of pulse oximetry readings. Now, the AMA would like to applaud the FDA for recognition of this serious issue that disproportionately impacts certain racial and ethnic groups and contributes to worse health outcomes and widening inequities amongst these populations. We appreciate the FDA's convening of this meeting today and very much look forward to working together to correct this issue. It is critical that the FDA ensures the accuracy and reliability of pulse oximetry readings in patients with diverse degrees of skin pigmentation. The FDA should require quantitative data on device performance across a range of skin pigmentations in clinical studies, particularly devices with color sensing technologies, to identify and mitigate the impact on historically minoritized communities. Studies illuminating varying medical treatment responses based on the color of skin warrant the design of regulatory pulse oximetry accuracy standards. Pulse oximeters that have been shown to reproduce racial bias should require a warning label that warns the end user of the risks of inaccurate readings. The FDA should also require additional collection of real-world evidence and increased post-market surveillance of these devices to ensure appropriate performance and help mitigate potential bias. Finally, the AMA also recommends that all healthcare providers be made aware of the limitations of pulse oximetry technology and be trained to account for the systematic measurement error when developing diagnosis and treatment plans. Earlier today, when I applied a pulse oximeter to a patient in the operating room, I paused. I asked myself, how can I know that this FDA-approved device will give me an accurate reading during the critical moments ahead in surgery? We need to take appropriate steps to remove the growing uncertainty in these devices and ensure the health and safety of the public. Thank you.
I'm Dr. Stephen Gay, Clinical Professor of Medicine, Division of Pulmonary and Critical Care, Michigan Medicine, and Interim Associate Dean of Medical Student Education, University of Michigan Medical School. The issue under discussion is complex and begins with the disparity in the accuracy of pulse oximeters that affects a unique group in our society. This complexity begins with the concern that a population may be experiencing greater morbidity and mortality related to the accuracy of these devices. That complexity is amplified by the concern that this segment of the population has been historically underrepresented in medicine and has experienced a complicated history with both access to care and fair and equitable treatment. This evaluation and investigation are not about assessing blame. We are clinicians, scientists, researchers, and care providers. It is our ethical duty to investigate these issues. We do not want there to be doubt in our interventions and our approach to care. When there is doubt about an intervention or evaluation in one portion of our society, the likelihood is that doubt spreads to others who may not be directly related, simply because something isn't working in the way we expect and hope it will work. We have had significant amounts of historical precedent in healthcare where individuals who are underrepresented in our society have been affected in such a way where doubt has occurred in their minds that they are being evaluated, treated, and cared for with equity. These are two separate and distinct phenomena. There is doubt that occurs when you feel an accepted practice does not include your best interests at the forefront of its actions. But there is also doubt from a scientific and investigational standpoint that a commonly used device is giving us correct information to best care for a population of our patients. We should not and cannot perpetuate this doubt in any way. We cannot simply dismiss the concerns and the unique findings of this data. As individuals at all levels who participate in the delivery of healthcare in all facets of our society, we must strive to ensure that we are always working to deliver the highest standard of care while putting our patients, their complexity, and their concerns first. We must work to ensure society's trust in our practice of medicine, especially in times such as the COVID-19 pandemic. Considerations going forward that are directly related to this issue include: 1. The ATS notes that the pulse oximeter can offer valuable clinical information to guide clinical care in the outpatient and inpatient settings. 2. The medical and scientific community is aware that differences in skin pigmentation affect and impact pulse oximetry results and that such effects may adversely impact clinical decision-making. 3. The COVID pandemic and increased reliance on pulse oximetry monitoring to initiate and adjust treatment for patients with COVID exposed the significant problem that clinicians see when treating patients with darker skin pigmentation, when they must rely on pulse oximetry readings for decisions on care. 4. Until the skin pigmentation pulse oximeter issues are resolved, interim guidance on the appropriate use of pulse oximeter monitoring for patients with darker skin pigmentation should be developed. And 5. The pulmonary community, in partnership with medical device industry and federal agencies, must collect appropriate data to understand how skin pigmentation impacts pulse oximetry monitoring and must develop improved methods to ensure accurate interpretation of pulse oximetry saturation levels for all patients, including patients with darker skin pigmentation. I am grateful to the committee for your diligent consideration on this complicated matter, and I thank you for the opportunity to address you.
My name is Julian Goldman. I'm a physician at the Massachusetts General Hospital and presenting to the panel today on behalf of the Anesthesia Patient Safety Foundation. I have no conflicts of interest to disclose. In June of 2021, the Anesthesia Patient Safety Foundation published an online statement on pulse oximetry and skin tone to address recent findings regarding pulse oximetry inaccuracy in darkly pigmented patients. Pulse oximetry is an essential aspect of modern anesthetic practice. In 1986, the American Society of Anesthesiologists published standards for basic anesthetic monitoring. As these standards were adopted, they led to a 5-fold reduction in catastrophic accidents. The current ASA monitoring standard requires the use of a quantitative method of assessing oxygenation, such as pulse oximetry. Naturally, pulse oximetry is what is used due to its widespread availability, low cost, and relatively good performance. When we use pulse oximeters, we consider 3 aspects of information. One is the trending of the saturation values. For example, we look at the patient's response to changes in inspired oxygen concentration, changes in ventilator settings, or other therapeutic interventions. The threshold value, coupled with an alarm, is vital to alert caregivers to respond to the patient's bedside. That response may be by an anesthesiologist in the patient's OR, or it might be to the bedside on a hospital ward by a rapid response team. But we have to ask the question, when we have a low saturation setting of 85% that's used for the alarm, what is the patient's actual saturation? How much lower is it than 85% in the presence of pigmentation bias? And should the set saturation for the alarm be adjusted to compensate for that error? Without further information, it's unclear what to do. Pulse oximetry is used, of course, for spot checks as well. The single value could be used for screening, diagnosis, longitudinal care on repeated visits to a clinic, and study inclusion and exclusion criterion. A very important use of spot check pulse oximetry is to assess criteria for home oxygen therapy, which is an 89% saturation value. So if a patient is evaluated and if their actual blood oxygen saturation is 80%, but their displayed oxygen saturation is 90%, that patient would not be eligible for home oxygen therapy despite needing it. The repeatability of oxygen saturation measurements is important and not very clear. Pulse oximetry statistics use ARMS, which is an inter-subject population statistic. But in a single patient, we need to consider intra-patient repeatability. We move probes from digit to digit, from hand to hand, as a patient is repositioned or to protect skin integrity. And we are making an assumption that in a single patient, ARMS is 0. Is this assumption valid? Can we depend on repeated measurements in the same patient? This should be investigated further. The Anesthesia Patient Safety Foundation recommends the continued use of pulse oximetry as an essential patient safety device, and that SpO2 value is but one piece of information in an overall clinical picture. Patients would not be well served if clinicians ignore pulse oximetry data or default to arterial blood gases in all darkly pigmented patients. The APSF further recommends improved education on the proper use of pulse oximetry, including the importance of following manufacturer recommendations for proper use, proper site selection, probe selection and placement, interpretation of data, and a deeper understanding of conditions that could impair performance, such as low perfusion. Furthermore, additional research is needed, better prospective well-controlled studies to obtain real-world evidence, better population definitions, patients with broad representation of skin tones are included, and that studies are sufficiently powered to support statistical analysis. We need objective methods to measure skin pigmentation and record that as part of the clinical studies. As far as the types of data analysis, ARMS is useful but may be inadequate because it may not represent the patient population on which the device will be used. Perhaps ROC curves and Clark error grid analyses could provide more insight into clinical performance. In conclusion, the APSF supports the renewed attention to the accuracy of the pulse oximeter, which has revolutionized medical care and augmented patient safety, and we should use this opportunity to more closely examine how we could improve pulse oximetry performance and its use, and work together with clinicians, manufacturers, and regulators to achieve this. Thank you very much.
Hello, my name is Garrett Burnett. I'm an anesthesiologist at the Icahn School of Medicine at Mount Sinai. I've prepared a short talk on pulse oximetry and skin pigmentation for you on behalf of the Society for Technology and Anesthesia. First, I'd like to discuss some basics of pulse oximetry. Pulse oximeters provide a non-invasive estimate of the arterial oxygen saturation, or the SaO2. Pulse oximeters were deemed a standard monitor by the American Society of Anesthesiologists in 1986 and continue to be an essential monitor for anesthesiologists caring for patients in the perioperative period. Pulse oximeters function through red and infrared light absorbance through a perfused tissue, which is typically a patient's finger or a patient's ear. Pulse oximeters are designed to focus on the pulsatile component of arterial blood, and oxygen saturation is provided by comparing the absorbance of light of oxygenated hemoglobin to deoxygenated hemoglobin. Now, anytime you're using pulse oximeters, it's important to recognize the limitations of that monitor. So factors such as motion, ambient light, and temperature can affect pulse oximeter function. Additionally, cardiac dysfunction, such as irregular heart rate or low perfusion states, can impact pulse oximeter function. Now, although pulse oximeters should focus on the pulsatility of the finger and not the finger itself, nail polish has long been recognized to interfere with pulse oximeter function. This is particularly true with nail polish colors such as blue, green, and black. Similarly, skin pigmentation and melanin have been shown to limit pulse oximeter function, which has become an increasingly popular topic of discussion. Although this has kind of been a more recent discussion, there's been concerns regarding the impact of skin pigmentation as early as 1993. Further, volunteer studies in 2005 and 2007 demonstrated pulse oximeter inaccuracies in patients with darkly pigmented skin, but this was mostly in the range of pulse oximeters less than 80%, so in kind of a hypoxic state. Now, the COVID-19 pandemic and the importance of pulse oximeters has driven considerable attention towards the impact of skin pigmentation on pulse oximeter accuracy, particularly in the more commonly encountered oxygen saturations above 90%. The first notable study focused on ICU patients, where they found a higher rate of occult hypoxemia in Black patients compared to white patients. Now, the term occult hypoxemia means a pulse oximeter value greater than 92% with an actual oxygen saturation of less than 88%. In one study comparing Black and Hispanic patients with white patients in the OR, Black patients had 44% higher odds and Hispanic patients had 31% higher odds of occult hypoxemia compared to white patients. Regarding outcomes of this discrepancy, there was one study that demonstrated higher rates of hidden hypoxemia in Black and Hispanic patients. Hidden hypoxemia, though similar to occult hypoxemia, has a slightly different definition. It means that the pulse oximeter value is greater than 88%, while the actual oxygen saturation is less than 88%. Further, in this study, they demonstrated that in patients with hidden hypoxemia, there was a higher incidence of in-hospital mortality, as well as organ dysfunction. Finally, in COVID-19 patients, one study demonstrated that the overestimation of blood oxygen levels were shown to delay or withhold COVID-19 treatment in Black patients and Hispanic patients. In conclusion, the influence of dark skin pigmentation on pulse oximeters may impact clinical care and outcomes disproportionately. Yet, pulse oximeters will continue to be a vital tool for the practice of anesthesiology. This limitation should be recognized by clinicians, and future technologies to adjust for this impact may be necessary. Evidence has shown that pulse oximeters have varying degrees of bias across all manufacturers, and while not every manufacturer has the same level of discrepancy, it's imperative that device manufacturers be held accountable for the performance of their product across patients of all ethnicities. Finally, considerations for increasing diversity of FDA validation and pre-market testing should be considered to further address these disparities. Here are my references, and thank you for having me.
I'm Dr. Liz Bridges, past president of the American Association of Critical Care Nurses. Thank you for the invitation to provide testimony. AACN is the largest specialty care nursing organization in the world, providing education and advocacy for acute and critical care nurses in the US. Why is this issue important to AACN? Because acute and critical care nurses are the professionals providing direct care to these individuals. We are the first line in the detection of clinical deterioration or improvement. Our advocacy on this issue reflects our over 10-year journey related to equity, diversity, and inclusion. In a recent EDI assessment of over 4,000 of our members, they told us they expect AACN to address these issues. Additionally, AACN is a member of the Critical Care Societies Collaborative, which is a partnership of the 4 major professional societies whose members care for America's critically ill and injured patients. AACN was a signatory to the CCSC letters in 2021 and 2022, urging the FDA to direct developers and manufacturers of FDA-regulated pulse oximeters to test all devices to ensure accurate and reliable readings for patients with diverse degrees of skin pigmentation. We continue to advocate for this action. As we address this complex issue, there are numerous areas for consideration. In the absence of immediate technical solution, the conversation on what to do right now in clinical practice is complex. A solution that has been recommended is a race-based adjustment, which based on the literature may mitigate or exacerbate the error. In considering this action, we need to ask who would be benefited and who would be burdened. The other solution is to increase the SpO2 target range to avoid occult hypoxemia. However, 2 recent studies demonstrated this strategy was associated with increased hyperoxia in Black individuals, which also has adverse effects. In clinical studies, there was a greater incidence of occult hypoxemia compared to laboratory studies of healthy subjects. We continue to advocate for post-market evaluations. It is imperative that we explore the effects of the complexities of critical illness and the social determinants of health, including comorbidities, on the observed bias. In 2007, Feiner published a laboratory study that found, in addition to race effect, in 5 of the 6 oximeter probe combinations, females had a greater bias in saturation estimates. In preparing for this testimony, I reviewed pulse oximetry studies. In the past 2 years, less than 50% of studies reported any gender data, and only 2 studies included gender as a model variable. None of the studies addressed potential gender disparities as a main effect. Finally, while acute and critical care are frequently considered as places of care, a more appropriate conceptualization is that acute and critical care follows the patient. As evidenced during the pandemic, individuals were monitoring themselves at home, and the decision to advise a patient to come to the hospital was based on pulse oximetry. To reiterate what we stated in our 2021 letter to the FDA, we remain concerned about the inaccuracies of consumer-grade pulse oximeters, and we continue to urge the FDA to use its influence with device managers to address this issue, as they are being used to make critical healthcare decisions. Pulse oximetry is a ubiquitous technology, but the discussion today is only the tip of the iceberg in the exploration of potential bias or inaccuracies in monitoring devices, embedded algorithms, and clinical trials informing the care of acutely and critically ill patients. This is an opportunity to set precedent to inform study designs to ensure the avoidance of racial and ethnic bias through the use of diagnostic assessments and algorithms that are appropriate for all patients. This is an opportunity for the FDA to lead efforts to identify these discrepancies across the board. This is also an opportunity for the FDA to partner with professional societies, journal editors, and organizations responsible for the development of guidelines and guideline standards to ensure we are not perpetuating bias through our dissemination efforts. Beyond the regulatory solution we're considering today, the FDA has an opportunity to set precedent through its powerful role as a convener, to bring together the scientific and professional communities. AACN, as a leader in critical care, stands ready to support this partnership. Thank you for the opportunity to present.
Thank you very much, and thanks to all of our speakers, outstanding presentations. And I'd like to open it up now to anyone on the panel who might have any clarifying questions for our speakers. We have about 20 minutes for questions. Yes, Dr. O'Brien. Go ahead, Dr. O'Brien.
Hello?
Yeah, we can hear you.
Okay, great. Joel O'Brien, patient representative. Thank you very much for all the presenters this morning. I have 2 questions, if I might ask. One for Ms. Hagan and the other for Paul Balchelder from Clinimark. First, for Ms. Hagan, we're just clarifying. Thank you again for speaking up and being willing to speak to us today about your patient experience. I don't know if I heard correctly. I was just curious as to why your friend was asking for their daughter. What was their concern that they wanted the oximeter for? Secondly, is that you had indicated that you didn't know that there was any issues. You were new with the oximeter. Your whole family had taken it to see it. It was sort of a toy. And then it was only after you went to your patient advisory group that you found out that there were some issues there. And I was curious, was there any instructions that came with it? Was there any labeling that came with the device when you bought it from CVS?
So to your second question, can you hear me?
I could hear you there.
The box is very bland, and I will admit that I didn't really look at the box. I tore it open, and then everybody proceeded to put it on their finger. So the only reason I bought it is because I had heard of it. And that's really, I guess, the only thing I can tell you. There wasn't a lot of deep thought that went into it when I purchased it. And I'm sorry, your first question again was?
You had said your friend from India had asked you about it for their daughter in college.
Right.
And
right.
I was curious as to what they were concerned about, that they were looking for the pulse oximeter.
Well, I think maybe it's more widely used there. It's a device that they knew about that I had never, ever heard of. And I think that she had packed up a box of medicines and whatnot for her daughter to have. And that's one thing that she did not pack up. And so she asked if I had one and could I provide it? And the answer was no, I'd never heard of it. And I could not provide one at that time.
Well, again, thank you very much.
Sure.
And for Paul Botschelder, I believe, sorry if I'm mispronouncing your name, from Clinimark.
Yes.
Thank you very much again for your presentation as well. I was very interested. Thank you actually for discussing accuracy of root management squared, because that's not something I knew, and it was very interesting to learn about that. I appreciate it. And talking about the difference between, which we've heard several times now, about the clinical lab versus patient data, et cetera. What I was interested in was, I mean, it's clear that there's an issue that I hear with pigmentation, but I was looking at causation, and you said something in your last slide, I believe it was, saying that maybe something else other than design or engineering was happening here. And I was just wanted to get clarified from you what exactly you were talking about. And I don't know if they can pull that slide up that you were referring to when you had made that statement. I think it was contrasting, you know, again, with that, is as I read through the pre-material data that was given to me, and I was surprised to find out that in the finger, it's only 5% of melanin distributed there, and yet we still have this disparity that seemed to be equal other places in the body. And I was very curious about that in terms of, you know, is it clearly the observation that skin pigmentation has an issue, but is that really the cause of the variance that we're getting? But
that's a very good question. Let's see, the host, I believe, has stopped my my video, but that's okay. Let's see, now I've got to start my video again. Here we go. Okay. Your question about what I meant when I said maybe it is something other than the design and engineering. Controlled clinical laboratory testing is designed to test the best case performance of the device. And then from that, after that, we're entertaining other types of tests as well. But the big question is, when these devices are performing this well on deeply pigmented subjects, you know, when we had 1,014 paired data points from deeply pigmented subjects in that data pool, what is it that creates this vast disparity in readings in the deeply pigmented subjects? And I believe Dr. Bickler will mention some things about that, but there may be combinations of low pigmentation, deep pigmentation with low perfusion, or even possibly, if you refer to one of my earlier slides, sensor placement. It can also cause that. So these are the conditions that we start with. We know the basic performance of the device in the clinical laboratory testing. Now, what's next? And how do we provide controlled laboratory reproducible testing that represents the egregious issues that we're talking about today?
Okay. All right. Thank you.
Dr. O'Connor.
Right. So my question is nominally for Jessica Colin. And before I ask it, I just want to make it very clear that I'm asking this question as much to get it on the table rather than any expectation that she'll be able to answer it. And the question is, you know, neonates have a lot of hemoglobin F, which has a lower P50 than adult hemoglobin. And so my question is, are we confident in the performance of pulse oximetry in neonatal patients? And once again, the issue of skin tone matters here because African Americans and people of color in general are at far higher risk of having children prematurely. And so as we go down the pathway of trying to understand this, we may require a different expectation or standard for preemies that are neonates. Or for a neonatal population altogether than what we do for the adults. And I don't see that she's here to answer it.
Oh, no. Hi, sorry, just hopping on the video now. So while I did practice in neonates, I haven't in a long time. It's been about 10 years. And I don't think I can say for sure what the current research is. And I don't know how you can accurately measure fetal hemoglobin, each ratio in each infant. So I'll be honest, I'm not sure exactly how accurate a pulse oximetry would be in that patient population. It is great concern though, especially in those neonates when you're trying to treat again, those issues with like retinopathy of prematurity and bronchopulmonary dysplasia, of course.
Great. Yeah, I didn't expect you to have the answer. I just wanted to get the question on the table. Thank you so much.
Thank you.
Thank you. Dr. Collett.
Yeah, my question is for Mr. Batchelder again. And it gets to the issue about the probe, I guess. You were insinuating, I guess, with the one slide that the tightness of the probe can make a substantial difference. I know there's at least one home sleep testing device that has a probe that's one size fits all. And I could imagine that may have some implications on its accuracy. I also wondered about like current oximeter probes, that there's lots and lots of choices out there. Do you have any comments about like how probes themselves should be analyzed, if that's important? And one other question is about like skin thickness. Could that actually have a difference as well?
Certainly, I'll start at the end. Skin thickness definitely can. I want to caution against using simulators with sensors, because it's the sensor skin interface that can only be tested in humans. The performance of the entire system has to be looked at in humans in this area. Is there another area of your questioning that you have?
Yeah, well, just I guess the probes themselves, like how well they fit, or how loosely they fit, or how should that be examined?
Right. Yeah. So that's a real big question. And the information that I provided in that one slide was taken from Dr. Mannheimer's dissertation, which is publicly available, where if the sensor is wrapped too tightly, in a disposable sensor, that can, and what has been shown in his studies, to increase the reading, increase the bias in all subjects, and in Black subjects, as you saw in that study, were much higher. So sensor placement has also been long known, that where it is placed on the finger can make a big difference in the SpO2 reading in a single subject. Dr. Goldman mentioned, inter-subject variability, and that is something that needs to be evaluated. And that is one of the things that can be part of this issue. When we move from carefully controlled laboratory studies where the sensor placement is assured before the hypoxia trial starts, then what happens in the clinic when we're in a hurry, the sensor may move. And then another aspect of sensors that you brought up is they can be significantly different. Some sensors have white background on the sensor, some are black, and those make a big difference in how the sensor will perform. And then also, if the sensor does not inject the light directly into the tissue, but allows some of it to go along the tissue and hit the detector without going through tissue, can cause a shunting effect, which also can make a significant inaccuracy. So all of these things are combinations of what might be happening from our carefully controlled testing of deeply pigmented and lightly pigmented subjects in the laboratory to the hospital environment.
Thank you.
Also, as you're talking, I was hearing other factors that might play a role. It made me think of things that I hadn't thought about before. For example, morbid obesity. I don't know how much subcutaneous fat there is, but how does that affect the saturations? And I think, I didn't know this previously, but when you don't have a good signal, the immediate knee jerk is to squeeze the probe harder or to wrap it tighter until you get a signal which appears that falsely elevates the SpO2. Would that be correct?
That is correct. That happens often, and that can cause a significant issue with the actual accuracy performance of the device.
Thank you. Dr. Loeb.
I guess I'd like to pick on Dr. Ehrenfeld if he's on, but one of his statements was that he now goes into the operating room and wonders, is this pulse oximeter going to give me accurate readings in this patient? And I would propose that that is actually a question that all clinicians should ask themselves about every non-invasive monitor. Pulse oximetry, non-invasive blood pressure, temperature readings should be known to be inaccurate, have limits of accuracy, I guess I should say. And to me, one of the problems is, well, I would ask Dr. Ehrenfeld, has his questioning the accuracy changed because of recent studies, or is this something that I wonder whether he asked himself all along? And I'd be interested in actually a response from any of the technology savvy clinicians, many of which are on this call.
I don't see anyone responding to that. I guess that's quite provocative for all of us to think about. I don't know if
I'm happy to take that.
Okay.
Steve Gay, Pulmonary Critical Care, University of Michigan. I think we have always assumed in all of the patients that we
I'm going to get a message to Phil Bickler that he can't ask a question.
Right. I was answering that question.
Go ahead, Dr. Gay. For folks on the call, it's only the panelists that can ask questions. The speakers can respond when called upon. Go ahead, Dr. Gay.
Right. I think we have always assumed that none of the devices that we use from a non-invasive standpoint are completely and totally accurate. I think the issue at hand here is that there seems to be a greater discrepancy in specific groups of patients as opposed to an overall variability that we would expect in every patient. And I think that is one of the things that gives us a little bit more concern in our approach of interpreting the data. We have now had to begin to wonder, at what point do we look at the normal amount of variability and it's okay? At what point do I start to consider, is this the amount of variability that's expected, or is this more than the amount of variability that's expected? I think because it's not a standard across the board in all patients, it makes it more difficult to interpret and use from a clinical standpoint from that approach.
It's interesting, though, that as clinicians, we like these hard guidelines. So if you think about the prescription for supplemental oxygen. It's if you're 88% or less, and there's no kind of continuum of that. And so people kind of hang their hats on that. And that, I think, perpetuates the fact that perhaps these devices are more accurate than is realized commonly.
So this is Dr. Rizzo from the American College of Surgeons. I'm a surgical intensivist, and I would like to echo what was just said. But we as clinicians are naturally dubious of any answer and continue to check it. I tell people you want someone who's paranoid and checks it again and again. And frankly, I don't prescribe home oxygen or not without getting a blood gas to corroborate the evidence. And it's with all of our devices. It's not just with pulse oximetry. We have to understand that there is a natural tendency for them to not be 100% accurate. And anything that industry can do to make these devices more accurate is an obviously welcome development for us as clinicians, because we strive for accuracy, but we also check and double check. And I'm sorry, I can't share my video because it's giving me the you can't share video message, but thank you.
Sure thing. Mr. Branson.
So I may be getting a little bit ahead of our discussion this afternoon. Health disparities are real in every part of healthcare. But as an editor of a journal, if any of these studies that have been presented outside of the ones by Bickler and Gibran and Tobin from back in the 80s were submitted to my journal as an evaluation of pulse oximetry, I would have rejected all of them, because the time of the recording of the SpO2 value and the recording of the blood gas value was anywhere from 5 to 30 minutes, or closest one available in an electronic medical record. I'm not saying that this is not real, but it does make it makes some sense that there are these large disparities that, again, we see from some of the other presenters, that there's no difference in studies related to race. And it clearly includes skin pigment, perfusion, temperature, and there are so many other things. And as a respiratory therapist, I find the root mean square to mean absolutely nothing to the people at the bedside. You know, you can all test it this afternoon, go into the hospital and ask the therapist what the root mean square is of the pulse oximeter device they're using. They don't know. They're taught that if the heart rate on the oximeter reaches the same as the heart rate on the EKG, then it's probably accurate. So I think there needs to be, I'd be interested to hear from any of the speakers that, is there a better way to discuss what accuracy is, so that people are well informed? And I was listening to Dr. Bridges, who I've known for several years, and I think about first principles, and that's that if there's a gender bias, what's the, you know, with skin pigment, we think it's the melanin. If it's a gender bias, the first thing that comes to my mind is the probes don't fit as well on women, who tend to be smaller than men. And there is this penumbra effect that Paul described, where the light shines around the finger and goes to the sensor. So I'm sorry if that's too much rambling, but I've been sitting here thinking about all this, that the data that we see from some of these studies really begs for, as other people have said, prospective data collection with measurement of skin pigment.
Okay, thank you. Dr. Loeb, go ahead.
I believe in the last presentation, we had some of the authors of the papers of the retrospective studies. But I was wondering about this outcome of occult hypoxemia, which I wonder how that was chosen. It seems like a lot of the studies have occult hypoxemia, which is a very compelling outcome to a clinician. But as was shown earlier by the statistician, positive predictive value is really the much more important outcome. It speaks to the same effect, but does it in a more statistically correct way than just looking at two boxes in the 4 box of outcomes. So this is a question for any of the people who have published using occult hypoxemia, how they pick that and why it seems to be, in my mind, perpetuated in many of these studies.
Any of our speakers want to tackle that?
Well, this is Julian Goldman. Regarding Dr. Loeb's question, I wanted to respond to the prior question as well. I think that there's a consideration that we've missed in the discussion, and that is that, as clinicians, we have a high index of suspicion that perhaps we cannot trust data from any monitor, any device, including a lab test. But I think the unique aspect of today's discussion is that our historical cues as to the performance of a pulse oximeter, as Dr. Branson said, for example, matching the heart rate display with the ECG, with the pulse rate rather with the ECG, or looking at the quality of the plethysmographic waveform, or looking at some other aspect that we've all come to learn over time, may not help and does not seem to help provide insight into this aspect of performance or the failure in performance in some subjects. I think that's really what we're wrestling with uniquely, and that has been pointed out that this is unique, and it's partially unique because our other cues no longer apply. And if we had other information that could flag or identify when a patient's measurements might be more inaccurate because of the skin tone or the pigmentation, that in and of itself could help assess or help us interpret the data. Thank you.
Sure thing. Dr. Yalmas, you did have your hand up, but then you lowered it. You're passing. Okay. Well, if there are no more questions from our panelists to any of the speakers, then we will close this session out. We'll take a 40-minute lunch break, or just under 40 minutes, and we'll reconvene at 1 p.m. Panel members, please do not discuss the meeting topic during the break amongst yourselves or with anyone else attending virtually. And as I mentioned, we'll resume the meeting at 1 p.m. Thank you very much. Good afternoon, everyone. It is now 1 p.m. and I would like to resume this panel meeting. It is now time for the second scheduled open public hearing as posted in the Federal Register notice. However, all requests received from the public to present to the panel were covered during the morning open public hearing session. So we gain an hour even without the official time change. We'll therefore move forward with today's agenda. So there's open public hearing that was scheduled between 1 and 2, nothing to fill that. We had a lot of comments already in the morning, a lot of information to process for the panel. And so we're going to move on to the invited speakers presentation. This next session is covering real-world evidence related to pulse oximetry. During this invited speaker session, we will be hearing from several researchers who have conducted studies to assess real-world performance of pulse oximeters and will present their findings to the panel, starting with Dr. Shoding.
Hello, my name is Michael Shoding. I'm an associate professor of internal medicine at the University of Michigan, and I'll be speaking about our research on racial bias in pulse oximeters. We started to get really interested in this topic during the COVID-19 surge at the University of Michigan. Myself and some of my colleagues noticed anecdotally a couple of times that we were caring for a patient who seemed to have a normal saturation based on a pulse oximeter value on the monitor. But during routine care, when we performed an arterial blood gas analysis, we saw the oxygen saturation was actually low. And we didn't understand why this discrepancy existed. And when we dug a little further, we learned about prior studies demonstrating pulse oximeters had different accuracy in white and black patients. And we got really concerned. That's what we were seeing at Michigan when caring for patients with COVID-19. And so for that reason, we set out to perform a study to evaluate pulse oximeter accuracy in clinical practice. And so we analyzed routinely collected electronic health record data, including patients who self-reported as white or black and were hospitalized in 2020. We compared pulse oximeter recordings to arterial blood gas measurements that were performed within 10 minutes. And using this criteria, we arrived at a population of 1,339 white adult patients and 276 black adult patients, including over 10,000 pulse oximeter and arterial blood gas pairs. And when we plotted the data, we saw that there was a clear difference in pulse oximeter performance in black patients and white patients. At the same pulse oximeter reading value, say 92%, the arterial oxygen saturation levels in black patients were lower than white patients. And this was true across the pulse oximeter range. And this suggested that pulse oximeters seem to be overestimating the arterial oxygen saturation in black patients compared to white patients. And to really make this clinically relevant, we ask, how often was pulse oximeters missing hypoxemia when they were reading at 92% to 96%? And this occurred really infrequently in white patients, 3.6% of the time, but in black patients, 11.7% of the time when the pulse oximeter was reading normal in a range of 92 to 96%, the actual arterial oxygen saturation was less than 88%. And when we also performed this analysis, adjusting for differences in age, sex, and cardiovascular SOFA score, we really arrived at the exact same conclusion, that there was a significantly higher rate of occult or hidden hypoxia where the pulse oximeter was reading normally, but the actual arterial oxygen saturation was less than 88%. We also asked another related question, which was, how well was the pulse oximeter at discriminating low oxygen, defined as an arterial oxygen saturation less than 88%? And we found that in black patients, the area under the receiver operating characteristic curve for discriminating low oxygen was 0.84, whereas in white patients, the area under the receiver operating characteristic curve was 0.89. Again, demonstrating that the pulse oximeter was less able to identify low oxygen levels in black patients compared to white patients. And finally, we replicated these main findings in a multi-center cohort from 2014 and 2015. And we published these findings in our research letter in the New England Journal of Medicine in 2020. And since that publication, we've replicated these main findings in several other patient cohorts. We found again that pulse oximeters were missing hypoxemia 21.5% of the time in Black patients and only 10.2% of the time in white critically ill adults getting evaluated for ECMO therapy. We also found that in Black children, pulse oximeters were missing significant hypoxemia 21.1% of the time in Black and 15.6% of the time in white children. And finally, in a recent analysis, we found again that pulse oximeters were missing hypoxemia 19.6% of the time in Black and 15.6% of the time in white veterans. And this analysis was performed across all US Veteran Affairs hospitals. And in doing these analyses, one thing that we've come to recognize is that potentially small differences in pulse oximeter accuracy can translate into really significant population disparities in hidden hypoxia. And we're illustrating that in this figure in a publication that recently came out in the American Journal of Respiratory and Critical Care Medicine. So what this figure is illustrating is the possible values for arterial oxygen saturation when a pulse oximeter is reading 92%. And so first, let's take a pulse oximeter that's very accurate with a precision of 2% and 0 bias. And that pulse oximeter would have an accuracy root mean square of 2%. And for that pulse oximeter, this occurrence of hidden hypoxia would be quite rare. When the pulse oximeter was reading at 92%, the rate of hidden hypoxia where the pulse oximeter is missing low oxygen is 2%. But if that pulse oximeter was slightly less accurate, if the precision increased to 2.5% and the bias increased to 1%, well, the rate of hidden hypoxia becomes quite a bit higher. Now, when the pulse oximeter is reading 92%, that could represent a true arterial oxygen saturation of less than 80%, 12% of the time. So this, again, just demonstrates how small differences in pulse oximeters can translate into significant population disparities in hidden hypoxia. So that's why it's important to recognize that these small differences at a population level can potentially have a big impact. So these analyses that we performed were all performed using retrospective electronic health record data. And this type of data has many opportunities and strengths, but it also has some limitations. It provides an order of magnitude more patient data to analyze. And so you can do analyses that we've never been able to do before. And it also provides real-world understanding of performance and the potential clinical impact of inaccuracies. However, pulse oximeter and arterial blood gas recordings are not performed simultaneously in this type of study using this type of data. And that will introduce some random noise because from the time that the pulse oximeter is recorded to the time that the arterial blood gas is collected, there can be some random drift in the oxygen saturation, which will introduce some random noise. But importantly, in our New England Journal of Medicine study, the difference in time between the pulse oximeter and the arterial blood gas, on average, was really identical in both Black and white patients. So the same type and amount of noise would be introduced. And so that just cannot explain why we would see these differences between Black and white patients. And then finally, these types of analysis are based on self-reported race, which is an imperfect measure for skin pigment level. And of course, we all tend to think that differences in skin pigment level are what's explaining these differences. However, we do think it's really important to recognize that those most impacted are racial minority groups. So I'd like to close by just offering some actions I really think the FDA should consider based on these analyses. First, it's paramount that manufacturers report pulse oximeter performance across racial groups. And then we must also power pulse oximeter testing studies to be able to detect the small but clinically important performance differences across these groups. And we have to ensure that pulse oximeter testing is aligning with our use in clinical practice. So in clinical practice, when I am caring for a patient, what I want to know is if that pulse oximeter is reading normally, how often and am I actually missing real low oxygen saturation? And that's the reason why we came up with this concept of the occult or hidden hypoxia rate. And finally, I think we have to start using routinely collected real-world post-market surveillance data more and more, including both retrospective and prospective data to evaluate how these pulse oximeters are performing in clinical practice. Because it's critical that these pulse oximeters perform the same for everyone, because they're such an important device for providing good patient care.
Hi, my name is Ian Wong, and I'm a pulmonary and critical care doctor in the Division of Pulmonary and Critical Care Medicine and the Division of Translational Biomedical Informatics at Duke University. Thank you again for the invitation to speak on pulse oximetry and hidden hypoxemia. Here are my disclosures and funding. I've been invited to speak to you today about some of my recent work published in JAMA Open about hidden hypoxemia and pulse oximetry. Thank you also to my colleague, Michael Schoting, who helped inspire our team in this line of work. To briefly review to ensure that we are aligned on terms, I'd like to discuss both the pulse oximeter and the arterial blood gas. On your left, the pulse oximeter estimates oxygen saturation by shining a light through tissue. Arterial blood gases, on your right, measure true oxygen saturation with blood taken directly from your artery. Pulse oximetry values are represented by SpO2 and ABG values by SaO2. For this study, we studied hidden hypoxemia, where patients had a pulse ox value of 88 or higher, but an ABG value strictly less than 88%. As you know, 88% is the threshold for when we consider patients hypoxemic clinically. For hidden hypoxemia, we sought to answer two questions. The first, what's the clinical impact of hidden hypoxemia? And the second, can we extend this work beyond Black and white patients? Our results across 87,971 patients in search of these answers led us to find clinical implications in terms of mortality, where the presence of hidden hypoxemia was associated with 70% higher mortality in all comers. Beyond mortality, we found effects on organ dysfunction, as evidenced by 31% slower lactate clearance, which is affected by oxygenation, and 14% higher organ dysfunction as measured by SOFA scores. We also sought to extend this work beyond Black and white patients, and not only found that hidden hypoxemia was 38% more frequent in Black patients, but also 23% higher in Hispanic patients. And another unexpected finding associated the patient's race to 40 to 66% fewer chances of having their ABG, the gold standard test, measured in Asian, Black, and Hispanic patients. So we found that the gold standard test was less frequent in patients of color. These are our three big takeaways. The presence of hidden hypoxemia is associated with 70% higher mortality. That hidden hypoxemia is 23 to 38% more common in Hispanic and Black patients, and that Asian, Black, and Hispanic patients had 40 to 66% fewer ABG tests. This makes sense given the physics behind pulse oximetry, since it's a simplified version of spectroscopy, as you see here. We see spectra of blood, water, and hemoglobin here, the most common skin chromophores, or pigment molecules. The pulse ox compares the red and green lines you see here in this chart, and you see that melanin overlaps with these lines here. The only way around this is to remove the melanin again. The ABG, which removes the blood out of the body, measures it. So although the pulse ox is more convenient, the ABG has inherent accuracy benefits. We've known that these complications have been present for years in both healthy and ICU patients. So what can be done about this? Are we hopeless? We believe that there are both potentials for solutions in hardware and in software. and we're happy to discuss this at a later point. But for the purpose of this talk, let's ask, what could the FDA do? The FDA had kindly released this pulse oximeter community safety communication in February of 2021, but there were no conclusions and no recommendations off that, just caution in considering the pulse oximetry results. So while these improvements are being developed, how can we help the FDA build the framework by which we can judge these improvements? Based on the results of this work, I believe we can consider reevaluating what the standards for accuracy are. Per the communication, this generally means that the accuracy requirements have 66% of SpO2 values within 2 to 3% of blood gas values. 95% of SpO2 values were within 4 to 6% of blood gas values, respectively. So if we look at this bell chart, looking at standard deviations, and with the pink showing 95% confidence interval could be as given the plus minus 6%, anywhere from a true ABG sat of 94%, range from 88 to 100% by the pulse oximeter. So if we're reevaluating accuracy standards, we could consider tightening accuracy requirements. The true value of plus minus 6% is permitted, and SaO2 of 94% could read anywhere between 88 and 100%, which means that's my entire clinical range for treating a patient and keeping their oxygen saturations. Additionally, to reevaluate accuracy standards, the FDA guidance suggests that these clinical studies were not statistically powered to detect differences in accuracy between demographic groups. Can we consider testing enough patients to do so? For example, just as we consider testing patients in drug trials, could we hold device trials to similar standards, or perhaps standards that focus on results in specific subpopulations? Furthermore, what context do we need to evaluate and test these patients? Just as we note that pulse oximeter accuracy is highest at saturations of 90 to 100%, intermediate at 80 to 90%, and lowest below 80%, pulse oximetry accuracy is affected by value here. So, can we challenge them to test across sufficient SpO2 ranges throughout clinical care? Specifically, we need to test ranges to put the lower range accuracies, or in this case, inaccuracies, to the test. The lower ranges, especially around 88%, are the most important in changing clinical management. For example, if a patient is at 90%, perhaps current standard of care may not consider turning up their oxygen therapy, whereas if they were 86%, they might. Furthermore, another consideration includes that the FDA guidance recommends that there are at least a range of participant skin pigmentations within the accuracy testing, including at least 2 darkly pigmented participants, or 15% of the participant pool, whichever is larger. But let's ask, what does darkly pigmented mean? How do we quantify that? And finally, how do we do so in numbers that allow us to draw significant conclusions from that data? We do need to test across skin chromophore ranges. As we noted, skin chromophores, like melanin, have reasonable ways by which they can bias our measurements. On one side, Fitzpatrick himself notes that ethnicity and race have no scientific basis. But the FDA's publication on reducing a bias in AI and ML research itself notes that there are many contexts to race and ethnicity. We need to test for accuracy within a range of skin chromophore concentrations to know when our results might be valid. Could we consider looking at both performance of pulse oximeters and performance of downstream care as possible results or metrics? But how do we do this? Is it the Fitzpatrick skin scale, which was developed originally across 4 skin types, and skin types for 5 and 6 were added for Black patients? But how do we do this? Is it the Fitzpatrick skin scale? But the Fitzpatrick skin scale was developed with ranges 1 through 4, primarily for white patients, and types 5 and 6 were generated just for Black patients decades later. Is it the von Luschen chromatic scale with 36 different options? Or is it the Monk skin tone scale, which was developed to help improve research in AI and ML, algorithms? Or conversely, do we realize that some of these may actually be subjective gradations, and that would objective measures be perhaps beneficial for helping us to remove the subjectivity and helping us deliver more objective, equal care to all patients? So, in summary, I think that the FDA's biggest effort here is to consider reevaluating accuracy standards. Could we begin to consider pulse oximetry disparities and their downstream implications as a quality improvement metric? The fact-of-life issues, for example, of CAUTI and CLABSI were resolved once we considered them as part of our QI system. Are these different ways by which we could consider judging both the technology and the systems of care themselves? I'd like to thank my colleagues, as shown here, and thank you for the opportunity to present with you. I remain ready to answer any questions you may have. Thank you.
Hello, I'm Ash Fawzi from Johns Hopkins University. I'll be speaking about the consequences of racial bias in pulse oximetry on clinical decision-making, highlighting our recently published research in patients infected with COVID-19. I want to thank the organizers for inviting me to speak on this important topic. I have no financial interest or professional relationship to disclose. Informed by Dr. Michael Schoding's study published in December of 2020, showing higher prevalence of occult hypoxemia among Black patients, I became more aware of the issue in my clinical practice. This graph displayed the oxygen saturations of a 50-year-old Black man with COVID-19 infection who I cared for in the intensive care unit. On the y-axis, you see the oxygen saturation, and on the x-axis, the time in hours. The readings from the pulse oximeter are represented by the blue line, while the gray line represents the actual oxygen saturation as measured by an arterial blood gas. As a consequence of consistent overestimation of the true oxygen saturation by the pulse oximeter and an error of up to 20%, the patient experiences 10 hours of occult hypoxemia, defined as an oxygen saturation below 88% despite normal pulse oximeter readings, highlighted here in red, before supplemental oxygen is increased to 100%. This 85-year-old Black man with COVID-19 infection spends over 5 hours with occult hypoxemia as a consequence of pulse oximeter error of up to 17% before this is recognized and the amount of supplemental oxygen is increased to 80%, improving the patient's oxygen saturation from 76% to 96%. Informed by our clinical experiences, my colleagues and I conducted a study using curated clinical data from patients infected with COVID-19 within the 5 hospitals of the Johns Hopkins Health System. We first aimed to determine whether and to what extent pulse oximeter accuracy differed by race and ethnicity. Second, we aimed to evaluate whether pulse oximeter inaccuracy by race and ethnicity impacted recognition of eligibility for oxygen threshold-specific therapy. By studying a group of patients who all had the same disease, in this case COVID-19, we were able to evaluate clinical decision-making. To accomplish the first aim of the study, we identified 1,216 patients with over 32,000 paired oxygen saturation measurements within a 10-minute window. We showed that while on average, oxygen saturation is typically underestimated by pulse oximeters for white patients, pulse oximeters routinely overestimate the true oxygen saturation in Black, Hispanic, and Asian patients, which can lead to underdiagnosis and undertreatment. We also note that extreme overestimation is more common among patients from racial and ethnic minority groups, with almost a quarter of pulse oximeter readings in Black patients overestimating the true saturation by 4% or more. Using a statistical model to adjust for demographics, comorbidities, and other factors potentially associated with pulse oximeter inaccuracy, such as temperature, blood pressure, hemoglobin, and bilirubin level, pulse oximeters continue to significantly overestimate true oxygen saturation among Black, Hispanic, and Asian patients compared to white patients by an average of 1 to 2%. As I'll demonstrate in the subsequent slides, this seemingly small average difference resulted in substantial clinical consequences. To accomplish the second objective of our study, which was to determine the clinical impact of pulse oximeter inaccuracy, we looked to the larger sample of patients with COVID-19 who were monitored by pulse oximetry but did not necessarily have an arterial blood gas measurement. Using the statistical model we created, we predicted patients' true oxygen saturation. Since the joint guidelines of the CDC and the Infectious Disease Society of America recommend starting COVID-19 targeted medications such as remdesivir and dexamethasone at an oxygen saturation threshold of 94% or below, we identified 1,903 patients breathing room air whose predicted oxygen saturation fell to 94% or below, while their pulse oximeter reading was above 94%. In essence, these 1,903 patients qualify for COVID-19 therapy based on guideline recommendations. However, this was not picked up by the pulse oximeter. These patients were further divided into two groups. Those with pulse oximeter readings never fell to 94% or below and were thus never recognized as needing COVID-19 therapy, and a larger group who eventually had their need for treatment recognized. Of the 451 patients who never had their need for treatment recognized, meaning that their pulse oximeter readings never fell to 94% or below, despite their predicted oxygen saturation falling to that level, half were Black, a quarter were Hispanic, but only 17% were white. The remaining 1,452 patients eventually had a pulse oximeter reading of 94% or below. Of this group, Black and Hispanic patients were significantly more likely to have a delay in recognizing their need for COVID-19 treatment. Black patients were 29% less likely to receive timely therapy, and Hispanic patients 23% less likely because of systematic overestimation of oxygen saturation by pulse oximeters. In conclusion, our study builds upon mounting evidence showing that pulse oximetry overestimates oxygen saturation in patients from racial and ethnic minority groups by demonstrating that despite small average differences, there were substantial delay in recognizing need for COVID-19 therapy for Black and Hispanic patients as a consequence of differential pulse oximetry inaccuracy. This may have contributed to racial disparities in COVID-19 outcomes reported in the literature. Limitations are similar to other retrospective analyses using clinical data, with the added limitation that treatment delay estimates depend on how often pulse oximetry was measured or recorded. The differential overestimation of oxygen saturation by pulse oximeters is part of a consistent pattern of systematic underdiagnosis of disease and withholding of therapy from patients of racial and ethnic minority groups. For example, determining kidney and lung function relies on equations that incorporate race, leading to underdiagnosis and either delay or withholding of life-saving treatments or interventions, such as transplant. However, unlike these examples, pulse oximeters appear to have a fundamental flaw in the acquisition rather than the interpretation of data. The evidence that pulse oximeters overestimate oxygen saturation among Black, Hispanic, and Asian patients in clinical settings has implications beyond COVID-19 and acute hypoxic respiratory failure. Diagnosis of chronically low oxygen saturation among individuals with COPD or interstitial lung disease in the outpatient setting relies on pulse oximetry. And insurance coverage for long-term oxygen therapy is based on strict oxygen saturation cutoffs, which can lead to inappropriately withholding this therapy from certain populations. Finally, it is imperative to ensure that pulse oximeters perform equitably before further expanding their use. For example, the diagnosis and classification of acute respiratory distress syndrome, or ARDS, a condition that can affect patients with severe COVID-19 and has a high mortality rate, is currently based on partial pressure of oxygen, which must be measured by an arterial blood gas. Some researchers have recently suggested loosening this definition to allow estimating partial pressure of oxygen from pulse oximetry. Based on the results of our study and others, it is likely that the scenario depicted here, where oxygen saturation is overestimated by 3%, resulting in misclassification of ARDS as mild instead of moderate, would occur more commonly among Black and Hispanic patients, leading to withholding life-saving treatments. I would like to acknowledge and thank my co-authors who made this research possible. We urge the FDA to strongly consider the real-world evidence presented today using data from thousands of patients in the clinical setting that highlights the challenge of relying on differentially inaccurate pulse oximeters. These findings raise significant concerns that the device class has substantial shortcomings that lead to disparities in the way patients from racial and ethnic minority groups are treated. Enhanced regulatory requirements are urgently needed to ensure equitable patient care. Thank you for your attention.
I'm Dr. Eric Gottlieb. I'm a hospitalist at Mount Auburn Hospital in Cambridge, Massachusetts, a lecturer at the MIT Laboratory for Computational Physiology, and an instructor in medicine at Harvard Medical School. And I'm going to share today our research on racial disparities in pulse oximeter performance and oxygen supplementation rates. I have no relevant disclosures. As we know, there's a growing body of research on pulse oximeter performance disparities by race, and in addition to studies over the past few decades that have showed that these disparities exist, a number of more recent studies have shown that these differences by race are associated with adverse outcomes, including mortality. However, what has not been explored until recently are the factors that mediate these adverse outcomes. In other words, if you know you have pulse oximeter performance disparities and you eventually get these adverse clinical outcomes, what are the factors in between that get you from point A to point B? This question led to our recent study, which was published in JAMA Internal Medicine. The premise of this study was that if there are disparities in pulse oximeter performance, and some patients have pulse oximeters that read artificially high, does that lead those patients to receive lower delivery rates of supplemental oxygen in the hospital? So again, the objective of this study was to determine if there are differences in supplemental oxygen delivery rates across races when controlling for blood hemoglobin oxygen saturation and other confounders. This study was based on data from the Medical Information Mart for Intensive Care, or MIMIC-4, which is a collaboration between the Beth Israel Deaconess Medical Center in Boston and MIT. For this study, we identified a cohort of patients from MIMIC-4 who were in the ICU for at least 12 hours prior to intubation, non-invasive positive pressure ventilation, or BiPAP, high-flow nasal cannula, or tracheostomy, or went for 5 days or more, and 5 days being the maximum amount of data in this study without receiving any of these treatments. For this study, we used time-weighted averages for the variables of interest, and we did this using an area under the curve. And these variables included oxygen delivery, hemoglobin oxygen saturation, or SaO2, and peripheral pulse oximetry, or SpO2. For our analysis, we used linear regression and mediation analysis to assess associations between SaO2, SpO2, and oxygen delivery rates according to race. This figure shows the selection criteria for our final cohort of 3,069 patients and the reasons that patients would have been excluded from this analysis. And this includes race other than Asian, Black, Hispanic, or white, patients missing key data points that were used for analysis, and then patients with an index period, which is the relevant included data period of less than 12 hours. This is our table 1, which shows the clinical characteristics of the patients in this study. And important things to note here are first the racial breakdown of this study up top, which includes 83 Asian patients, 207 Black patients, 112 Hispanic, and 2,667 white patients. And the other thing that's very important down here is the SaO2 averages, the hemoglobin oxygen saturation, and the SpO2. And we can see, again, differences according to race. And what's notable is that if you compare, for example, Black patients and white patients, Black patients had lower average blood oxygen saturations, SaO2 compared with white patients, and Black patients also had higher SpO2 conversely than white patients. This is Table 2, which provides some additional information on the clinical course of patients in this study, including when blood gases are drawn and how many for various patients. For those patients who required advanced respiratory support, what that consisted of, and also the care units in the hospital where patients were admitted. This is a visual summary of our main findings. And walking through this figure, in panel A, we see on the x-axis the average hemoglobin oxygen saturation, and on the y-axis is the average SpO2. And if you look at this figure, the light blue is Black patients, and the dark gray is white patients. And what we can see here is that Black patients for a given hemoglobin oxygen saturation have a higher SpO2 than white patients. In panel B, we have box plots showing the average oxygen delivery rates, and we can see that there are many more white patients receiving high rates of supplemental oxygen compared to Asian, Black, and Hispanic patients. In figure C, we have hemoglobin oxygen saturation versus average oxygen delivery rates. And it should be noted here that for this visualization, we excluded some outliers. So the cutoff for oxygen delivery is 0 to 10 liters, whereas in panel B, we showed all the way up to 20 liters. And we can see that for when looking at average hemoglobin oxygen saturation versus oxygen delivery, Black patients who had higher SpO2 than white patients have lower oxygen delivery rates. And then in panel D, we again are looking at oxygen delivery rates on the y-axis, but on the x-axis is SpO2 pulse oximeter instead of SaO2 or hemoglobin oxygen saturation from the blood gas. And here we don't see a significant difference across races. And what this suggests is that patients are being treated according to SpO2 readings. So it's not that a doctor is saying, well, this patient is white, so I'm going to give them 2 liters of oxygen, and they're Black, so I'm going to give them 1 liter of oxygen. But it's what these providers are not seeing that leads to these disparities across race. This table is showing the linear regression results supporting our above findings, and we have 5 models that we share, Model 1 to Model 5. Model 1 has the outcome of SpO2 corresponding to the prior panel A. And this is showing that Asian, Black, and Hispanic patients, again, have higher SpO2 than white patients, controlling for hemoglobin oxygen saturation and other confounders. The next 3 models, Model 2 to Model 4, all have the outcome of oxygen delivery rates. And we show that these 3 minority races all have lower oxygen delivery rates than white patients. In Model 3, we have a similar regression, except we include the additional term of the discrepancy between the SpO2 and the SaO2, or the gap between the pulse oximeter and the blood gas. And we see that when we include this, the differences according to race are no longer significant. So this explains that this difference between race manifests itself in terms of this gap in performance, and there's not some other inherent quality about race that affects the ultimate results. Model 4 then corresponds to panel D, which is the SpO2 on the x-axis and oxygen delivery on the y-axis. And again, we show that when we're titrating oxygen according to SpO2, which is generally what's done, differences according to race are not significant. And model 5 is what's called a falsification test, where we just use a totally different variable instead of oxygen delivery rates, in this case heparin rates, to show that our findings are not just chance findings, and with heparin rates where we would not expect there to be a difference between races, there in fact is not a difference between races, and that's what we saw here. We then conducted a mediation analysis, which is a type of simulation, to show that the discrepancy between SpO2 and SaO2 mediates the differences according to race that we're seeing. And in this first line here, ACME is the average causal mediation effect, and the significant p-value less than 0.05 shows that this gap does mediate the differences in oxygen delivery that we see according to race. So these are our conclusions. First, we confirmed the previously reported pulse oximeter disparities according to race and ethnicity. We showed that minority patients received less supplemental oxygen than white patients for a given hemoglobin oxygen saturation, but not for a given pulse oximeter reading. And then we showed that the gap between the pulse oximeter reading, the SpO2, and the hemoglobin oxygen saturation mediates the observed racial disparities. Limitations include that this is from a single center. It only includes data on nasal cannula oxygen delivery and not other forms of support. There is no minute-to-minute temporal matching of blood gas and pulse oximeter, and that race is self-reported, and that's imperfect. And with that, I can take any questions. Thank you very much.
I'm Dr. Phil Bickler, representing the EQUIOX study group at the University of California at San Francisco. The EQUIOX study is a prospective clinical study of pulse oximeter errors in hospitalized patients. Today, I will provide the background for the study and the trial design. My co-investigators are Dr. Michael Lipnick, Dr. Carolyn Hendrickson, Dr. John Feiner. We all hail from the UCSF Hypoxia Research Laboratory at UCSF, and our partners include the Center for Excellence in Regulatory Science and Innovation at UCSF and Stanford, the UCSF Hypoxia Lab, the UCS Center for Health Equity in Surgery and Anesthesia, and the U.S. Food and Drug Administration. Why are we doing the EQUIOX study? EQUIOX is in part a response to 5 retrospective clinical studies showing an apparent racial bias in pulse oximetry. In each, pulse oximeters were reading in the 90s in Black patients when their oxygen level in the blood was actually in the 80s, missing a diagnosis of hypoxia or occult hypoxemia. Data from the first of these studies, that by Schoeding College at the University of Michigan, is seen here. I must say that in some quarters, such results were explained away by the limitations of retrospective trials, including imprecise pairing of saturation measured by the pulse oximeter in blood, hemoximeters set to fractional and not functional saturation, self-reported race without skin pigment data, and the presence of interfering pigments, anemia, low perfusion, and motion. However, this bias, albeit smaller, was described in the 1990s and detailed by our work from UCSF in 2005 and 2007. Pulse oximeters are a marvelous invention, but they require calibration involving healthy subjects, arterial catheterization, and controlled hypoxia, conditions only achieved in a handful of labs in the world. In lab testing, pulse oximeters can be calibrated quite accurately and perform well across all skin pigment types, as in the analysis of 4,000 paired samples from Clinimark in collaboration with Paul Batchelder, which we presented in a paper by Okanyola and her UCSF colleagues in earlier this year. So in these data, we can identify a gap between the real world and the lab. And it has been our mission at UCSF to identify what is amplifying this gap. Notice that I said amplify, not create. This gap has been seen before. Next, I want to tell you about results from a new UCSF study that goes a long way toward explaining this gap between testing in the lab and in the hospital. We examined in detail the performance of the Nellcor N595 and the Masimo Radical 7, oximeters which are in widespread use in North America, Europe, and the Asia Pacific region. We put together 9,800 data pairs that match hemoximeter values and corresponding pulse oximeter readings in the NELCOR and the Masimo. And we subjected these data to a multivariable model, including skin class measured by Fitzpatrick, perfusion index, hypoxia, sex, and age. And we segregated these data by according to the perfusion. And perfusion index was chosen by a sensitivity analysis and its relationship to bias, and furthermore corresponds to our clinical experience with these devices. The study that I'm going to describe has just been posted on medRxiv, so you can refer to it for the details. It's the first author is Koa Gutalunas, and the Gutalunas study has been critical to our design of the EQUIOX study. The results of the multivariable analysis were striking. The bias in both the NELCOR and Masimo were highly associated with skin pigment, perfusion, and hypoxia. Let me explain these complex plots. On the horizontal axis in each, we have arterial oxygen saturation spanning the entire range we studied from about 68% all the way up to 100. The vertical axes in each are the bias of the oximeter. That is the difference in the reading between the pulse oximeter and the true value in the blood sample. The data are segregated by skin category, so light, medium, and dark, and also importantly by the perfusion index. The red points indicate perfusion index greater than 2%. The cyan color is 1 to 2%, and the X's mark low perfusion, where the perfusion index is less than 1%. And you can just see by simple visual inspection how in the darkly pigmented group, there's a much wider spread of data, and the particular outliers on the high side are represented by data points with low perfusion index. The relationship between perfusion level and bias was also striking. Here we plot perfusion level on the horizontal axis and the bias on the vertical. And in the darkly pigmented subjects, you can see the distribution of bias is clearly in the positive range, where it is more neutral in the lightest subjects and intermediate in those with intermediately pigmented skin. Indeed, low perfusion explains the misdiagnosis of hypoxemia in our data. We are convinced that low perfusion is the key factor that is amplifying bias in those with dark skin and low perfusion. Here, hemoximeter readings and saturation values are compared, showing that in dark subjects, the rate of misdiagnosis of hypoxemia in healthy subjects in a lab environment are up to 30%. It was 30% in the Masimo and 8% in the NELCOR. With that as a foundation, the EQUIOX study is designed to address the most pressing questions related to pulse oximeter accuracy in a real-world setting. Our prospective study includes synchronous paired samples, so we have a very tight knowledge of what the oximeter is reading at the time the blood sample is taken. Functional saturation is measured, what the pulse oximeter sees. Skin pigment is quantified by the Fitzpatrick chart, the von Luschan chart, and by the Monk scale, as well as quantified by the Konica Minolta and Delphin skin colorimeters. Perfusion is quantified, optical signals are measured. We have an inclusive network of collaborators, stakeholders, and statisticians, and our target population is one that is representative of a full range of skin pigmentation. Dr. Carolyn Hendrickson is the clinical lead at San Francisco General Hospital. The study designs includes the primary aim of measuring bias in pulse oximeter performance in hypoxemic patients across varying skin pigmentations. Our secondary aims include determining what skin pigmentation metrics best correlate with pulse oximeter bias, determining if pulse oximeters perform in clinical use at regulatory standard level, and third, we will test if low perfusion state is a possible mediator that might explain differences in oximeter performance across categories of skin, different skin pigmentation. Our study timeline is rapid. We began this study in August. We obtained IRB approval. We have national clinical trial registration. We have 55 patients enrolled as of now. Our interim data analysis with the aid of our FDA partners will be in November 2022. We're considering possible site expansion depending on enrollment numbers and data distribution, and we anticipate that the study will be completed in July of 2023. All our data is being posted on the Open Oximetry Project website, and we want to thank our collaborators, Cersei at UCSF and Stanford, and the UCSF Hypoxia Lab, as well as the U.S. Food and Drug Administration, the Gordon and Betty Moore Foundation, the Patrick J. McGovern Foundation for essential help with the project. Finally, I want to acknowledge my enormously talented research group, without which we would not be able to tackle this task. They have been incredibly helpful as we initiate the project and will carry it forward. So special thanks to all of them. Thank you very much.
Hi, it's a tremendous pleasure to be here. My name is Chris Allman from Stanford University, and I work in the Division of Pediatric Cardiology at Lucile Packard Children's Hospital. I'm going to take a few minutes and just talk about the launch of a prospective clinical study to evaluate the accuracy of pulse oximeters in children. I should mention that none of the study investigators has any relevant financial disclosures. Just to introduce some of those investigators, this includes Desiree Conrad, who's currently a third-year cardiac fellow with us, two cardiac anesthesiologists, Chandra Ramamurthy and Manchula Navaratnam. Rebecca Kemeny is in the cardiac ICU and also leads our DEI initiative. Greg Adamson is in the cath lab. Rohan Taneja is our study coordinator. And this all began with a conversation with Michelle Tarver. And so we kind of consider her to be part of our team as well. So I'll briefly review the motivation behind the pediatric study to describe its design, and then finish up with some reasons why this project may be a little bit more challenging to conduct in children relative to adults. So I think, as has been mentioned before, this is also true in children, that pulse oximetry is used widely to determine whether children are adequately oxygenated. But recent studies suggest that pulse oximetry systematically overestimates the true oxygen saturation in children with darker skin pigment. Now, this error puts children with darker skin pigment at considerable risk for health disparities by failing to detect important levels of hypoxemia that may drive critical treatment decisions like COVID medication usage, hospital admission, ICU transfer, intubation, and even consideration for ECMO or life support therapies. And prior studies have understandable limitations related to their retrospective design, including using race and ethnicity as a proxy for skin pigment, O2 saturations that appear to be paired in the electronic health record, but might not be either simultaneous or performed at steady state. And it isn't very good at analyzing technical factors like motion artifact and perfusion that could also affect the pulse oximetry values. As has been discussed, there are a number of studies in adults that have really looked at this, especially in the COVID era. And then probably the best pediatric study was conducted by Erica Andrist at the University of Michigan, who looked at it in 1,000 children and found similar findings, the discrepancy in the accuracy in patients with darker skin pigment. So the primary rationale for the pediatric study is to overcome these limitations with a carefully designed prospective study and ultimately to determine if hospital-grade pulse oximeters perform outside FDA specifications in children with darker skin pigment. So the design of the pediatric study actually parallels the adult study with a primary hypothesis that the mean bias or error in FDA-cleared pulse oximeters increases with increased skin pigmentation as measured by colorimetry, as well as standard pigmentation scales, most notably the Fitzpatrick and the von Luschen. And then we will look at other pediatric specific factors like age and the presence of cyanotic heart disease, an important subset of children, in how it influences the degree of error, So this is a prospective single-arm clinical trial in children in a real-world setting, which is Lucile Packard Children's Hospital. And the total sample size is 154 subjects. Children will be eligible for inclusion in the study if they are up to and including the age of 21, which is the definition of pediatrics at the FDA's Center for Devices and Radiologic Health. They must already have an arterial vascular access line as part of their routine clinical care. And they must not be anemic, have hemoglobinemia, or a skin condition that could complicate probe attachment or interpretation. And they cannot be non-pulsatile patients. This is mostly relevant for our heart failure patients on left ventricular assist device support. Patients will then be recruited generally in the clinic the day prior to their scheduled cath procedure. They'll provide written informed consent, and for older kids, there will be an opportunity to provide assent for participation. Skin pigmentation will be measured 3 different ways at the potential site of a pulse oximeter. This includes colorimetry and 2 pigmentary scales, and this will be performed at up to 5 to 8 different locations where the SAP probe may be affixed. We will then conduct 5 paired measurements of SpO2 and SaO2 that will be obtained simultaneously under steady state conditions, and technical factors such as perfusion and temperature will also be captured at that time. The total sample size for this study is 154 subjects. You can see the assumptions that are listed here. Perhaps most importantly, the sample size depends on the interclass correlation coefficient of the clustered measurements within a patient. And since this is something, the sample size can vary considerably, the plan is to actually start the study, verify that the ICC is roughly around 0.15, and if needed, adjust the sample size either upward or downward based on the preliminary results. So to this extent, the study has somewhat of an adaptive component to it. We've been lucky with our great team and the collaboration with FDA and UCSF to have been able to get this study almost ready to be up and running. We received the notice of a grant award from the FDA in August. We had Stanford IRB approval in September. We have a study coordinator that's now been hired, Rohan. We've had 2 site visits with UCSF, both to their hypoxia lab and to San Francisco General, where the study is enrolling, and we are nearly ready to enroll. So lastly, just wanted to touch briefly on a few reasons that this project may be harder to conduct in children. So we need to be prepared. The first is that unlike adults, pediatric diseases in general are relatively rare, especially including children who require arterial lines. And this could complicate enrollment. Now, we think we have more than enough patients at Stanford to enroll the study, even if we need to augment the sample size, but it's something that we want to be aware of. Secondly, there's quite a bit of color variability in children, most of which is related to vasomotor reactivity that's normal. So children, and especially infants and newborns, can be very red. Perhaps someone has seen an infant who's crying or is stooling, and they can turn a very beet red, which is not in the von Luschen or Fitzpatrick scales. They can also be mottled, so kind of a variable skin tone, and they can also be pale depending upon their state. And this could complicate things somewhat. It's also very age appropriate for infants and toddlers to not follow instructions. So for example, in an adult study, you might be able to say, please hold your hand steady or steadily while we make 3 careful measurements to ensure their reproducibility at steady state. And of course, this can't really be done in children. And then cyanotic heart disease, which represents an important subset of patients and probably introduces another set of variability that can increase the degree of bias. And just to highlight this group, I think many of the clinicians here are familiar, but these are children born with cyanotic heart disease and typically have saturations in the 70s and 80s rather than 90s, where most SAP probes have actually been well validated. And this is typically caused by a structural shunt inside the heart that redirects blue blood out to the body rather than sending it to the lungs where it gets oxygenated. On a historical note, the first blue baby surgery to redirect blood back to the lungs was initially performed at Johns Hopkins University and credited to Alfred Blaylock and Helen Taussig, but later was credited to Vivian Thomas, who was a research lab assistant who actually really had perfected the technique. And just as a side note, the story has been very well retold in the 2004 movie Something the Lord Made, which I would commend to people for their review. So at Stanford, we actually have conducted 2 studies in hypoxemic children looking at the accuracy of pulse oximeters and found that FDA pulse oximeters overestimate the arterial saturation in children with cyanotic heart disease very much in the way that's purported to occur in children with darker skin pigment. So there may be a couple of layers to the error that we see here, which needs to be troubleshot in this population. So in conclusion, we suspect the problem of pulse oximetry errors may be as prevalent in children as it is in adults, and this study will help to confirm it using a prospective data collection. We are working closely with both the FDA and the UCSF teams, which has really been wonderful in helping us to get up and running as quickly as possible to ensure our methodologies are harmonized so that we can pool our data across age groups and for analysis and make them available to the public for secondary analysis. And we do acknowledge that several factors may make the study harder to perform in children related to cooperativity, vasomotor reactivity, and cyanotic heart disease. This may require an adjustment to the sample size population and perhaps some troubleshooting of our methodologies along the way, but we have a great team and are certainly prepared for that. So just want to thank again the U.S. Food and Drug Administration, especially Nilsa and Michelle for their wonderful support and the rest of the team, and then the UCSF group that has really been exceptionally helpful in getting us up and running, and we really credit this study being where it is really because of their support through the UCSF Stanford CIRSE. So thank you very much.
Thank you for the opportunity to speak today. I'm Michael Lipnick. I'm an anesthesiologist at UCSF. I also co-direct our World Health Organization Collaborating Center for Emergency, Critical, and Operative Care, as well as our Center for Health Equity and Surgery and Anesthesia. I'm also an investigator in the UCSF Hypoxia Lab, which I'll be sharing a bit more about. Most of the focus of my talk today is going to be about a new project that we hope will accelerate and support improvement in pulse oximeter performance, especially as it relates to performance bias due to dark skin pigment. And at the end of the talk, I will invite collaboration and share information about how to do so. I have no conflicts to disclose. First, just a bit of brief background. Our lab was founded by John Severinghaus and has been doing healthy human study subjects of pulse oximeter performance for 50 years. We've studied thousands of subjects and hundreds, if not thousands, of devices. Most of the focus historically has been on device development and performance verification testing, though over the past 10 years, and especially the last few years, we've been working closely with a number of organizations operating at the intersection of pulse oximetry and health equity. Through pulse oximetry, there are many opportunities to improve patient safety and reduce health and healthcare disparities, not only in the US, but worldwide. Especially on the global scale, access remains a major issue and is worth noting, not just access to any device, but a safe and reliable device. We've worked with collaborators like Lifebox and the WFSA, as well as leading aid agencies and global health authorities to advocate and explore ways to improve access to pulse oximeters as an essential patient safety tool. More recently, the impact of oximeters and specifically poor performance in populations with dark skin pigment has become more of a focus for us. The issue of performance and impact on health outcomes have likely been covered adequately by other speakers. So I just wanted to share a couple of statistics highlighting the potential for oximetry to improve safety in this issue of access. About 30 million surgeries are performed worldwide without pulse oximeters. 70 to 100,000 operating rooms and intensive care unit beds worldwide do not have pulse oximetry. And for a large proportion of the world's population, access to pulse oximetry is not there, even in primary care settings. We're trying to, with all this in mind, approach the problem considering all of these objectives, trying to find a balanced approach that maximizes safety for all communities, but with special attention to populations at most risk. Now, during the COVID-19 pandemic, our team was inundated with inquiries to the lab, which have made us double down efforts and look for new strategies to accelerate work in this area. And here are a few examples. The skin pigment effect seems pretty small. Does this make a clinical difference? Should I be concerned? What methods are you using for skin pigment quantification in the lab? What is the best? What's the most reliable? What are the pros and cons of each technique? Do you have independent data that you could share, raw data on device performance? Have you tested certain devices, fingertip devices, handheld devices? Can you share those results? Maybe from another team, we figured out how to solve the issue of performance of bias in dark skin, but we need help verifying it. Can you help? And then finally, this example, during the biggest surge in South Asia, NGO was looking to donate a large number of units and asked if we could test several units rapidly for donation. One of the devices in the center you can see is sitting in the mid 80s when the real sat was 70%. This poor performance was very concerning, but even more concerning was the uncertainty around how this would be this information would be utilized, how it would be shared with the communities receiving the donations, and whether or not it would be impactful in changing the donor's mind. So collectively, all of these experiences highlighted to us the need to explore better ways to share data that we have, figure out how we could open up our lab infrastructure and make it more accessible for other teams, and also to cross-pollinate with other fields and disciplines that were approaching this problem in different ways. So this was the genesis of the Open Oximetry project, which we're launching thanks to multiple supporters, including catalytic investment from the Gordon and Betty Moore Foundation and the Patrick J. McGovern Foundation. The project has 6 components, and we're actively building collaborations in each of these core domains. I'll walk through these going clockwise, starting at the top left. First is the real-world clinical prospective studies. Equiox is the first of these, which you've already heard about today. This is supported by the FDA CIRSE and is a real-world clinical study at San Francisco General Hospital in adult intensive care unit patients, trying to better understand the magnitude of potential bias, as well as potential root causes of this bias. In the lab, we're exploring new protocols to better account for factors like skin pigment, but also low perfusion, severe anemia, other factors that we know that we hypothesize that if better accounted for in device development and regulatory guidance could potentially improve device safety as well as equity. We're also working on new techniques, in vitro techniques, phantoms, device testing products that could help us identify in less resource intensive ways poor performing devices and potentially some of the root causes for those. Now, for these 3 core domains that I've shared, all the protocols we are also sharing online and co-developing with other collaborators. We hope to be able to use these, the data collected and the tests to work with regulatory bodies and to test proposed frameworks and to better understand the impacts. The final 2 components of the project, and a large part of why I'm talking today, is our plan to create a mechanism for collaboration and an open data repository, both attempts to harmonize efforts and create transparency around pulse oximetry safety and try and accelerate progress on improving safety. The next 2 slides before I wrap up, I'll go into this in a bit more detail. So this network that we're trying to build for collaboration really is a mechanism for those who are already working in this space or perhaps starting to work in this space of improving pulse oximeter safety or improve access to pulse oximeters with an emphasis on the issue at hand, which has to do with performance bias and skin pigment. This will be a forum that meets regularly. We recognize that there are many efforts working in this space, unfortunately, often in parallel, and we're actively trying to figure out how we could leverage the strengths and resources of other ongoing efforts, acknowledge work to date, and to prevent unnecessary duplication moving forward. The forum is going to include international multi-stakeholder engagement. This includes diverse disciplines, geographies, industries, viewpoints, and populations. Again, trying to avoid duplicating, but rather be additive and complementary with existing groups. We're already actively working closely with the leads for the ISO-IEC Joint Working Group on oximeter performance. Some preliminary objectives for this forum include developing a research agenda for both regulatory and technology standpoints. We hope to not only identify potential regulatory changes, but also be able to test them and view data in real time with the group, as well as to account for international perspective and attention to communities at risk in the US as well as globally. The group hopefully will account for total product life cycle, design, distribution, utilization. This would include not just the performance, but also procurement guidance, as well as education and communication and implementation guidance. We recognize there are a number of entities working in that space and are, again, actively working, trying to find ways to work together across these different groups. And then finally, promoting clear and consistent communication, not only in the education piece and advocacy pieces, but also as relates to data collection, definitions, best practices for data, and doing all of this in a way that doesn't unnecessarily erode confidence in this essential patient safety tool. This forum will have its first of regular meetings in December of 2022. Through this QR code or through the Open Oximetry website, you can sign up or forward this to other groups or individuals who might be interested. And we welcome participation from anyone. So please check that out and share it. The final piece of the project that I'll be sharing about, again, in this effort to leverage multiple collaborations and stakeholders, is data sharing. We're building a couple of different mechanisms to try and promote data sharing. One is an online visual summary that would be more consumer oriented. This would show device performance data for a large number of devices that we'll be testing. Testing has already begun in the lab and will continue for the next 2 years. The second is a raw database. This will be raw performance data. Again, we'll be collecting these data in our real-world clinical trial infrastructure, but also in the laboratory infrastructure, and putting all of this into one data set where individuals who are doing research and development can access the raw data and query for the study questions that they want to ask. We also invite other researchers to contribute to both of these data sets, and please reach out and we can share more information about how to do so. Of note, the current site is a demo site, and although we'll be adding and updating to it continuously, a full new version of the site will be launched in January 2023. So with that, I'll conclude. I'd like to thank our multiple supporters, as well as the numerous collaborators here at UCSF and multiple organizations around the world who are contributing to this work. Thank you very much.
Thank you, Dr. Lipnick, and to all our speakers. Outstanding talks to everyone. I'd like to open up to the panel to see if anyone has any clarifying questions that they'd like to address to the speakers. We have about 15 minutes for this. I know once we get going, people will start asking questions. I'll ask the first couple. I was taking some notes or questions as I was going. It's a question to Dr. Wong, and I guess Dr. Shading can also answer it. For the this entity of hidden hypoxia with the increased mortality rates, I think you said it was 70% higher mortality rate. Who was the reference group? Was it other patients who were not hypoxic, or was it patients who were hypoxic, but it was evident? I think that becomes important because I don't know if the hidden hypoxia is just a surrogate for disease severity, or if the inference is if it was treated, then it would have made a difference in the outcomes.
Thank you so much, Dr. Nathan, for your question. With regards to the comparison group for hidden hypoxemia, the reference was, so all patients had a pulse oximetry value that was greater than 88%, as you well understand. But the reference group was the intervention group, or the case group was those with a hidden hypoxemia, so those with a arterial sat below 88. The reference was those with both a arterial sat and a pulse ox sat that were both above 88% as the comparator.
Because I guess that gets to my point that these were sicker patients, you just didn't know about it, and it would have been interesting to compare their mortality to other patients who were hypoxic but where it was known, to make the next assessment or the next inference that, gosh, if they had been treated for their hypoxia, they would have had a different outcome. I think maybe all you can say at this point is they had a worse prognosis that was unknown because their O2 sat was falsely elevated.
Correct. And so I believe that perhaps Dr. Gottlieb or others in this who were working on perhaps causal inference techniques, right? As we and many understand, this is an inherent limitation to retrospective analysis. But the best we can do, I believe, is through causal inference techniques, as I'm sure he'd be willing to talk about.
Now, it doesn't diminish the importance and the impact of the study, but thank you for clarifying that. Okay, Dr. Yarmus, you're up next.
Hi, thanks. So I think this is probably directed at Dr. Almond, but open for anything. So as an intensivist, I think most of this has been focused on critically ill patients, given probably the access to arterial blood gas monitoring. But clearly there's some historical data out there, like the study that Dr. Almond had quoted from Harris for the, looks like outpatient oximetry monitoring and less severe illness patients. So I'm just curious, probably for everybody, are there working solutions out there where, this, for example, in that study, you have outpatient patients being monitored. So have there been workarounds or any types of parameters that clinically has been utilized to help sort of adjudicate some of these issues? And that also transcends beyond other diseases like sleep medicine, for example. Has this been recognized previously? And are there any efforts, for that patient population, interstitial lung disease, 6-minute walk testing, kind of all the more geared towards, I suppose, an outpatient monitoring program?
Thank you very much for the question. I think, can you hear me okay? I think, most of the attention so far has really been focused on describing the scope of the problem. And I think, like many things, we're always a little bit behind in the adult world. I would say that in the study that Bronwyn Harris did, 3 different oximeters were tested, the Nellcore, the Masimo, and one called the Masimo Blue, which is kind of appropriate, I guess, for blue babies. And that one actually did have relatively good performance, or better performance, I should say, relative to the other 2. So in some ways, there's some sense that there might be some oximeters that perform better. You're right about the outpatient oximetry study, which I didn't mention, but was looking at kind of the home monitors. And I think has been pointed out elsewhere, this is a very large business in pediatrics with kind of worried families related to SIDS and other things, where these over-the-counter machines are used quite frequently. And I think, as the point was made before, they're not being used kind of for casual reasons, but really for health monitoring. The results were relatively similar in that there was still this bias in the bluer kids. I think what we're hoping to do in the context of the prospective trial is to sample both those kids that are kind of coming in for elective procedures and are quote unquote wellish, although they have heart disease or other reasons that they're getting cathed, as well as patients in the cardiac ICU and those undergoing cardiac operations. So hopefully we'll have the span there. I think some other people on this panel and some of the distinguished speakers have more experience in terms of monitor development, and I would defer to them.
Thank you, Dr. Almond. You were a little bit soft there, but I think everyone heard that what you had to say. But just in case you ask another question, if you can just get closer to the mic, please. Dr. Lowe, why don't you go ahead?
I have a question for Dr. Wong. One of your slides showed the absorption spectra of water and of melanin. And I wondered if that was accurate, where that spectra came from, because I noticed that they're quite similar, melanin and water. And if that's true, I'm just trying to get my head around the difference between laboratory findings and the clinical studies, and how it can be that melanin makes such a big difference, especially if it has a similar absorption spectrum to water.
Thank you so much for your question. In reference, I believe that was from was Baxter. Was a therapeutic laser reference, but it's one that would actually examine the optical properties of tissue. And that's where that came from. In terms of the reference of melanin to water, I think the question, so this, maybe I might ask or maybe rephrase your question. There are effects of melanin and water, and to some extent we have compensated for water and the presence of water in the design of pulse oximetry. But one of the differences that we may not always control for is many people have different, although the spectra can overlap, the concentration and as you may differ, and that as the concentration of melanin differs, right, and is, I believe, inversely proportional to the or the concentration, right, and the force, etc. The Beer-Lambert might dictate how much effect it may have on the resulting spectral comparison.
Okay, yeah, certainly concentration makes a difference. I have one follow-up question, if that's okay, for Dr. Bickler, because I thought that there was a very well described how, again, the lab studies and the clinical studies could have very different results. Perfusion and therefore the signal to noise ratio obviously is very important. And I notice that's one of the variables that you're looking at. And I think that's great. I wonder about the ratio of DC absorption at the two different, at 660 and 990. I'm sure I got the numbers wrong there, but the red and infrared, the DC absorption, if the ratio of those has been looked at as an explanatory factor, and if that could be, put in the model. And Dr. Lipnick, I think, at the hypoxia lab probably knows the physics and how this all works better. I just wonder if that's been looked at as an explanatory factor.
Thank you for that question. Yes, the ratio of the pulsatile and non-pulsatile light signals in the red and the infrared forms the basis for pulse oximetry. And as I mentioned, all pulse oximeters need to be calibrated in humans in order to translate the optical signals that's seen by the electronics to a saturation reading. And you can imagine that if all the calibration procedures are done in subjects with low skin melanin, you produce one R curve. That would produce a pulse oximeter that would be accurate in individuals with lightly pigmented skin. And what has become apparent is that it's been insufficient to account for the presence of melanin. Now, you could do another calibration for subjects with darkly pigmented skin, and you would get a different calibration curve. So that is possible. And almost 20 years ago, we advocated for something like that. But something that's really important to point out from our recent work is that it is more complicated than just skin pigment. Because our work on low perfusion shows that there's a third factor, which is incredibly important, and that is the perfusion index. So if you don't account for that, the simple correction for skin pigment alone would be insufficient. So that represents both I think, a fundamental discovery and also an engineering challenge to incorporate multivariables in the way that pulse oximeters determine the saturation value.
Thank you. Let me move on to Dr. Collett.
Yeah, I wanted to keep going on that subject, Dr. Bickler, if you don't mind. Can you explain, I guess, how you calculate the perfusion index? If you said, and I missed it, I apologize. But, and where you also, I guess, I was trying to understand the relationship between dark skin and the perfusion. Are you suggesting that people with dark skin have low perfusion, or that that's just a complete other variable in evaluating these oximeters?
Thank you for that question. That really gets at the heart of what's going on here. So first of all, with respect to understanding perfusion index, you can think of it as the pulsatility of the blood. because the pulse oximeter requires seeing pulsatile blood in order to estimate saturation. It's based on a ratio of ratios of pulsatile to non-pulsatile blood at the red and infrared wavelengths. So if there's no pulsatility, it can't determine that ratio, and it will produce a wildly inaccurate estimation of saturation. Now, skin pigment, melanin absorbs much more red light than infrared. So the effect on that ratio of ratios is to shift the ratio to lower values, and that means higher saturation. So you can understand how absorption of red light would shift the calibration curve to the right and produce an abnormally or an inaccurately high saturation value. So that's really the essence of it. So think of perfusion index as just the amount of pulsatility of blood in your finger. Now think about sick hospital patients. They don't have normal perfusion. They're presenting with all sorts of illnesses that make them have less pulsatility. They could have low blood pressure, they could be in shock, they could be dehydrated, they could be critically ill. There are numerous factors which produce decreases in perfusion in hospitalized patients, especially those in the ICU. So it's easy to imagine that in a population like that, pulse oximeters would face much greater challenges to reading accurately. In fact, you can imagine how that would bias their readings toward artificially high saturations. It's informative to think about some of the other retrospective clinical studies. We heard from Dr. Burnett at the University of Michigan. He studied patients in the operating room. It's our clinical experience that anesthetized patients actually have higher perfusion because of the vasodilating effects of the anesthetics. What he found was that the errors were sort of intermediate. They weren't as grave as those identified by Dr. Schoeding, Dr. Fawzi, Dr. Wong, Valbuena, and all the others. And so that fits with the idea that perfusion is a critical factor in affecting the performance of oximeters.
Thank you. Mr. O'Brien.
Yes, thank you very much. Joe O'Brien, patient representative, National Scoliosis Foundation. Another question for Dr. Bickler, following up. As I said earlier, ever since I read that there was only 5% melanin in the finger, I was very confused as to what the causative factor is with skin pigmentation. And as I listened to your presentation, as we just discussed, I was thinking, okay, perfusion, that makes sense, that may resolve it. One of the studies I had read, I was just wondering if you could address it, was Thyssen and his group in the Netherlands. And they looked at critically ill people in the ICU and had looked at perfusion index versus the gap between that we're talking about. And they found that there was no correlation between it, that they could not use it for any indication whatsoever. I was wondering if you could just address that. And
I'm sorry, I'm not familiar with that study. But I would say in general that low perfusion produces errors in pulse oximeters. Anybody who uses them clinically knows that. And in fact, clinicians often have to change the site where we're performing the oximetry measurement. We often use the ears or the nose or other places on the body if the oximeter is not producing a good signal from the fingers. So this is a common part of the clinical experience with pulse oximeters in general.
Okay, thank you.
That was going to lead into my question. Actually, in the context of the EQUIOX study, are you looking at just one site, or are you comparing sites like a finger probe versus an ear probe versus a forehead probe? And does that help with the perfusion aspect of this? And I don't know, in individual patients, the perfusion in these areas might be a little bit different.
Well, that's right. The EQUIOX study is designed to capture whatever is the prevailing perfusion index. And some of the patients that we've already enrolled have ear oximeters because the oximeter has not been working well on the fingers. So in the course of the study, we're going to capture different sites.
and different levels of perfusion. And we will incorporate that into our analysis. Now, that complicates matters statistically because our study design is confined in time and space. And the FDA wants an answer from us by next summer. And I think that some of these detailed questions, like the one you posed, perhaps will not be answered in that frame of time. We'll have some ideas, but probably no firm conclusions.
But wouldn't it be easiest if you just try to get 3 readings on the same patient once you have them consented, because that might enable you to answer additional questions rather than one patient earlobe, another patient the finger?
We are actually doing that. We
Okay.
are primarily focusing on standard clinical care. So we're not walking into the patient's room and saying, oh, can you please switch the oximeter? Because what we want is a real world clinical study. We want to understand what's happening in real clinical environments without that kind of interference from the study.
Sorry to keep you on the hot seat, but being that it's real world between the 2 centers primarily, is it going to be a standardized pulse ox that's going to be used, or it's whatever's on the patient?
It's what's ever on the patient. And this is one of our goals in expanding clinical sites where these kinds of studies are done. We're sharing our protocols on open oximetry. We're inviting anyone, anywhere in the world to adopt these protocols and to incorporate their specific patient populations and clinical environments into the information that will really benefit a more global understanding than just 2 centers, like you've said.
Right. No, I think that's valuable. Dr. O'Connor.
So yeah, I want to go in again, a question for Dr. Bickler. So there's literature that suggests, for example, that there may be a difference in accuracy between the finger and the ear in people with heart failure. And you can imagine that heart failure is more prevalent in some populations than in others. And so I'm going to encourage you to prospectively get as much data as you can, not just about the patient's demographics, but also their comorbidities, because particularly with the heart failure, I mean, it's a small study, but it's a real signal, or it seems to be a real signal, and it would be worth exploring on a larger scale. Because just like the perfusion index, if we can say, if your patient has heart failure, put it on the ear, that would be very useful guidance to providers like me.
Thank you, Dr. O'Connor. That's an excellent point. As you can imagine, we are recording a wealth of demographic and medical information about all the patients in our study. I think maybe Dr. Allman might want to comment about that, because he has a population where there's a high degree of cardiac problems, and he alluded to some of the challenges related to heart disease. Maybe he could provide some additional comments.
Sure, Dr. Allman, if you want to weigh in.
Sure. Can you hear me better now?
A little bit closer would be helpful, I think.
Yeah, I think, we do find in pediatrics that we need to oftentimes move the probe around. A lot of the kids that we have that are blue, some of them are quite healthy running around clinic and at school, and then others are critically ill in the ICU with low cardiac output or other perfusion problems. So it's such a common thing to do in pediatrics, to basically rotate the probe around until you find a signal. And I can't say that we've seen that especially true in heart failure patients, pediatric heart failure patients, but that certainly is a common practice. Thank you.
Sure. Dr. Klein.
Yeah, I have more of a comment than a question. I really, for Dr. Allman, as we've talked a lot today, we talked a lot about adults and a little bit about neonates. So I just wanted to thank you for the pediatric perspective as a fellow pediatric person, and thinking about those kids with congenital cyanotic heart disease, and our patient population that probably has skin thickness somewhere between the neonate and the adult, and so all of these other confounding pieces. And so I just wanted to say I look forward to the results for your pediatrics work.
Thank you for that comment.
Thank you, Dr. Klein. I just have an additional comment I'll give. other folks time to think if they have any last questions they want to ask. But as you're talking about the pulsatile flow, it's not an aspect of pulse ox that are fully understood or appreciated, but there are populations. I think what has been alluded to already are heart failure patients who are on vasodilators. We have patients with pulmonary hypertension who are on very potent vasodilators, and we base therapy based on their pulse oximetry reading sometimes. And then another population of interest would be LVAD patients with left ventricular assist devices who don't have any pulsatile flow. And I'm guessing, I hadn't thought about this, you can't get a pulse ox on those patients then?
That's right. You're right. You're generally out of luck. Sometimes you'll get a little bit of a signal on an LVAD patient, but I would most of the time I think they're highly unreliable.
And even patients who are on VA ECMO, depending on how much pulsatile activity there is, I should imagine that affects the signal as well, the sickest of the sick patients.
That's right.
All right, Dr. O'Connor, your turn.
So with the LVAD patients, if the blood pressure cuff works, the pulse ox is probably not bad. If you can't get a blood pressure on them, you probably should just take the pulse ox off the patient. That's kind of how we take care of these. And I want to ask Dr. Almond a question, since I was inspired by the other question about pediatrics. What are you going to do about hemoglobin F in your very young patients? Is there a way that you're going to deal with that in your data collection?
Yeah, that is a great question. My general sense is that is that most of the studies that have been out there have not found that the bias seems worse in that population. A lot of the kids that we see do have significant congenital heart disease, so their saturations are abnormal, especially in that newborn period. We are just launching this study, and it may be that we want to consider collecting information about hemoglobin F. I think at the same time, most of the cardiac anesthesiologists here have said that they just haven't really kind of empirically observed an issue related to that. So I think that's something we should definitely take under consideration.
Dr. Kapotic, you can't ask a question as a speaker, but if you want to pass a comment in response to one of the other questions, please go ahead.
Yes, I do, and it was related to the neonatal issues where a preterm may have some level of fetal hemoglobin. That was well investigated in the mid-80s and shown not to have an effect. However, in Dr. Almond's condition, where there are cyanotic heart defects, polycythemia can be an issue. And with polycythemia, there's reduced flow to peripheral sites. So some of your challenges in placing a sensor and getting an adequate value, or at least a signal sufficient to give you a derived saturation, may be associated with that poor peripheral perfusion, much like a sickle cell child not being able to get the hemoglobin to flow well because it is so thick in the polycythemic state. And then just in follow-up to the ECMO situation, having been an ECMO coordinator at a few hospitals, it is very much the case in VA ECMO that you will not get an adequate signal. And again, no saturation can be derived by a pulse oximeter. But that's also the case on any patient that's on cardiopulmonary bypass in the operating theater.
Okay, thank you very much. Dr. Hennessy, will make yours the last question before we close out the session.
Great, thank you. So this is a question for Dr. Schoeding. I'm wondering if any of the higher rates of occult hypoxemia in Black patients compared to white patients is due to a higher prevalence of hypoxemia in Black patients versus white patients, or if it's all due to the pulse oximeters functioning worse in that population.
Thank you for that important question, which was also discussed, I heard earlier, among the statistical analysis. So the occult hypoxemia rate, also known as the false admission rate, can be influenced by the prevalence of hypoxemia. And so if, as a population, a group of patients had lower or higher prevalence of hypoxemia, that would affect the result. That's one of the reasons why we did the additional analysis by looking at receiver operating characteristic curves, showing that there was still a signal. I would want to make one other point about this, which is we heard from others that in hospitalized patients, the rate of pulse oximeters, SpO2s, being low is extremely rare. So basically, once a patient comes to the hospital, the oxygen saturation is immediately titrated to get the pulse oximeter to a normal range. And we heard from Dr. Gottlieb that, everyone titrates oxygen to pulse oximeter. So what it means then is that if there is residual occult hypoxemia, it's probably more likely related to the fact that we're not actually titrating oxygen to the SaO2, which is low. We're titrating oxygen to the SpO2. So I think in general, that's the more likely mechanism here, rather than a difference in baseline prevalence of hypoxemia.
Great. Can I ask one follow-up question?
Sure.
Sure. You noted that there was a lower use of arterial blood gas in Black patients and white patients. Do you have a hypothesis as to why that was?
Yes, that was one of my colleagues. I can't remember who it was.
Sorry.
That was Dr. Wong.
Okay, Dr. Wong?
Yes, thank you. So we were unable to distinguish why this happened. So rather, in short, in terms of adjusting for the risk of, for example, like for SOFA scores or dysfunction, for other patient characteristics, we were still unable to show a difference in the underlying population. So in short, we don't have a clinical reason as to why this happened that would otherwise explain it, besides some of the other factors that we noted here.
Okay, thank you.
Okay, thank you. Dr. Gottlieb, I see you have your hand up. You may answer any of the questions posed by the panel, but please don't add any new questions. You muted. Was your hand up inadvertently?
Oh, I'm sorry. Thank you. Yeah, I just wanted to follow up on a few of the comments that were just made in response to Mr. Hennessy and Dr. Schoding's comments. We also showed in our table 1, and this is somewhat generalized data because it's just a single median with a Wilcoxon's test. But we did show overall lower SaO2s and higher SP. So that Black patients had lower SaO2 than white patients, but had higher SpO2. So overall, I don't think the main effect is that the Black patients were more hypoxemic. And one other thing that I wanted to mention in response to what Dr. Schoding had said was in terms of oxygen titration. There are a couple of papers and some work underway, though I don't know if any of this is actually in the clinic yet, is devices that would auto-titrate supplemental oxygen rates according to SpO2. So I think it's really important to stress that with these new sophisticated technologies, if these disparities become very much baked in and could really be amplified with devices like this.
Okay, thank you very much. And that brings us to the end of this session. It is now 2:44 on my watch. So we'll take a 5.5-minute break and let's reconvene at 2:50 p.m., please, Eastern Time. Thank you. Welcome back, everyone. This next section is going to be devoted to panel deliberations. But before we get to that, there was a question this morning that was addressed, that hasn't been addressed, that I'd like to ask Dr. Lee from the FDA to answer.
Thank you, Dr. Nathan. Yes, I believe the question came from Dr. Loeb regarding whether devices are on market cleared that simultaneously measure skin pigmentation and just. So while devices and LEDs can adjust the intensity, it does not do that actively, meaning that they're basically a finished device that FDA reviewed. The intensity levels of the LEDs can vary. That is something that a medical device developer would choose and make through design specification, but they don't actively adjust due to any perceived differences in pigmentation or other adjustments. Hope that makes sense. Thank you.
Okay. Yeah, that was mine. Thank you, Dr. Lee.
Oh, thank you, Dr. Connor.
Thank you, Dr. Lee. We'll now begin the panel deliberations. Although this portion of the meeting is open to public observers, public attendees may not participate except at the specific request of the panel chair. Once again, that'd be me. Additionally, we request that all persons who are asked to speak identify themselves each time. This will help the transcriptionist identify the speakers. Let us begin. Do any panel members have a question or comment to discuss? And panel, please turn on your videos if you don't have them on already. Unmute your computer when you speak. Raise your hand. I think everyone has the hang of that with Zoom, and I will call on you in turn. So we have up to 2 hours for the deliberations. Let's see how it goes, and looking forward to a lively discussion from our panelists. Dr. Alam, I see you have your hand up first.
Thank you. My name is Murad Alam. I have a couple of comments to make. I'm a dermatologist, so unlike many of you, I have limited expertise with using these devices in a highly acute setting, but there were a couple of things I thought I might be able to contribute. The first was regarding pigmentation. I just wanted to give some background. I'm a dermatologist. I'm also a laser person. I'm the president-elect of the American Society of Laser Medicine and Surgery. I do clinical research in that area. We, in fact, have devices here. I went and looked at one in one of the breaks, an IPL device, which does have a mechanism. It's used for skin treatment of red and brown dyspigmentation, and it has a mechanism for doing what I think Dr. Connor was referring to, where it picks up what the person's skin type is and then adjusts the frequency adjusts the filters and the fluence of the IPL so that it doesn't burn someone, for instance, with a darker pigment. So those sort of devices do exist. They're relatively crude. But I also wanted to raise the issue of the precision of pigmentation. I think FDA was giving us a description of devices that are very precise in measuring pigmentation, and they were being somewhat skeptical about the Fitzpatrick rating scale, which is, of course, subjective and somewhat imprecise. But I did want to raise the issue that we might be trying to measure precisely, at least hypothetically measure precisely, something that is inherently not precise. So I think there's a danger of having a very, very sensitive instrument for measuring something that varies dramatically in the same individual, given the time of day, given how it's applied. So for instance, I'll give you an example. Some Asian people are very photoprotected just because they're out of the sun a lot, but they might go to the beach one day. And as a consequence, their pigment can vary quite dramatically from time to time. So unless you were having a device that was ascertaining this every time, very precisely, I think you'd have to be very cautious in trying to slice something very finely when really it's big categories, and that's probably as accurate as you can be. So that's one observation. In terms of the devices, I think there were a couple of types of devices that were considered. One would be something that would have an algorithm. I think Dr. Connor again referred to this, where it'd be kind of working the same way, but it would use information about your skin type to somehow adjust the number that was the output number to compensate for that. So I think if you were doing something like that, that might sound a little bit more feasible. I think if you were actually having a device that had different mechanisms for everyone with every single different kind of skin type. Just based on what little I know about how these devices work, that's a very complicated optical imaging device. I do some optical imaging research too, and to have that in a relatively small box, I mean, some of the stuff we have, costs several $100,000 and fills a room, and I'm just not sure those would be very robust devices if they had 15 different laser frequencies embedded in something and some way of deciding which 3 to use based on fine distinctions and pigmentation. So again, I think the algorithmic mode makes more sense, and I don't think you should slice pigment too finely. I think you'll get into trouble. The other comment I had, but I'll try to be brief, was pertaining to the many devices that FDA considers non-medical that are available at CVS or Walgreens. And I think much of the discussion, the research that's been done today and presented today, rather, has focused on devices for use in the hospital for patients who are very ill in some cases, who are on ECMO or LVADs, etc. But I think we shouldn't forget that a very large percentage of the population comes in contact with those inexpensive, quote unquote, non-medical pulse oximeters that they buy in department stores and grocery stores and pharmacies, and they rely on those to even decide whether or not they should present to the physician. So I think we have to really think about those as well. There's a risk of ignoring them, and there's also a risk of saying, well, those are so bad, let's just stop making them, which I fear might be throwing out the baby with the bathwater, because they're providing some information to people, and it might be a matter of trying to figure out some way to make them not as dangerous. Maybe instead of giving a number, it should give a range, 91 to 96 or something like that. But some way to also think about those devices, because while they're not maybe what you all who are very, very smart are dealing with, a lot of us who are lower tech people, that is what we deal with. And I can speak from personal experience. I have skin of color. I had long COVID. I relied on those things a lot, and that's what we have access to. And I think there have to be more safeguards there, because you all who are intensivists are highly sophisticated and able to compensate for the errors in your devices in the hospital just based on your own intelligence and experience. But the average user isn't. And what you don't want to do is either make those devices go away or make them or improve them so much, the common man's devices, that they get out of the reach of the average person, right? I mean, if we made them much better and each one cost $1,000, there'd be an access issue where people really wouldn't be able to get a pulse ox at CVS anymore. Thank you.
I think you make a lot of good points. I want to give other folks a chance to weigh in, but just to pick up on what you said, I think obviously an objective assessment of skin pigmentation is key. Your point's very well taken in terms of complicating it and making it too complex and having different devices for different folks. I don't think that's going to work. And what you alluded to, and I'm just not sure where the technology's at, is some kind of internal device that measures or gives some measure and corrects for the pigmentation that it senses. I think it also is helpful if you have a patient who you put it on the finger and you're not getting a good read, and then you put it on the ear and you get a good read there, and the pigmentation might not be the same. But if you have some kind of internal correction through available technology, I think that's probably going to be the best way to go. I don't know if anyone else wants to weigh in on that particular aspect of pulse oxes and where the future should go. And then we'll talk about the consumer device as well. That's a whole other can of worms. I think they're out there. You can't stop them being out there. I don't think I've ever bought one over the counter. I should imagine they come with a big warning, but like anyone else, you tear open the box and you throw away the warning, and then you're using the pulse ox and you're taking the number for what it is. I think that's natural for most people. I don't think that there's actually a warning of any sort on the device itself. Anyone else want to weigh in on the pigmentation issue and how best to go about evaluating that? Dr. O'Connor, I see you next. I don't know if you want to stay on topic or if you had something else you wanted to. I think maybe we should try and just, rather than jumping around, let's stay on topic for now and everyone have their say about.
So Beverly raised her hand in response to your query.
Oh, who was that? Ms. Edwards, did you want to comment on the pigment issue?
Well, I wanted to comment on the over-the-counter.
Okay.
oximeter, because so my mom recently passed, and she had heart disease, and she had an oximeter at home, and this was in May. And the morning that I called the ambulance to coming to take her to the hospital, I remember that I don't remember what the oximeter read, but that wasn't what caused me to call them. It was the blood pressure reading, and it was her obvious labor breathing. And we did have that oximeter, and we did check her. And I guess my question would be, or my concern would be that, yes, I do believe that that over-counter oximeter should be reevaluated too, and maybe come with some kind of warning, or someone suggested earlier, maybe it should be behind the counter in the pharmacy lane, the use, and what the consumer should consider as far as the readings.
Yeah, first of all, I'm sorry about your loss. It's always very difficult losing a parent, so my condolences to you on that. You bring up another very important aspect, that low oxygen is not the only cause of shortness of breath. And I think for the consumer, they shouldn't get faked out by thinking, oh, I'm very short of breath, but my saturation reading 92%, therefore I'm okay. There could be many other things going on that require a trip to the hospital. So I'm not sure if consumers are lulled into a false sense of security by that. Thank you for that comment. All right, Dr. O'Connor, over to you.
So I'm going to summarize a lot of what I think for the group here. So first of all, I think that the ARMS of 3% is too permissive. I think we should instruct the FDA to think about using an ARMS of 2% or even 1.5%. The second thing I'm going to say is that it sounds silly, but many of the studies done to validate these devices, all the data is north of 92% saturation, which is frankly a domain in which the differences make no difference with respect to clinical decision making. So I'm going to say that they need to have a statistically significant data set between a saturation of 75 and 92%, because that's the domain where pulse oximetry will in fact inform changes in decision making. The third thing I'm going to say is, as I've already said, they should actually ask people to do phase 3 trials rather than try to subject volunteers to ridiculous hazards. We should in fact instead conduct phase 3 trials in ICUs and in operating rooms where we're really looking for data from patients who are hypoxemic. And we have very precisely matched specimens from a blood gas that's contemporaneous with a pulse oximetry reading. I don't think you're going to solve this problem with 2 LEDs. I think that the only way you can, in fact, make forward progress on it, both with respect to dealing with the pigmentation and, frankly, getting a better signal, is to, in fact, add LEDs. You know, co-oximeters don't have 2 LEDs. They have multiple LEDs. And I think some of our problem is methemoglobin, some of it's carboxyhemoglobin. And, frankly, these devices need to perform reliably in the setting of anemia, which is also another as yet kind of recognized confounder of this data. And I think that, you know, they need to think very seriously about a standard that is derived from data in patients with multiple LEDs. And the other thing is, I think they need to do better signal processing on the signal they get, particularly for things like venous pulsation in patients with pulmonary hypertension, which is an increasingly prevalent problem in the society. And finally, speaking as an anesthesiologist and an intensivist, designs that mitigate the influence of ambient light on the signal, both in adult and pediatrics, and designs that mitigate RF interference from pulse oximetry, which is a huge problem in the operating room, would be hugely helpful. And finally, displaying SQI, signal quality index, and perfusion indices prominently alongside the pulse ox, and perhaps some sort of guidance for end users in the clinical domain would be very useful to front-end providers like myself. So I think that's a pretty good summary of all the things I wanted to say. One last thing. With respect to ARMS, the other thing that the manufacturers should provide us with is a positive predictive value at basically every point between 92% and 77%. So if the pulse ox reads 77%, what's the positive predictive value that this patient's going to have pulse ox reading saturation that's within an arterial blood gas saturation that's within 2 or 3% of this?
Yeah. I think you make a lot of valuable points and summarize a lot, perhaps, of what many of us were thinking in terms of really needing to tighten it up in terms of the arms. And I don't know what the right number is, 1% or 2%, but clearly when you look at those Gaussian distribution curves and you recognize that even if it's just 2% of patients with the, who fall below that 88%, 2% of thousands, tens of thousands of readings is a lot of patients. So that really needs to be tightened up. I don't know, to your point, rather than getting a single number, I don't know if the Gaussian distribution somehow could be shown. This is where we think you are, you're 94, but there's a 10% chance you could be 88. Just to give people an idea of a spectrum, that it's not an absolute number. And your other point, I think that you made and I reinforced in the earlier session, is these healthy control studies are like phase 1 studies of medications, and then you go on to phase 2, and then you need the phase 3. So I really think that the FDA needs to, before they endorse any of these devices, needs to have, call it real world, but it'll be well-controlled studies in sick populations, a diverse population of various disease severity, various illnesses, to validate that, yes, to your point, they perform well over 92. That's where all the data lies. But what about less than 90? What about going into the 70s? How do they perform there? Because that's where the interventions start. And it's interesting to me that the interventions that start when they desaturate all have to go through very rigid phase 3 clinical trials. You look at remdesivir, whatever disease entity you want to pick, putting patients on inotropes. So even though it's not just a device, very important decisions about medications that come under much greater scrutiny are going to be implemented or not based on the reading of these devices. So your point's very well taken. Thank you.
And I want to also point out that, when patients are profoundly hypoxemic, I mean, if somebody's 87, you can wait for a blood gas. But if they're 55, you're going to act, right? And so the idea that it's not a diagnostic tool is, in fact, not really correct in the clinical context. The reality is that if the SAT's 65%, I got to do something.
And to the same point, they might be 85, but you're going to watch them because you don't want them to be 75 in an hour's time as well. So, the trend and the accuracy of the trend becomes very important. Dr. Wilson, you are next up.
Thank you. First, let me say I agree with all the points that were just made by Dr. O'Connor, but there are a couple additional points that I think, so I'm not going to amplify on that, except one regarding the ARMS, that just to emphasize that it's critical that not only do we narrow it, and the FDA is going to have to determine, is it 2%, is it 1.5%, but it's important that continues to be within that range in the important portion of the oxyhemoglobin dissociation curve. So when we're in SATs between 70 and 80, just as well as between 80 and 90 and 90 and 100. So that was just that emphasis. The other point is related to how we evaluate skin pigment and what the percentage or number of patients required for studies. So currently it's 15% or 2 subjects, and that number needs to increase. The FDA needs to determine how much, but I would say we need to at least double so that you can get data that is going to cross more bins in terms of skin pigment. And now a related point to that, in addition to increasing the percent and number of dark skinned patients in the subject pool, is how do you evaluate that? And I think Josh Pfeffer did a very nice job early in reviewing how do we look at skin pigmentation. And clearly a quantitative methodology that takes the viewer out of it is optimal. But I do think that we need to have some sort of a Fitzpatrick or a Massey scale or Von Luschen scale also, as follows. In the laboratory data and in the data that goes to the FDA, quantitative data with some sort of a corresponding table is important. But in the clinical space, if we're going to give some guidance to individuals, they're not going to have, at least for some time, quantitative measurements on patients' skin. They're not going to have a spectrophotometer necessarily. So it would be helpful for us to have that. And so I would just like to explore a discussion about which of those three that have been mentioned a bit, the Massey scale, the Fitzpatrick, and von Luschen. The von Luschen is nice. It has lots of gradations, but the trouble is when you look at it, as you go from 1 to, it's I think 36 as I recall, sometimes you go into increase in tone and then you have a drop in tone. And so it's mixed. It's a little bit problematic. And with the Fitzpatrick, the problem is that there's not enough gradation and that it's overemphasizing the light skin. So from that perspective, and I don't know if Josh is still on or if he wants to maybe show up in the image, I could bring it up for the group of the three, the three that we mentioned, that the Massey does a better job. It's not perfect, of going from light to darker to darker to darker with 10 bins, so that you can then better differentiate where those patients are and perform histograms and ensure that those patients that may be in a certain ethnicity also have a spectrum of color, skin color subjects that is appropriate.
Thank you for those points. I just want to build on some of what you said, and that 15% number, I think, maybe is historical and derived from the proportion of the population that is minority. I'm not sure where it comes from, but is that the right number, especially when that 15% is potentially so different in terms of, where they're at and what their pulse ox will read in relation to everyone else who is more lighter toned. And for those 15%, it's 100% when the pulse ox is put on them. So I would venture to say that there should be equal representation so that they have equal chance. Anyone of a different skin tone has an equal chance of having as accurate measurement as someone of a lighter tone, rather than 15%, because that's what the proportion is.
Agree. Could we have some discussion on maybe comparing those charts that Josh Pfeffer mentioned? The Fitzpatrick scale was determined initially as a way of determining how people would become sunburned in the first three, and in some cases, even the fourth version of scale, it goes from 1 to 6, but 1 through 4 is more in light skin, and only in 5 and 6 is dark skin. It doesn't seem to really cover the spectrum. And
I just want to give a word of caution about the scales, which can be subjective. And the more categories you have, the more opportunity there is for discordant reads and less chance for concordance. And there's the kappa statistic. And I think if you go from 5 to 30, there's going to be less concordance. And I don't know from Dr. Alam, our dermatologist, if anyone's done a study looking at kappa concordance, assessing skin coloration, because I don't know if it's standardized that you look in this area, that area, people might look different, and it looks a little bit different there versus here. And I think that really where it needs to go is, some kind of more objective, some of these technologies that were mentioned, rather than a visual scale. Anyone else care to comment on that?
I'm happy to comment.
Dr. Alam, yeah.
Yeah, I think the concordance that you said, those sort of studies really haven't been very scientifically done, in part because the Fitzpatrick scale, as someone said earlier, I think it might have been Dr. Wilson, is not really meant to be based on skin color. You're supposed to ask people how they burn or tan, under what circumstance. It's more about the reactivity of the skin and the color of the skin. Two people could have skin of identical color and actually have different Fitzpatrick types. However, in common usage now, it's sometimes also used as a scale not pertaining to sunburns, but just pertaining to what skin color people have. I think one other issue of complexity that you might wish to consider is that color and ethnicity are actually different. And so you could have two people whose skin is the exact same color. One is maybe dark-skinned Filipino Indonesian person, one is a light-skinned African American patient. The color number might be the exact same, but the structure of their skin, its ability to transmit light, its likelihood of reacting to sun or getting an injury might be quite distinct. So I wonder if really when we're working on this, we might need to expand our concept beyond just color, right? Because really what we're talking about, the reason we even care about color is somehow whatever is going, whatever LED is transmitting through the thumb or the ear or the forehead is not getting the same signal because the color varies. But I suspect the thickness of the skin, the density in which, I don't know, the keratinocytes are packed together, a variety of other factors could probably also affect transmission. So I just think we have to be careful there, that even if we came up with the world's most precise colorimetry, we still wouldn't take into account, for instance, ethnicity, anatomic site, a variety of other elements.
Thank you for that. Dr. Wilson.
Yeah, I don't want to monopolize, but I just wanted to respond to your really good point about some problems with concordance, and if you have too many choices, people are going to make more mistakes. The one point that maybe I didn't articulate as well is with the Massey is that of the of the Fitzpatrick, of the 6, 4 of them are light skinned and 2 are dark. So you're just skewing the data one way. And so if there was something that maybe was an 8-point scale, but it was just more graded and uniform the way that the Massey scale is fairly uniform, that has some attractiveness. But I do agree that a quantitative methodology is going to be essential for FDA's. I would hope the FDA would require that for future submissions. Do you not also though agree that initially there should be some correlation between the quantitative scale and some nicely graded version of a color categorization?
I think that would be a good study to do, just to see how these different scales perform. Dr. Casera, I see you have your hand up.
Yeah, I'm going to be a contrarian. I'm going to say self-identifying what your color and race is has generated pretty good data, and we have a meeting from the FDA around it. What I'm worried about, and this speaks to the fact that different skin tones from different races, ethnicity may also matter. We don't know that yet. So jumping right to asking the FDA to use a scale, whether it's a visual scale or a device that measures your melatonin level, and assuming that that's going to translate into some of this disparate data we're seeing on pulse oximetry is really a leap. And I think we're doing a pretty good job having people self-identify themselves what they are in terms of skin color and ethnicity. And I think we should stick with that until there's studies that show that that can correlate and you can put it into a numerical value or look at a Massey scale or Fitzgerald scale. So I'm going to be a contrarian and say, I don't think we have that data yet to translate what the data's been driven, that there's disparities in pulse oximetry in certain populations. And now we're taking the leap that we can pick those populations out now using a numeric scale and even a visual scale.
Let me be a double contrarian and say, do we need a scale? As long as the manufacturers can show that it performs adequately and as well in people of different colorations, then it's up to the manufacturer how they get there rather than mandating you use this scale or that scale.
I agree. That was my point. My point was we're doing a pretty good job right now showing such discrepancies between dark pigmented, self-identified race and ethnicity. Why are we leaving that? until we have studies that actually go head to head comparing, I designate myself as a certain race, here's a numeric value or a scale, and that kind of correlates and the data is still generated. So I'm agreeing with you with that. We should not abandon the self-identification because there's a lot of other things in the skin besides the melanin. And my dermatology colleague, and excuse me for forgetting your name, has kind of pointed that out very succinctly.
I think at the end of the day, as long as the device performs in everyone through a wide range, then I think everyone's going to be satisfied with that.
Yeah, the other thing I wanted to say, again, not to, I just want to make this other point. All the money is around abnormal low PaO2s. We should have an expectation that the device companies look at those levels of to show that in the lower ranges of pulse oximetry, it's reliable, it's precise, and it's accurate. And we should definitely lower the expectation of a 1.5 to 2%. Again, we need to have some type of number, call it a reliability index of how reliable is that number that I see based upon all that information.
Okay, thank you for that. I'm not going to go in order because I want to make sure I give everyone an equal say. So I see Dr. Connor has his hand up. Do you want to go next, please?
Sure. I think just, sharing some ideas about, like Dr. O'Connor mentioned, the idea of, not all differences are the same. If you're a 95 and you're off by 2, it's not clinically relevant. But if you're truly an 87 and you're off by 5, that's where you're certainly now treating or possibly treating patients differently. So I was just going to point out, there's some literature on this method too that Ken Sherlock, myself, and Lee Bragg, when we were all together at Cleveland Clinic, did this in INR point of care devices. When INR devices were first being produced point of care, we looked at this versus like going to the Coumadin clinic and saw, different values result in different clinical decision making depending upon where you are on that scale. So we basically, took a Bland-Altman plot and created regions that led to discrepant clinical decisions, not just discordant numerical values. Published that and then also actually ran some trials comparing different point of care devices using that methodology versus just looking at concordance. So I just wanted to, put on record, maybe this is a shameless plug, but there are ideas that like don't just look at something like ARMS, but look at really when do clinical decisions differ, and you could use that sort of methodology by race, and see when these things are breaking down, or by race or by skin tone, that sort of thing.
Thank you. Let's go back to Dr. O'Connor.
Yeah, I just wanted to make one additional comment, and it's about the coloration issue. 15% is, in fact, the wrong metric. What we need to instead say is that they need to have a statistically significant number of patients, one that allows them to assert a 95% confidence interval that's no different than Caucasian patients. So it doesn't matter, I don't, other people who are wiser than me will decide what we're going to do about coloration. But for myself, I think we need to have statistical criteria, not percentage of population. And what that may mean is that, you end up with 200 Caucasians, 200 African Americans, 200 Asians, 200 Hispanics, or 200 grade skin 1, 200 grade skin 3, 200 grade skin 24.
Agree. Thank you. Dr. Loeb.
I'm going to be a little contrarian about the coloration because in my mind, all of the studies do show gradation between darkly pigmented, intermediate pigmented, and lightly pigmented people. So I don't see, unless you're building a model that's going to count on a per person basis, why you need to put the intermediate people in there. In my mind, there are two problems with the way that the breathe down studies are done. The one is they've predominantly been done in lightly colored people. And I would propose that equal numbers of darkly pigmented and lightly pigmented people be used. And by darkly pigmented, I'm saying the people who are on the highest or the next to the highest in terms of pigmenting. And secondly, that they're done under best best possible conditions of good perfusion. And I think it's vitally important that the breathe down studies be done in poor perfusion conditions. And for instance, that could be done by cooling the hand or possibly having a cuff above the pulse oximeter up to something that will increase venous congestion, but it will also decrease the pulsatile amount. So I think that's important because each pulse oximeter should be able to demonstrate giving an equal distribution. I think that's very important what Dr. O'Connor just said. An equally bad distribution, I guess it is, in different racial groups.
Yeah, I agree, and I think that point. Hopefully, this is getting over to the FDA. These devices need to be studied and validated across a wide spectrum of coloration and a wide spectrum of disease severity. It's interesting if you compare. We've made some comparisons already to typical drug studies where you have a phase 3 study. And even in drug studies that are phase 3, they're not really real world because they've got so many inclusion-exclusionary criteria. It's typically a very highly select population. What I would venture to say is in the equivalent of a phase 3 study with these pulse ox devices, you need the phase 3 to be real world. You can't have a very highly select population that doesn't have any peripheral vascular disease or any kind of circulatory problems, isn't on any kind of vasodilators. And the device, might not perform quite as well, but at least have some transparency around it and some warnings around it that, hey, if this patient's blood pressure is less than 80, whatever the case might be, then your reading might not be accurate. So I think the equivalent phase 3 study actually needs to be real world, not a highly select population. And oh, by the way, after approval, go do your phase 3 study. You go do your real world studies. All right, let's see where we're at. I see Dr. Wilson, you have your hand up.
Yeah, thank you. I wanted the opportunity to agree with the contrarian Hugh Kasari earlier about it may not just be the pigment. Totally agree. It may not even just be the skin. We know that within every race there's a wide gradation of skin pigments, but also in terms of the static absorbers, which can be even more important than skin pigment, according to our research, bone density, thickness of the digits, all of those factors. And that's the reason why it's really important to have the full spectrum across, however you can characterize various races, ethnicities. Just wanted to agree there. Also, one aspect that became very prevalent with COVID was we found in critically ill patients a number of publications that showed very high methemoglobin numbers, higher than was expected, and carboxyhemoglobin. There's endogenous carboxyhemoglobin, which is related to the breakdown of hemoglobin. And we'll need to make sure. I think if we do phase 3 trials, there has to be precise quantification of the hemoglobinopathies that and above some number would need to be evaluated, so you could have a fractional, not just a functional saturation.
I want to just bring up another aspect. Actually, before I do, Dr. Kirsch, we haven't heard from you yet, and I see that you have your hand up.
Yeah, thanks. I agree with all the great comments that have been made. I just would like to emphasize the disparities of weight or BMI across different populations, and I would strongly encourage us to consider having the FDA take that into consideration as they look at different types of patients, not only their skin tone, but also their BMI, which will add significantly to the problems of measurement.
Yes, agree. That would be the broad swath of patients that we're suggesting. So no BMI cutoffs as well as patients are hopefully included in these types of studies. Dr. Collip.
Yeah, I just sitting here, again, a lot of the focus has been on critically ill patients or healthy patients, but another area that you see relatively healthy patients that have significant oxygen desaturations are in the sleep lab. And we have patients every night that desat into the 80s and 70s, and I think that might be another area that might be fruitful to look at, because as far as your phase 3 trials, because there is a lot of variability in the sleep lab and with regards to saturation and BMI, and there's obviously a diverse patient population as well.
Yeah, thank you for that comment, because that does remind me as well of the patient population that we see in our clinic, patients with advanced lung disease. About at least 1/3 of them are on oxygen, and we base it on 6-minute walk tests for the most part. And it's a big decision, do you put patients on a therapy, which is a significant imposition in terms of quality of life, having to lug around oxygen or not? Or, are you withholding supplemental oxygen to patients who really are desaturating and might develop end organ dysfunction or pulmonary hypertension because they haven't been put on oxygen in a timely fashion? So I think when we think about and mention broad populations, it also has to span disease severity from normal healthies, where it's always resided, to patients who are on the fringe of needing and need supplemental oxygen, to the critically ill patients. So I think that is just another aspect of these broad kind of designs to validate these devices. Thank you.
And I would just add in a CPAP machine too. We base a lot of it on, when to give somebody a CPAP machine.
Dr. Branson. Mr. Branson, I'm sorry.
That's okay.
Otherwise, make you honorary doctor.
That's all right. I always tell the guys at work, just give me the doctor's salary. You can call me whatever you want. I don't like the idea of using self-identification of race. And I have talked about this enough that I use the same examples, and at least I'm always looking for who are two people might know who they are. But think of Colin Powell, who self-identifies as Black, and Tyreek Hill, who plays wide receiver for the Miami Dolphins, who's a young man from South Georgia who's very Black. And those two patients, or those two individuals in self-identification, have significantly different skin colors. And I really do, I'm worried that, I'm not saying that we don't collect the self-identification data. I'm just saying that something about the skin color is more, I think, is as important or more important. And I'm with you that one of my other tasks recently has been working on new orders for home oxygen therapy for CMS. And I would hate for us to finish this and find out that oximeters aren't accurate enough to prescribe oxygen therapy for patients, or, as you said, Dr. Nathan, are depriving some patients of oxygen who should be getting it. And we have to revert to having to have a blood gas to determine who needs, who's going to be allowed or is going to be permitted by CMS to get home oxygen therapy. So I think overall, the thing that's for sure is we need to have better designed oximeters that take these factors into effect.
Thank you. Dr. Connor.
Thank you, Jason Connor. Yeah, I was just going to reiterate that I think that, the way these are tested could be much more rigorously done. I agree that the ideal situation, like Dr. O'Connor said, is to test these, in the ICU, to test them in the OR. I think that's really hard to do, and I can imagine even IRBs not granting permission for that, because there's little advantage to the patient in having just this done for research purposes, but there wouldn't be potentially any medical value to them, given that the timing of the blood reading may not help them, especially in the OR. But I think there are plenty of analogous examples. The gentleman from Massimo kind of described and tried to put down some of the research we saw earlier, but I think he validated the point that I have no doubt that Massimo is really, really good at getting very accurate measurements. They're better than anyone else in the world. So, meaning in the lab. So that, when we're actually using these in emergent situations in a hospital or in an OR, all these situations that the readings aren't nearly as good. but I think FDA, can now mandate ways this is tested. I mean, I think, analogous situations. I have a friend who is a pilot and trains other pilots. So in the simulator, he doesn't take them up and simulate windless, cloudless days, right? He simulates really hard situations. And if they don't handle that, he can ground them and they can't fly commercially. Similarly, in the opioid side, in CEDAR, when a new long-lasting opioid comes out, the company basically has to try to break it. Meaning, they have to try to break it down so the long-lasting opioid can't, be used all at once, which is, what used to happen. So people could essentially make their own home heroin or get a much more rapid high than that allows. So there are instances even within FDA where, a company is forced to challenge their own product. And I think that needs to be done here with, tightness, with more light, less light, different color skin, the example of using the cuff to limit oxygen flow. I think all these situations, we understand they won't be perfect, but at least this way that clinicians for, particular patients can realize, oh, for my types of patients, I should choose this device over another device.
Thank you for those comments. Let's go to our consumer representative next, Ms. Edwards.
Okay, I just wanted to comment on the pigmentation, the darkly pigmented that FDA has asked for in the 15%, right? And I guess I want to know how FDA defines darkly pigmented, because in the African American community, the pigmentation is as diverse as the people are. So I think that would be interesting to get a definition of what they define as darkly pigmented.
I think it's a very valid point, and maybe one of the things that comes out of here, because I'll bet going back, it just says exactly that, without being more specific than exactly that. And I think that in terms of new guidance, in addition to increasing the 15%, and I think a couple of us had thrown out it should be 50% or thereabouts. There need to be specific guidance, not just dark skin, but maybe that's where the scales come in, that whichever scale you choose, there's got to be a broad representation across that scale. I think the companies can use the scale to say, yes, we did study X number of individuals across the spectrum of the scale, but I don't think the scale itself can be incorporated into any kind of clinical practice, but it just helps them to validate their device. So I think that's very helpful information or suggestion. Thank you very much. Dr. Yarmouth. Oh, sorry, go ahead.
So why not have a test of just people with color, and to figure out, where the issue lies, right? Because we already know that it's in the people of color, right? You get what I'm asking you?
Yeah, I think what
why
you're
not go
asking
back
is
to
maybe a separate study in people of color. I think you can either have it as a separate study or wrap it into one study where it's equally divided, or kind of nuance it that way. But I see Dr. Edelman has her hand up. Maybe she wants to address this issue from the FDA standpoint. You're muted.
Sorry. Yes, thank you, Ms. Edwards. I just wanted to clarify, you are correct, as is Dr. Nathan, that up to now we did not have a definition of what that meant. And that is why this, we believe, is one of the key questions that we would like the panel to deliberate. And that's why Josh presented that, had the presentation where he discussed all the different ways to subjectively and objectively assess skin pigmentation. And we're very interested in hearing panels' recommendations as far as preclinical studies for for pre-market assessment. In other words, not just after they get on the market, but what, if any, of these scales should be utilized in assessment of how good the pulse ox is before we make a decision if it gets to the market or not. Thank you.
Thank you for that clarification. And I guess that'll be one of the questions we'll be addressing But just to build on that and just to kind of plant the seed as we try and answer this question, I'm not sure if we as a panel can come up and say, well, this scale's better than that scale, and we recommend the FDA use this scale. And maybe the important message is use a scale, whether it's a 6 graded scale or a 30 graded scale, as long as there's representation and good representation across the spectrum, maybe therein lies the answer rather than, you know, being too prescriptive. That's just my viewpoint, and maybe it's because I don't know enough about these individual scales. I've never used them or seen them in action or know what the concordance is and how difficult or easy they are to use. But anyway, I know Dr. Yarmus has had his hand up for a while. His hand must be getting tired. So why don't we let him have a turn?
Yeah, I'll put my hand down. Thanks. I'm actually going to shift a little bit. Hopefully that's okay, which is to talk a little bit about the outpatient setting again, where we know medicine in general is moving very much towards outpatient practice, right? And trying to ideally keep patients out of the hospital, and more of the hospitals are being taken up by ICU beds for the critically ill. So I just want to be mindful of that as we just sort of continue to navigate through the pandemic, where many of us have utilized these outpatient oximeters as indicators for outpatient management and potentially inpatient admissions. Many of those are these devices that are not as regulated, right? They're the ones that you buy at CVS. And we've looked at this through the years. I know our CF group, for example, has looked at different devices to sort of assess patients in the outpatient setting. So I think we'd be a little remiss, and I don't think we're ignoring it, but just to sort of put aside those devices and the importance of them. If you look at watches, and right, everyone that has an Apple Watch now can monitor their oximetry, and how we manage those. So I think part of this discussion should also importantly be focused on the regulatory aspects of how those devices are assessed, because that may actually be an equal or greater problem as this progresses.
Agree, yeah, and I think that becomes part of the questions in terms of differentiating between the consumer and medical grade devices. So that lays the foundation for us to try and answer that one. But you're right, I mean, those are probably used much more perhaps than what we use in the hospital, and people make their own decisions about triaging and what they're going to do based on what they're reading from those devices on.
And
Dr. Hudson.
Sorry,
Dr. Yarmus, why don't you go ahead?
Yeah, thanks. I think it's also I'm not sure it's just the patients, right? I think providers are increasingly utilizing that information and may or may not understand the regulatory aspects of the differences of a clinic, a pharmaceutically purchased device at a drugstore versus one that's in the hospital.
You're absolutely right. I know we do for our lung transplant population. Everyone goes home with a pulse ox, and then if they call short of breath, the first thing is, well, what's your pulse ox showing? So you're absolutely right. Dr. Hudson.
Yes, thank you, Dr. Yarmus. As I was listening to this discussion, I was thinking all the same thoughts. I actually think the iceberg is really the outpatient devices. And I will second, and this is why I raised my hand, is I don't think most physicians know that these are really fully unregulated devices and untested. And all of that is now feeding into apps and into medical decision-making. They are not used recreationally. They are used for medical decisions. And I think we would be indeed remiss to not be addressing that issue with this panel. I'll also say that as clinicians, we use pulse oximetry very differently than patients do. We use it as a device to monitor trends. We use this information in conjunction with a lot of other clinical information. I think the devices that are sent home with patients and that they purchase, I think should not be a number. I think it should be something that takes into consideration the perfusion, averaging over time to give an indication of whether there's concern or not for connecting back with their care team. And I think that would be a great advance for home use of these devices.
Thank you, Dr. Hudson. Dr. O'Connor.
I agree with what everybody said about the outpatient devices. I mean, two things astonished me in COVID, one of which was how many patients who were profoundly hypoxemic were not dyspneic, right? And the second thing is, honestly, I think we should think about offering the FDA the opportunity to create a third class of device, which is a device intended for use at home by patients in collaboration with their care providers for decision making. That is to say, we might not need all of the features for an inpatient device, but we may well need to provide them with guidance because, you know, one of the other lessons we learned in the COVID-19 pandemic is that when the hospitals get filled with people who are mechanically ventilated, we're going to send people home on oxygen by nasal cannula in large numbers using these devices to guide their care. And anything we can do to improve that performance would be a benefit to the public.
Yeah, I think we can certainly suggest that. I'm not sure the feasibility of a third category of device, but for one, the medical grade ones versus the consumer ones need to be very clearly marked. I know I bought a couple of pulse oxes, and to be honest, and maybe I should be embarrassed about this, I didn't know that there were more medical grade ones versus more consumer ones. I just thought there were cheaper ones and very expensive ones, and you get what you pay for generally. You figure that the cheaper ones just don't perform as well. So I think there just needs to be clearly delineated that this is consumer grade versus medical grade would, I think, be a very good first step. Mr. O'Brien. Oh, sorry, before we go there, Dr. Edelman, you wanted to respond to one of the comments or answer a question? Are you muted?
Sorry. I just wanted to clarify that prescription use devices can indeed be used at home with prescription.
So that's the difference. If we want to be sure that our patients are getting a medical grade one, we write a prescription.
But you are allowed to do that, so there's no need to create a third class.
Okay, thank you. Mr. O'Brien.
Yes, thank you. I just wanted to weigh in with the discussion about the use of it. And clearly, from a patient perspective, I think we have to include the home devices that are being done, because the proliferation is just amazing among the patient population. You know, I have my watch, I have my spirometer, etc. I'm a 72-year-old male with anemia, with pacemaker, atrial fibrillation, sleep apnea, and I'm also on pain meds, opioids for a significant amount of time. I have a lot of issues with it. And not to mention that, thank God, I'm not female and not Black, because I am concerned about what do I do with this, et cetera. And, you know, part of the discussion we're going to have is education around that and what that means. And actually, I just realized that Eileen Hudson, I think you're the one that is one of the authors for what I saw in the Annals of Family Medicine, which seemed to suggest that these non-medical devices could be used to rule out hypoxemia in that. So I think there's education on a lot of levels, both at the physician level and at the patient level, in terms of what these actually mean. And, you know, there was a discussion earlier about perhaps putting that on the pharmacist. I think that would be a tragic mistake, because the pharmacists are already overburdened, and I don't think we can rely on our patient education from the pharmacist's perspective on each individual purchase of a, first of all, they're on the shelf. The pharmacist is going to know, and I think to put it would be an unbearable burden. And I hope to God we don't just come up with another label that is 19 pages long that you need a magnifying glass, and that most people just take and throw out anyways. And it also doesn't address what's happening in today's world. Today's world is about audiovisual. It's about a 1-minute video that explains, hey, here's the critical issue. This is how to use it. This is how not to use it. This is what it means. This is if your skin color, et cetera. It should be something that is very easily identified for the education today, what it means, not what we've used 100 years ago, et cetera. So I think that has to be part of the discussion for the FDA looking forward.
Thank you for that. Dr. Casera.
I guess I'm going to be a contrarian again. I think what we're talking about is trying to regulate devices for non-intended uses. In other words, these consumer grade oximeters are not medical grade oximeters. There should be no expectation that they are. And it's going to be very difficult for us to go to the consumer oximeter companies and say, you have to make oximeters that are close to medical grade because the public is using them in unintended ways. I don't think that makes regulatory sense. I think it's an education, and I think you should not obviate or sidestep your provider, whether it's a nurse practitioner, a physician, or a PA. Just because you have access to something that can simulate a medical device doesn't mean you should be using it outside of medical supervision. So again, I'm going to be a contrarian that I think, to speak to what the FDA said, you can order and prescribe a medical grade oximeter for patients at home. So we're talking about making another category that actually doesn't make much clinical sense to me at this point.
Your point's well taken. I think the consumer devices are going to do their own thing, but I think where it becomes important is education and what the limitations are, and perhaps better labeling. I don't know what the labels look like now, but some kind of warning on the box in terms of caveats about the reads, and they might have them now. I'm just not sure how bolded they are, to the point of you don't want to read a 19-page little insert and at the bottom of page 8 there's a little warning that it might not be accurate and call your doctor. So I think just more visibility, and which goes to the education of the consumer about the pulse ox is exceedingly difficult, because how do you educate everyone? I think it's got to be written in bold on the little box that it comes in, and even then people are going to throw away the box and not remember a month later. It's a difficult issue. Dr. Hudson.
Yes, I'd say I know a lot of individuals that have pulse oximeters at home, and I don't know anyone that uses it for recreational purposes. They all use it for medical decision making. So I think it is not in the public's best interest to have these devices that we all do understand are being used for medical decision making and turn a blind eye and say they're just for recreational use. I don't think that is in the public's best interest.
So actually, I have a question for the FDA then. For these wearable devices, how much oversight can the FDA have over these devices in terms of requirements, etc.? Is it the same? You can put in the requirements that are needed, or because they're consumer wearables, falls under a different category?
I'm going to ask Dr. Lee to apply. Go ahead, Dr. James.
Hi, Dr. Edelman. Dr. Nathan, thank you for the question. So, as we discussed at the beginning, actually, we do regulate some of these products under different product codes. And as you can imagine, particularly if there's importation of the devices, there are requirements of registration and listing. So they are falling under, particularly even if they are, sports and aviation, we've defined that. But as you can imagine, there are other devices I think that you guys are discussing that could fall under general wellness, where a company is not claiming medical use, but that it's only for health and wellness, and that would fall under our wellness guidances. So either they would have to oblige by the wellness guidance and follow the parameters in which medical, non-medical devices are regulated for, health and wellness, or they have to register and list and fall under sports and aviation. It is a little bit complicated, and we certainly appreciate the discussion because there are a lot of different label technologies out there. But Dr. Nathan, does that answer your question?
Yeah, it does. It seems like it is quite complicated, and your reach seems to only extend so far. If it's under wellness, then it seems like you might not be able to mandate exact requirements, is my understanding. I'm sure it's very complicated, and there's probably workarounds that these companies can go to get their device on the market, I suspect.
If I can just add, just 1 second, this goes back to the comment made earlier. We can ask for a particular labeling on OTC pulse ox if that's the recommendation of the panel, and then there's a way for us to require that. We just did that for OTC hearing aids. So we would be very interested to hear if that is what the panel is recommending.
It sounds like that's, let me speak what I think for the panel. I think that there should be some kind of verbiage on these devices that aren't quite as accurate, that they are not meant for medical purposes, for medical usage. I don't know what defines recreation, looking at what your oxygen level is. And I'm not sure what the right verbiage would be, but certainly if you have a low saturation level, or if you're short of breath, or you have any other symptoms of concern, you need to follow up with your PCP. You should not, goes back to the point of what I said earlier, not all shortness of breath, all causes of shortness of breath are accompanied by low pulse ox. So I think there's just got to be more warnings around them that these are recreational and shouldn't be used for health purposes.
And one last comment before we leave this. In general, I just would like to emphasize that we're mostly interested to hear what the panel recommends. And then we will take all of your recommendations into account and try to delineate the best path forward.
All right, sounds good. I think we have a bunch of people lined up to weigh in. So let's start with Dr. Hennessy. I think you're muted.
I'm sorry, Sean Hennessy. If I go to a pharmacy and buy a digital thermometer, I assume that it's accurate. If I go to a pharmacy and buy a pulse oximeter that gives me a digital reading, I'm going to assume that it's accurate, despite what you put on the box and despite what you put on the machine itself. I think it sends a mixed message to allow companies to sell something, yet to not have confidence in the results of what it puts out, particularly because we know it's being used for health reasons. So when these things were first sold, it was before the COVID-19 pandemic, and they didn't have the use that they do now. I think that the environment has changed, and as a result, the regulatory environment should change.
I think in the ideal world, we would want these devices held to the same standard as the medical grade ones, and we're going to be recommending, it sounds like, to the FDA that they tighten that one up. So is it feasible to mandate that of these consumer devices in terms of rising to become medical grade because people might use them for medical purposes? I don't know if that's going to be pragmatic or feasible.
Well, so somebody else proposed, and I think it makes sense, is if it doesn't have the degree of precision to warrant giving, a number that goes from 75 to 100, to band it, to say that it's within this range or within this range or within this range without giving a digital number, or to give a range, right?
Okay, Ms. Edwards, why don't you go ahead?
Okay, just again from a consumer perspective and back on my mom. So my mom was under a doctor's care, and I'm almost certain that her doctor was the one suggested that she get the oximeter and the blood pressure machine to measure it daily. And as I said, up until the very day that I called the ambulance, she had not had any signs that her oxygen levels were low, not by the oximeter. So I guess, again, I go back to there needs to, again, as others have said, there needs to be something on there that alerts the consumer to the deficiencies in that oximeter, or either make it the same grade as the medical grade, but then that too has deficiencies. So, I guess in correcting it, they both should be corrected.
Agreed. It's just a question of to what level do you mandate that the wearables be corrected? So we can discuss that further and try and help the FDA find direction in that regard. Dr. Klein.
Sure. So just tacking on a little more to the discussion about the consumer products. And initially, when the suggestion was made about they should be behind the pharmacist and there should be a conversation, I thought that was a really good suggestion, though I do recognize that that's very challenging and it's already onerous. We already have a workforce shortage for pharmacists, and that's additive work. But I am concerned about, we're talking about pretty complicated messaging. If we have consumers that are low literacy, unable to read, how are we going to message all of this so that they can be informed when using these products? So that was one comment. And then this is something that might be a little bit in the weeds, but unrelated to that comment. And this may be more specific to pediatrics, I'm not sure, but for pulse oximeters that have a disposable probe, and so now I'm getting away from the actual device, but talking more about the probe, and I'm not sure if, but in this cost-contained environment post-COVID and health systems trying to use things longer and longer. So it can get complicated by, is the probe wearing out? So it may not be the actual oximeter machine if a new fresh probe was put on the patient, or for patients that are at home and also trying to extend the use of these, particularly the wrapped probes for pediatric patients, if they're trying to extend the use so that they don't need to buy a new, spend money on a new probe. So I just wanted to add those 2 cents.
Thank you. Dr. Alam.
Yes, thank you. I agree with a lot of what's been said already. That's Murad Alam. And I guess I just want to call a spade, if you will, bear with me, which is that I'm not sure what the history is, but I think it's clear based on the panel's views and certainly mine that the OTC devices are misclassified. These are not health and wellness items. This is not a toy. This is not, it just isn't. The only reason people use pulse oxes is because they or their doctors or their loved ones are concerned about their health and want to be alerted as to when their health is deteriorated to a point where they need to seek immediate medical attention. So this is a misclassification problem. I don't know how it happened, but this is not shampoo. And so FDA will need to find some way to regulate these. And if the current scheme is insufficient to dramatically improve the products or regulate them with a level of vigor that would be sufficient to actually make some difference, then FDA might have to reclassify them. There is one caveat to that, though, which I think is very important, that FDA should not complicate the reclassification so much that the cost of a pulse ox goes from $10 to $1,000, because that will create an access issue, right? We'll have great pulse oxes, but 99% of people who could benefit from even a not very good pulse ox will not have access to any pulse ox. So there has to be some nuance as to maybe giving a range, maybe clarifying that, numbers, will be 85 to 90. If your lower number is 85, you should give someone a call, whatever. Maybe make them a little bit better to work slightly better overall, be slightly more reliable, be slightly better in skin of color patients, but not make them completely disappear. But I think the main thing is reclassification is essential, because I just don't see how these are health and wellness devices.
I think you hit on a very important point, and that's to find the balance between being too idealistic and being pragmatic at the same time. Because if we impose the same hurdles in terms of these consumer devices as we are medical grade, then we're going to put it out of reach, arguably, of people who need it the most, and then people don't have access to these devices potentially unless they get a prescription, which may or may not be paid for. So I think the FDA should continue what they're doing in terms of having a different threshold for the consumer devices, but much like we will be recommending tightening the requirements for medical grades, it seems like the same holds true for consumer grade devices, that the requirements for approval need to be a little bit more stringent, which might make them a little bit more expensive, but it's a question of trying to thread the needle and do, find that balance, which is going to be quite difficult. Mr. O'Brien.
Yeah, Joe O'Brien, patient representative. I will echo what was just said 10 times, and then I think it's absolutely essential. And, just in terms of looking at it, I don't think there's any finger pointing here. I think what's changed is the environment has changed. All of a sudden now, a problem that existed for 30 years, the risk benefit ratio changed. We now know that there is a life-threatening risk that's associated with misrepresentation of a number, with the accuracy of a number. That changes the dynamic, that changes the views perspective that we had. So previous panels, previous decisions about wearables and non-wearables, was looking at one thing. Now we know that there's perhaps a much different perspective that we have to look at and protect the patient in that particular environment that's going forward. And a lot of that will be, and I absolutely agree, we cannot overprice the market, and access to healthcare is essential for the patient population. Otherwise, we defeat the purpose for that. But if you look through the internet, you look at the websites for both the IT companies that are using wearables and that are developing wearables, and you look at the major companies that are in the field already that share this space, they do wonderful jobs on videos for marketing and advertising, et cetera. And I think certainly if they use their brain and their money with the right influence, they can come up with very specific, appropriate for the population, educational material that can be given to them quite quickly without giving without all that documentation that we typically do, and have it so that we can have a much better device and a much better patient going forward.
I think that all resonates. I do want to make an important point that I think is the undertone of what everyone's saying, is that even though the mandate of this advisory committee is to address the disparity in terms of pulse ox reading of folks who have different skin tones, the one number that resonated with me was one of the studies that was presented that for white people, there was a 10% inaccuracy rate, and for Black people, there was 15%. Well, we shouldn't strive to get Black folks to 10%. We should strive to get everyone to 1%. So even though our initial mandate was to look at disparities based on pigmentation, I think what you're hearing from the panel is to take it up a notch for everyone, make everyone equal and better in terms of the pulse oximeters that are out there.
I agree.
Dr. Hudson.
I'd just like to add to the discussion that the companies that make the medical grade pulse oximeters are the same companies that make the non-medical grade pulse oximeter, the recreational ones. So I don't think it's a big leap for them to up their game. I think it's probably more of a marketing issue than anything else. I don't think we'd be asking too much of the companies. I think one thing that we would potentially consider is taking the number off of those devices that are used at home to not give individuals that false sense of security that it's exactly the same thing as what we use in a hospital setting, and just give some kind of warning that is taking into consideration these other factors. And then you eliminate the issue with literacy and understanding complicated labeling for the FDA if it's something as simple as red, yellow, and green.
Thank you. Dr. Yarmus.
Thanks. So maybe I'll play a contrarian for a little bit and just sort of refocus a little bit on why this may not be as importantly on us as physicians and the FDA, because this is already out there. So I guess the question maybe focused to the FDA is, if we're here to opine on these inpatient parameters and research design, do we have the power to do that in the outpatient realm, right? So should we be looking at these types of study designs from for outpatient patients. I know there are some studies that have not shown much difference, but that's in a, in a select population. So should we just be looking at this more broadly, both for all oximeters, I guess?
I think that's where the conversation is has gone, is that once we start looking that we're going to render some advice on all oximeters that are out there. Yeah, Dr. Yarmus, you had one more point.
Yeah, just to follow. Yeah, I guess where I was trying to head is, there's certainly discussion that most of the onus is on the industry. But I sort of, in fairness, feel like that's swung a little bit for the outpatient, where maybe it's not, they should be equal, right? Inpatient or outpatient. I think that assessment should be treated the same, and we should hold those standards probably equally.
Yeah, absolutely. I think it's hard to come up with different, not to address the whole thing, and to pass it out will just complicate matters. Mr. O'Brien.
My apology, I forgot to take my hand down.
Oh, okay. Then Dr. Loeb.
I hope I'm not being simplistic, but to me, the problem both with the racial disparities and consumer grade versus non-consumer grade is the signal to noise ratio. The problems with skin color, the problems with anemia, the problems with low perfusion, all of those give a lower signal. And some companies call the signal to noise ratio the perfusion index, and other companies have another name for it. As that number goes down, the accuracy goes down. To me, the consumer grades are supposed to be used in outpatients who are otherwise healthy. And so they should just be regulated to be accurate with good perfusion or good signal, and not give a value at all when there's low signal, as opposed to hospital grade, which would give a value when it's low signal and try to deal with those types of patients. And to me, the oximeters should have a display of signal quality, because, as Dr. Goldman said, the waveform and the pulse might be right, but the signal quality might be low. That's the clue that you need that it's not being accurate. And again, my guess is that the amount of static absorption, that DC component at the two different wavelengths, could be used as a correction factor as the signal gets lower.
The questions and the comments have been very engaging, to the point that I've neglected checking on my chat, but I see someone who would be very appropriate to come in here and weigh in. I want to ask Dr. Pfeffer from the FDA, who's not on the panel, but perhaps he can weigh in. He gave us a talk initially on skin pigmentation, and based on the discussion we've been having, I think he might have some important insight to add.
Well, yeah, there are a lot of issues that have kind of come through over the past, half hour or so. Not sure quite where to start or what you'd like me to address first. Certainly there was a fair bit of discussion on the objective versus subjective approaches for evaluating skin pigmentation and what is really necessary. Certainly one of the problems that we see with approaches like Fitzpatrick is that there seems to be a lot of variation in pigmentation level or melanin content just within the very highest levels, the types 5 and 6. Whereas I think some of the objective approaches might be able to give you a finer gradation based on actual melanin content, and maybe even correlated directly with concentration of melanin in the epidermis, especially if it's well standardized with respect to some of these biopsy approaches. So I think there is a a potential to improve the ability to ensure that we're really doing a comparison against with the people who have the greatest skin pigmentation, and not missing those folks. I wrote, there was also discussion about, whether we need to keep this concept of the racial or ethnic categorization, because we don't really know that skin pigmentation is the main factor. And I would just point out that the vast majority of the literature that we see in optics, shows a lot of the a lot of approaches are impacted in terms of optical measurements are impacted by melanin, and there's a lot of evidence on that. Whereas for other ethnically correlated or racially correlated biological factors, I'm not aware of many studies that really show that there's a correlation between those factors and optical signals. So I don't think we should toss out certainly the idea of evaluating skin pigmentation. Possibly you could evaluate both ethnicity as well as skin pigmentation. But I think it could be quite useful. And of course, different scenarios, one of the points I was trying to make in my presentation is that different scenarios may require different approaches. There may be certain studies where you want fine gradation of skin pigmentation, whereas in others it might be less important. So I think we need to be somewhat flexible about that. And a part of
Thank you, Dr. Pfeffer. Thank you very much. Was there a particular slide that Dr. Nathan, that I believe that's how we asked Dr. Pfeffer to present a particular slide?
Did anyone on the panel want to see a particular slide that would be helpful in our deliberations?
One type of subjective approach that may be more.
Oops.
That's it.
Right. So this one and the following one showed some of the subjective scales. So certainly the Fitzpatrick scale, one of the problems with using it, I kind of consider Fitzpatrick to be two different scales. It's the original scale is this essentially sunburn scale, and it's based on interviewing the subject and asking them questions about their sunburn, suntan habits and what their skin does. And it was originally used to just evaluate light-skinned people. And so some studies have used that approach, which is the original Fitzpatrick approach, whereas more recently it seems like there's been a trend to kind of convert this approach into colors. And these are not well standardized colors, certainly. I think often people may take a certain range of colors and print them out on a piece of paper and compare them to patients. I'm not really sure how it's done, but certainly the colors can't be assumed to be very consistent. If you go to the next slide, yeah. So in some of these, the approaches, the color levels may be a little bit more standardized, but for example, Massey Martin, I don't think there's like an official Massey Martin scale that's printed in consistent color levels that can be distributed. I believe people are just taking a paper that's been published and printing it out and comparing it to people's skin color. So it's certainly not ideal in terms of standardization. And I believe one of the panelists talked about the Van Luschen scale, and it indeed pointed out that it's not monotonic. You're not increasing essentially pigmentation level as the numbers go up. It seems to kind of be combining the effect of both melanin and erythema. And that's actually something that we've, in our evaluation of objective methods, we look at erythema as a confounding factor, and we try to, or a lot of prior studies have looked at the degree to which these melanometers can evaluate changes in melanin or differences in melanin content without being affected by the redness of the skin, the blood content, essentially. So I think there are flaws, and certainly there could be a way to come up with a more optimal subjective approach, and maybe something like Munsell is a little more well standardized, where I think you can actually get a Munsell color chart that's more well standardized. But I don't know. It's a soil color chart, so
when I look at this, sorry, I'm echoing there. It just reinforces what I said in terms of the subjectivity around this. It looks fine on the chart, but you can just look at 4, 5, and 6 and imagine that in practical application and people asking to differentiate between 4, 5, and 6 when you're actually faced with someone, and where do you look, or even 7, 8, 9 and differentiating there. So I think what you alluded to in terms of those objective assessments, I think are going to be much better at the end of the day in terms of trying to standardize things. But I'll go back to what I said earlier. It's should we really care about being prescriptive? As long as the companies can show that their devices work in everyone across a broad range from 1 to 10, if you call it a 1 to 4 scale or 1 to 10 scale, and across a different range of pulse ox readings, then it doesn't matter how they got there, as far as I'm concerned.
Well, but the problem is you have to be able to make sure that it works in the people with the highest pigmentation level. And if you have a large bin for that top range, you might miss the people towards the top at those highest concentration levels, the highest epidermal melanin concentration levels. And that's the group I think we need to make sure are included in these studies.
Dr. Nathan, thank you very much. Dr. Nathan, any other clarification? If not, we'll let you return back to your deliberations.
All right, thank you very much. Thanks for weighing in there to the FDA. Dr. Connor.
Yes, thank you, Jason Connor. Yeah, I wanted to politely but strongly disagree with the idea of recommending for the home use devices or the commercial grade, if that's what we're calling a device, is the idea of a range or like a red, yellow, green situation. Like as a statistician, I think giving people less information is never really that valuable. But also, I think it doesn't fix the problem. I mean, if there are true 88s who say 88 in white, but say 93 in darker skin, a color or a range doesn't fix that problem. It's still wrong in those people. More so, if there's a situation where, we would show yellow when we hit 88, it might still be yellow, but someone else is 78. If they're at the bottom of that range, there may be a very different decision. So I think the idea of giving less information is bad. And I've also said this to FDA. There have been times where I've been involved in diagnostic devices and FDA wants it to read out for the clinician as high or low. And I think that's generally a terrible idea, or almost universally a terrible idea. But the idea that it still doesn't fix the problem of bias here is the key, that's still an error, even if we give ranges.
Thank you, Dr. Connor. Let's go to Dr. O'Connor.
Great. So I'm just going to push back as well, but from a slightly different direction. It's 2022. LEDs have never been less expensive. Electronics have never been less expensive. Manufacturing's never been less expensive. Frankly, we could design something we think of as a garbage device with modern technology, and it would be better than the best pulse oximeter of 15 years ago. And it would be better than any clinician who ever lives, right? And so I think, honestly, this is an instance where the reality is this. The patients who are asking to use these things at home are in fact quite sick. They're going to have poor perfusion. They're going to have really a risk for real hypoxemia. And the price on all of this stuff has plummeted. I mean, I can tell you what a home pulse ox cost 5 years ago or 10 years ago. It was out of reach. And the reality is that in the modern world, we're managing so many sick people at home that the price on these devices will be within reach within a year or two. And I think if we set a standard now, the device manufacturers will be able to hit it, not tomorrow and not easily, But I think in 2 or 3 years, I think they'll be able to hit it, and they'll hit it very well. And I think that our public deserves that.
Thank you. Dr. Alam.
My comment is about something else, but I would like to agree with the previous speaker, Dr. O'Connor. I think that's very wise and probably quite achievable. And to your point, Dr. Nathan, we shouldn't prescribe how people should make things better. We should just create standards and let them work towards them. I wanted to speak to the issue of Fitzpatrick scales again, which is something I know a little bit about. There was some concern that it's very hard to classify people. It's actually not. Most dermatologists, I think, almost always would classify people correctly on that scale, which is only 6 items. Basically, if you're redhead, you're 1. If you're blonde, you're 2. If you're other Caucasian, you're 3. Everybody else is 4, 5, or 6, and it's pretty straightforward how to do it. But I agree with your point, which is that it's probably not necessary to, again, be prescriptive or use that. It's a relatively blunt tool to really assess, to really apply the technology and make sure it's working for everybody. So I wouldn't use it for that purpose, but I agree with the previous speaker that we should probably look at both skin color and ethnicity in terms of showing that whatever mechanism companies, manufacturers choose to use to increase the efficiency and accuracy of their devices, that at the end of the day, they have made a change that is sufficient such that now pulse oximeters work well for people of all ethnicities. And the reason for that, I think, is because now this is out there that they don't. So we have to reassure the public that they do. And so to that extent, Fitzpatrick's skin types should be used just to make sure that when these devices are tested, we can say, well, sure enough, they work for all these Fitzpatrick types. And then we can also say, sure enough, they work for all ethnicities. So again, it wouldn't be a method of using the skin type to create the technology, but once the technology is fully operational, it just has to be shown that it does in fact work for all skin types and ethnicities. Thank you.
Let me ask the panel this, because we'll come up with recommendations for the FDA at the end of the day based on the questions. And I'm not sure if this is one of the embedded questions or not, but let's say the FDA takes some or all of our advice and there's new standards for medical grade, which hopefully there will be consumer grade as well. That takes a while to kind of operationalize and become available. What do we tell the FDA to do in the meantime? I mean, these devices are out there.
If I can interject, I don't think they should take any devices out of commission that are currently being available, because that would get us to the issue where people wouldn't have access to any device in the interim. So I think it's the approach maybe that was mentioned before sounds reasonable, to create some specific standards that are reasonable, achievable given current technology, and then come up with a date, kind of like the car manufacturers do with regard to gas mileage. By 2030, your devices should be meeting these performance criteria.
All right, sounds good. Dr. Connor.
Yeah, I'll answer my first question. This is just general. Maybe someone can answer this another time. But I wondered how close the at-home devices were. Like I just went on Amazon, the cheapest one was $10, and on average they seem to be about 25. I wondered how different the technology was from what you have on in the hospital. I mean, it's so cheap these days, as Dr. O'Connor said to make. I was just curious, like the technology was really fundamentally different. I assume it's different in an Apple Watch because it has many purposes, but in a dedicated at-home device, I was wondering if someone in the know could even speak to how different that was. But regarding your question of kind of like retrospectively versus prospectively, what do we do? I don't think we should ignore the retrospective. I 100% agree with Dr. Alam that, we don't want to take devices out of rural hospitals that don't have the ability to, buy brand new devices really quickly or something like that. But I think, maybe recommending a situation where, makers on the market have 1 year or whatever it is to test under these more, real life situations. And then that's either published or FDA publishes a list of people who ignored that guidance. Seems relevant. I mean, we all talk about health disparities. Usually it's not very clear what the root cause of those disparities are, or it's a whole bunch of things contributing to disparities. Here we have a pretty, we agree that this is a problem and we understand what the problem is and the bias due to skin tone. So to ignore these devices, which I assume get used for years and years without replacement, seems to be ignoring a problem that will continue to contribute to health disparities. So I think that something needs to be done, even retrospectively.
I think just to layer on another level of complexity, the devices have been approved based on studies in healthies, 200 reads, 10 patients, 15%. Now we're going to ask them to take it up a step and really regard, very similar to drug trials where we want a study in sick patients, the patients who this is going to apply to. It takes a while and it's going to cost money for these companies. And even though the technology might be a whole lot cheaper, it's kind of like becomes similar to a drug study in that, you need centers, you need approval, you need IRB, costs a lot of money, and eventually they're going to have to recoup the money. So it will add and layer on to the expense of the pulse oxers who have to get over these additional hurdles in the future.
Yeah, and I think it's appropriate to still do the lab studies, but just do a more real world setting where you're not warming the finger up. I mean, hospitals are cold, but I guarantee you Massimo is warming people's fingers before they do the official test. I'm willing to take that bet. So I think just FDA prescribing a more real world list, putting the cuff on to limit blood flow, these sorts of things seem appropriate and actually wouldn't increase cost much beyond what they're doing today. Because they're, bringing in, maybe a few more people of darker skin, but, it's still in a controlled setting where, you have to pay to bring people in, but, doing a few more iterations under more real life circumstances.
Well, I was thinking if you want real life, you're going to the hospital. You can simulate real life, but it's still not real life. So, and maybe I'm being too idealistic. I don't know. Dr. Yarmus.
Thanks, Lonnie Yarmus. I guess a couple of questions for the inpatient data, right? We've just heard all these outstanding presentations and great studies that have illustrated, I think everybody agrees there's clearly a problem. So in the interim, while this gets studied prospectively, what is the downside or process of putting a black box warning as an example for education? And the same in the outpatient setting, right? We don't have that data, but there's, I would argue in the outpatient setting, the messaging that seems to be more important is to the physicians, right? So we have a whole group of experts here. I think most of us were not familiar enough that, how these things were regulated. So messaging on both ends, and I guess that's probably a direct question to FDA.
Dr. Adelman, do you want to address that? The equivalent, I don't know if it would be a black box or the equivalent of a black box. What would that entail?
So rather than, discussing the mechanism by which we will execute it, I think it is most beneficial for us to hear what exactly is the intent and the messaging is. And then once we hear that, we will figure out the best way to communicate it.
Okay, thank you.
Can I follow up on that?
Sure.
So I think part of the issue is, not going to speak for the group, but I think there's clearly indication for prospective data. That's going to take time. And we know from retrospective data and some prospective data that patients are being harmed. So I think that's the urgency, right? In that time frame where we clearly have to figure this out. Is that an appropriate mechanism to use for patient protection?
So Dr. Yarmus, I believe what you're recommending is some sort of labeling / warnings in the labeling and specific education. So if you wanted to give us a more insight into what kind of education to whom it should be directed, I think that would be very beneficial.
Sure. So outpatient, I think, at least from my perspective of these conversations, clearly physician education, which presumably would be an easier access point, right? So if I get a message from FDA that says, look, these things are not as regulated and they should be used under caution and under what they're intended for, I'm going to take note of that. On the inpatient setting, I think we don't have that time, right? Because we already have data that suggests that there's patient harm. So at least in thinking of timelines and regulatory aspects, I would think a much stricter and more straightforward message to clinicians and patients that say these devices don't have the accuracy that we thought they did under these circumstances, and they should be used under extreme caution. In my world, I'll just give an example, as I'm an interventional pulmonologist by trade, and airway stenting remains an important patient care aspect. When stents were first introduced, they were primarily uncovered stents in the airway that caused significant problems. That was recognized. That led to an FDA black box warning, which almost overnight, led to pretty significant change in practice. So I use the same analogy here.
Thank you very much. So the more specific the panel could be about the key messages that you would like us to communicate, as the better we'll be in position to hopefully make it happen.
Okay, maybe we can formulate something during the question period and come up with some guidance on the kind of statement. I do wonder as well, and I should know this, but I don't, between all the great societies that presented to us today, I don't know if anyone has a position statement on the use of pulse oximetry, but it might be, in addition, as a bookend to the FDA coming out with something, maybe there should be a position statement from all the societies together, and that might carry a little bit of weight as well in the medical community. Okay, Dr. Yarmus, you had your turn, Dr. Wilson.
Yes, just in response to the question you asked a moment ago of what could the FDA do in terms of the consumer products. And so in addition to developing the new standards, as has been mentioned by a number of our colleagues, the FDA could promulgate or send out a notice warning to physicians and to patients that if the patients are being monitored at home for a medical condition, that it should be with a medical grade pulse oximeter. And that would at least elevate further the concern to those that just think they're getting something off the shelf that should work, like a thermometer.
Look, I agree with that, and that can be a recommendation. The thing that I don't know is medical grade. Are you always going to get one if you write a prescription? Because people might be under the impression that, this is a big device, I play it a lot, but it might not be medical grade. So I don't know if there's, aside from getting it via prescription, if there's a clear designation on the box or otherwise that this is medical grade.
So perhaps Dr. Edelman might have some advice on that. There might be some new requirements for labeling.
Yeah, Dr. Edelman, if you want to.
So prescription use devices. Once the physician writes a prescription for prescription use device, only the sorry, my mic doesn't seem to be working well. Okay, Dr. Lee, why don't you answer while I fix my mic?
Dr. Lee, I think you're muted.
Yeah, thank you, Dr. Nathan. Certainly I think the requirements on accuracy, that's something that we would look at in the final labeling for a cleared medical grade device. I think we had discussed this at the top, is that, we do publish, we have the manufacturer publish even the Bland-Altman information. So as a clinician, you could see information, and some companies do disclose very granularly information about performance across different deciles. Dr. Kasari, did that answer your question?
I didn't ask that question. I had a different comment. That was another speaker.
Okay.
Okay, Mr. O'Brien.
Thank you, Joe O'Brien, patient representative. My only issue, I'm listening to Dr. Wilson. We have a problem where we have a diversity problem due to a medical inaccuracy in a device that is a real contentious issue because it's life-threatening and it's disparity that shouldn't exist, that we're trying to eliminate in all aspects of our life that is there. And I'm afraid that having a simple solution that just says, listen, if you're using this for medical reasons, you got to go back and get a prescription or a non-prescription and pay now X number of dollars more for it. And we're going to create access problems to people who need it and create a political diversity that is really going to be problematic. I think there has to be a lot of thought given to what seems to be a simple action before that's taken place.
Thank you. I'm keeping an eye on how long we've gone, and we've gone about an hour 45 of discussion, and we can spend a long time discussing this. It was allotted for 2 hours, so we'll probably go for 15 minutes more. But I do want to make sure that everyone on the panel feels like their voice has been heard, and I think everyone has participated, but I want to just put it out there for those folks who maybe haven't said as much. If there's, just don't want to leave anyone out of this discussion. So, Dr. Kirsch, I see you have your hand up.
Yeah, the one thing that I don't think has been touched on is for those of us who have non-medical grade pulse oximeters, which I have one, it can result in bad information that would then result in additional testing that is costly and may actually cause harm. So the case study is I happen to enjoy skiing at high altitude, and I took my portable non-medical grade pulse oximeter with me at high altitude skiing and found that despite without any shortness of breath or any problems skiing pretty aggressively, my pulse oximetry reading was in the mid-70s while I was skiing. And that resulted in, I just continued to ski, but went back to my internist afterward, and that resulted in a variety of tests, all of which were negative. So a bad result resulted in tests that were unnecessary, very costly, and theoretically dangerous, might have caused me a complication from the additional test itself. Just another perspective on why I think tightening up on the quality of the device would be important.
I think a very good and valid point. It's that cascade effect. You find that something is, you have an erroneous read, and before you know it, you have the mega workup. I was going through our list of participants, and I want to give Dr. Lynch an opportunity to weigh in.
Hi, thanks. Yes, I agree with all the comments so far. I think one thing that, thinking about how these can be improved, I believe the dermatologist, I'm sorry, I forget your name, as he commented, it'll need to be an algorithm correction and not an instrumentation correction. So just understanding how that would have to play out. And I'm trying to think of another comment. I think someone also commented on structural heart disease in the pulse oximeter, problem with the pulse oximeter and congenital heart disease, potentially missing a diagnosis, which I think is a very theoretical problem and probably not an actual problem, because you're comparing differences in a pre-ductal and post-ductal stat in someone with a certain skin pigmentation. And so you're comparing two values on that same patient. So it's probably more of a theoretical issue than an actual issue. But there are clearly problems. I think the bigger problem is when we set these guidelines, below a stat of 92 or below a stat of 87, you need to get this treatment. That's when the absolute number needs to be more verified in the different skin pigmentations.
Thank you. And Dr. Katz, anything to weigh in from the pediatric neonate side?
Yes, in fact, there's a couple of things that struck me. The first is that we have a lot of children or preemies who are hypoxemic or not hypoxemic, but when they're ready to leave their hospital, they're often at the nadir, the physiologic nadir of their hemoglobin levels. So not only are they potentially hypoxemic, they're also anemic. And that's a population of kids who are never going to be able to be studied. because they don't have arterial lines. So we're never going to be able to get data on that. And then we have a large population of older kids who are asthmatics and have various conditions also who are not going to be amenable to this kind of study. So the other thing I wanted to point out is that we've experimented in our sleep lab. And looking at data which is taken 5-10 minutes apart is very foreign to us in pediatrics because they're moving around so much that the data has just got to be very, very inaccurate. So it's going to be really important to get this FDA-sponsored data out of California to see if you can get some clarity on this issue. But to me, I think the point is not that, almost certainly these over-the-counter products are going to work less well. And in fact, some of them have been taken off the market because parents were using them in pediatrics as apnea monitors. And they cause more anxiety, and they've never been shown to have any beneficial effect on outpatient and infants. So I think that, but the bigger problem from my standpoint is the fact that even the medical grade machines are demonstrating errors in 15 to 20% of patients, and even in Caucasian patients, they're causing significant harm potentially. So I do think that a warning is indicated, because if people are relying on these devices and are comforted by the fact that these things are accurate, we become so accustomed to using them that we've forgotten that there are strengths and weaknesses of this.
Thank you very much. I'm not seeing any other hands, and I think there will be opportunity to further.
I'm sorry, I've had my hand up for literally 20 minutes.
I apologize. It's hidden in the corner there.
Yeah. Sorry, I don't mean to interject. I just wanted to kind of make a comment and a reset. And I think the comment and reset is just looking at the total data we're looking at. We're looking at, we're trying to tell companies FDA recommendations on what to do moving forward and what to do retrospectively. I think, and I hope people can agree with this, that what we're really looking for is desaturation studies under low perfusion states. That can be simulated in healthy people. And that should be the expectation and the bar that these companies need to meet in all skin tones. And there should be an even distribution of all these normal volunteers under desaturating states and hypoperfusion states, with equal representations in every single one of those groups. And in particular, desaturation states where it's going to make a difference in terms of therapy. And that is, and I think we could agree, we could pick 94%, 92%, but I think that's really what the FDA is looking for. What do we want companies to do prospectively that we're going to approve their device? And what are the expectations going to be retrospectively for the devices that are out there? And I think looking at all the data that was presented, it's not just about skin tone. It's about skin tone and hypoperfusion. I think that's been hammered home a bunch of times with the speakers and some of my colleagues on the line. I just wanted to kind of reset. I think that's what we kind of need to look at moving forward.
No, I agree. And I think right from the get-go, we had outlined that broader group of patients for sure, and under different conditions. Now, whether or not you can accomplish that in normal healthy volunteers versus in a hospital setting, I'm not sure we need to be too prescriptive. I worry about making patients normals hypoxic and hypoperfusing them, although I guess you could do it locally, but I'm not sure how much that replicates in hospital conditions, but it is a start. And as I say, I don't think we can be too prescriptive in terms of these are the exact studies you need to do. They could, and maybe I don't know enough about what can be done in outside clinical research facilities where normals come in all the time for these phase 1 studies. Maybe this can be accomplished, or it could be accomplished in a hospital setting, whatever works best, as long as there's good data to support beyond the 92% where and above where most of the data sits, and to represent all racial tones, I think is the important messaging that will come out of this, and a tighter range in terms of the arms. So I think we started with that at the beginning of this, I think, very productive discussion, and we can bookend with the same message. And there was a lot of great discussion in between. So if everyone's okay with it, I don't see any other hands up. Oh, but we've got two hands. Okay. Mr. Branson, you did have your hand up before. Go ahead.
So I agree with what was just said. I would, in all deference to the physicians, in the hospital, the nurse and the respiratory therapist are the two people who put the oximeter on and look to the oximeter, and the messaging about this inaccuracy needs to go directly to them and to know what they should do about it. I think that's going to be an important part of whatever FDA does. I agree with making the arms be better. And I did want to comment with, for those of, one of the speakers talking about skiing, if you're healthy enough to ski at fairly high elevations in the mountains, I've spent a lot of time in the back of an aircraft or an altitude chamber at 18 or 20,000 feet with an O2 sat of 82% and have no shortness of breath whatsoever. So it's not just a matter of hypoxia, it's also a matter of cardiac output and the ability to hyperventilate. So it may have been possible that Dr. Kirsch's SpO2 was 77%, but he's healthy enough that it didn't affect him at all. And the device may have been actually accurate.
Yeah, and there is the entity of high altitude pulmonary edema, and so it could well have been that as well. All right, last, Dr. Wilson, our industry representative, in terms of what we are saying and what the FDA will say to industry, maybe you can speak to that.
Yeah, thank you. The first point, of course, is that not all pulse oximeters are created equal. Some are better than others, but we can hold all manufacturers to a higher standard. And so that just, that should be the goal, is whatever the FDA believes is the new percentage or number, or if, as Dr. O'Connor said, let's just make sure that the p-value is powered properly so that we can ensure that we have the right mix. So that has to happen. FDA has to make a decision on how to tighten the ARMS, because as somebody said, even some of the commercial, the over-the-counter pulse oximeters are better than what we had a couple decades ago, because of the miniaturization and so forth. But the point that I wanted to make, though, but thank you, Stephen, for inviting that comment. But the point I did want to make was a little bit related, and it has to do with the FDA does allow for extremities to be warmed, and I think that the FDA should tighten that requirement. That in general they should not allow it. No warming of extremities. If it's there should be just basic conditions about room temperature and so forth. But then the endpoint should be what's the perfusion index, so that, or some measure of perfusion, so that if the goal is to have higher than normal perfusion, then the FDA could allow some warming in order to get a perfusion number. But in general, it shouldn't be allowed for, unless you're trying to augment that upwards. And if you're trying to decrease perfusion, of course, then there's other factors that one would need to do in, say, a laboratory setting to decrease that. It might be colder ambient temperatures, things like that, in order to bring the perfusion index down. But those would be better endpoints, and I think just a little bit more scrutiny about when, if we want to be able to compare apples to apples, then the experimental conditions need to be clear. And if we do use perfusion index, let's say as a measure, then that can cross over into the clinical domain in terms of the measure of how well the extremity is perfused.
Thank you for those comments. All right, we'll end off with Ms. Edwards, and then we will adjourn this session. So Ms. Edwards, why don't you go ahead?
I just have a question. So are we moving away from the ethnic implications to more of a pigmentation And I guess the reason I'm asking you that is because I'm thinking about COVID-19 and how we got here to this point. So I guess you get what I'm saying. That's my question, and that's my concern.
Well, I think
pigmentation versus ethnicity, and I'm wondering what the implications are there.
I think that the two are closely linked for the most part. I think that ultimately what the goal is to make sure that anyone who has a pulse ox put on them has an accurate representation, be it couched as different ethnicities or skin coloration or pigmentation. So I'm not sure we need to, kind of divorce the two. I think if we get at the one, we get at the other. But ultimately, if you look at the objective measure, which is measuring the amount of melanin in the skin, that's a skin pigmentation thing, which is linked in many ways to ethnicity. Hopefully I addressed that okay.
Yeah, that was my concern, just because the ethnicity, the African American community, the disparities in care there is how we got here. I just wanted to make sure it wasn't lost.
Absolutely not at all. But I think, to the point that's come across is we want to raise a rising tide carries all boats. We don't want to get the African American community to where the white community is now. We want to get everyone better than that and equally.
Oh yeah, I get you. Okay. Yeah.
All right. Well, with that, let's adjourn the panel discussion. It's been very interesting, a lot of great comments. And I have 3 minutes to 5. I tell you what, why don't we make it 5 after 5 to get back together, and we'll address the FDA questions at that time. Thank you. How's everyone doing? Welcome back, everyone. We're on the final leg of a busy day, but a very interesting day. At this time, we're going to focus our attention on the FDA questions. Panel members have received electronic copies of the FDA questions, and they are posted on the FDA website. I would ask that each panel member identify him or herself each time he or she speaks to facilitate transcription once again. All right, please present the first question. All right, I'll read the first question. Please discuss the clinical evidence from the scientific literature about the accuracy of pulse oximetry among patients with darker skin pigmentation. In your deliberations, consider the strengths and limitations of the studies, including study design, outcome definitions, and potential confounding factors that can impact interpretation of the evidence. Specifically, please address, and we'll break this out, but specifically, please address, this will be the first part. Does the currently available clinical evidence demonstrate disparate performance in patients with darker skin pigmentation? Maybe we can break it down even further, and that'll be the first question for us. And I'll speak for the group because I think you heard, you didn't hear any dissent, but I think the current available clinical evidence does demonstrate disparate performance in patients with darker skin pigmentation. I think we saw a lot of studies and everything pointed to the same. I know that some of them were retrospective, and some of them, the ABG wasn't done exactly at the same time, but how much can it change in 10 minutes? In some cases it might, but for the most part, it's a relatively stable thing. So in my mind, and I don't want to speak for the group, if anyone else from the group has a dissenting opinion about that, I think the evidence we saw was quite clear. Anyone want to add to that or reinforce that? Dr. Luke. Loeb, sorry.
Robert Loeb. I would only add that the early literature, I did have questions about confounding variables and covariates and things like that. But I was convinced in the presentation as the presenters went, and they generally went in order of when studies were done. I am now convinced. I wasn't initially, but the latter studies that were doing more sophisticated analyses, yes, I agree that there is a problem. It has clinical implications.
Right, and I think they brought us along very nicely in terms of the presentation, but there's biologic plausibility why that should be the case. It's not just it happened and it was a chance finding. I mean, there's good reason for this. Dr. Hennessy, do you want to weigh in on that?
Yeah, I agree with everything that you said. In addition to being a higher risk for hypoxemia, Black patients, the performance of the test is measured by the area under the curve is worse in Black patients than white patients.
Mr. Branson.
I agree. There's definitely a single signal there. All I would say is that I think it is exacerbated in COVID-19 because so many of those patients were not only hypoxemic, but they also had viral sepsis and poor perfusion.
Right. I think certainly COVID brought this issue out to much more to the open. Dr. Connor.
Yeah, just to strongly agree, but also to reiterate the point I heard, during some of the open session, I think some of the makers of these devices point out some of the imperfections in the real world clinical studies. But I think the fact is, while that adds noise, we repeatedly saw that the performance was worse in darker skin patients. So, I just wanted to point out that we shouldn't let some of the imperfections in those studies, which are really just real world experience, negate for what is a clear signal with skin pigmentation leading to poorer performance.
All right, thank you. So it seems like, actually, Dr. Collip, let me address the second part of the question, because then maybe I should have done that initially, because the two kind of build into each other, and then I'll come back to you. If so, do you believe such disparate performance may lead to increased risk? And I think my bias is that the evidence was there that it does lead to increased risk. Dr. Collip, why don't you go ahead and weigh in?
Yeah, I guess I have more of a question than a comment. It looks like most of the data that we heard today, which I agree is overwhelmingly that the darker skin pigmentation makes a difference, was on prescription use oximeters. And I don't know how much data we got on the over-the-counter pulse oximeters. And if I missed it, I'm sorry, but it seemed like all of the real world data was on the prescription use. So I don't know how best to comment on the over-the-counter part.
Yeah, I think you're right, because all of this was in hospital data, so it had to have been prescription use. I think the assumption is if that's how prescription use oximetry performed, then consumer devices, which aren't held to the same standard, have to perform just as bad, if not worse.
No, we would assume so, but I don't know that we know that for sure. But I agree.
Okay. Does anyone dissent from the fact that these devices and their performance in folks of darker skin lead to increased risk and worse outcomes? Okay, so I think we do have consensus there. Please include prescription use. Actually, this is getting to what Dr. Collip mentioned, and over-the-counter pulse oximeters when used for medical purposes in your deliberations. We had a very robust discussion about the over-the-counter pulse oximeters. And so actually, Dr. Collip, I think you helped me answer the third part of that question. I think the assumption is that over-the-counter devices perform, in all likelihood, even worse, without seeing studies specifically attesting to that, because they're held to a very different standard. Does anyone else want to weigh in on over-the-counter devices? Okay, let's move on to part B. Do you believe the reported disparate performance or increased risk may be explained by factors other than darker skin pigmentation, such as perfusion index and motion artifacts? Dr. Casera, do you want to try and tackle that first? You're muted if you're talking. Dr. Casera, I don't know if you wanted to weigh in or if your hand.
Can you hear me now?
Difficulty.
Can you hear me
now? We can hear you.
Yeah, my computer literally booted for an update, so I'm on a new computer.
Oh gosh, sorry. Okay.
So what I wanted to say is that at least the way I read the data and the way it's played out, it looks like it's not just the a difference in skin pigmentation, but combined with a low perfusion index. In other words, patients who are critically ill or sick in the hospital, by definition, there's perfusion issues, and this is amplified by the fact that there's also racial skin pigmentation issues that exacerbate it. So I think that those two hand in hand are the source of the problem.
Yeah, I do remember that. I think that one slide with the Bland-Altman plot showing the combination of pigmentation and perfusion really caused a very wide dispersion there. So I think the one feeds into the other. I would agree with that. Dr. Yarmus.
I agree. And I would just add that I think the signal of that 13% versus 10%, right? That 10% difference in white patients still shows there's an issue beyond race. And that should be, based on what we heard, include these types of parameters.
All right. Can you, some more comments. Dr. Connor.
Yeah, strongly agree with the perfusion index comment. I think one other thing that would be interesting to make sure it's studied, though, is finger width. We saw in some of the papers that we were given to review that there was less of an effect with race or skin pigmentation in infants or kids, which has a smaller path length for the laser to travel. And because, obesity, at least in the US, is higher in some, non-whites, I think that could confound that. I certainly believe that skin pigmentation is the root of the problem, but we also wouldn't want to ignore, for example, if this didn't work as well in, light-skinned people or white people who were heavier as well. So I think that's something else that was unclear to me or not yet quantified that's worth considering.
I think it's worthwhile reinforcing, and it did come up in terms of just studying these devices in a wider population, including different body habituses, especially because of the obesity issue in the US in particular. Can you go down a little bit on the question? I just want to make sure we're not missing anything. There isn't a part C, that's all there is. Okay, I can see. Dr. Collop, once again, why don't you weigh in?
Yeah, the only other thing I think we heard also about maybe the probe itself may play a role in some disparate performance, which gets to the prior person's comments, about maybe bigger fingers, littler fingers, what have you. But I think one of the presenters suggested that, if it's too tight or too loose or there's light coming in, those also seem to may cause some error.
All right, let me try and summarize this. So Dr. Adelman from the FDA, with regard to question 1, the panel, not the channel, I've been going at this too long. The panel believes that clearly there is a disparate performance in patients with darker skin pigmentation, and this does increase these patients' risk for whatever disease they might be having. Even though we didn't see data on over-the-counter usage, we know that these are used by patients on the outside to gauge whatever illness they might have. So we believe that there is this effect that we're seeing from the numerous studies on inpatients probably has ramifications for the outpatient over-the-counter devices as well. And so that's the summary with regards to A. We do believe that other factors, and clearly perfusion plays a role, not just pigmentation, but perfusion. And I think something that the panel picked up on, not only perfusion, pigmentation, but other perhaps demographic factors in terms of the width of the finger, the breadth of the finger, obesity being an issue. And so there are factors just beyond pigmentation, but certainly pigmentation is the main reason why there is this disparity. But as the FDA looks at this, I guess we would encourage the FDA to consider these other issues as well that can be addressed perhaps in some fashion at the same time.
Thank you very much, Dr. Nathan.
All right. Dr. Edelman, is this adequate, or is there anything further you wish us to address with regards to question 1?
No, that is definitely adequate. Thank you very much.
Dr. Nathan, may I add?
Is that Dr. Wilson? Please, please go ahead.
Just from, so 2 points. First, that we're all in agreement that the preponderance of data shows that there is a signal. However, and that's despite having some methodological concerns. However, all of the clinical data had all of the various manufacturers combined. They were not separated out. So it would be appropriate to say that there could be differences between different manufacturers that you don't really know. On average, there's a signal.
I don't need, no, we need to drill down on that.
I just want to finish the point, which is that since we think that perfusion and motion is important, that some manufacturers have worked on those problems and they're less problematic. So it is important to not call out not to say that any company has a solution just yet, but that there may be more work to be done by some manufacturers than others. That's essentially the point.
Right, and as I said, I don't know that we need to drill down to that. I think everyone will be held to the same standard eventually. Some companies might be closer to that standard or at that standard already. But your point is taken that different devices were probably used in these clinical studies. Doesn't take away from the signal that was ubiquitous across these studies. Okay, let's move on to question 2 then. Question 2, there are several tools to assess skin pigmentation, including but not limited to colorimetry, spectrophotometer, better just to say it, Melosome volume fraction, and skin color scales like the Fitzpatrick and Van Lucian color scales. Please provide recommendations for studies evaluating pulse oximeters for the following. Standardization of skin pigmentation assessment. Dr. Kasseri, why don't you go ahead?
Yeah, I'm going to be the contrarian again and say that I think looking at the totality of data and what's been reported, it's mostly self-reporting. We should have an expectation of self-reporting, and if you'd like, pick a color scale to go along with that to report both. Maybe have some kind of standardization, because we just do not have enough studies that point to skin color scales to pointing out the problem that we found. We found the problem through self-identification, not through skin tone studies.
Dr. Lowe, why don't you go ahead?
I agree, and I think that the patient advocate, Ms. Edwards, brought up a very good point that this began, or really is seen as a racial/ethnic issue, and that it would be wrong to just go with some standardized assessment of color. So I agree that two standards should be used, both some sort of a standardized assessment of color, and I would add color at the site of the measurement, and also self-described ethnicity, and that the analysis of results would be on both subgroups.
Okay. I'd venture to say that I'm not sure how prescriptive the FDA should be, as long as the assessment spans a spectrum of coloration and skin pigmentation. I'm not sure, primarily because I'm not sure what the best scale is, but whatever scale is used, it should go from 0 to 100, lightest to darkest. And I'm not sure if the FDA wants to be prescriptive to the manufacturers of this is the scale to use. I think the objective measurements that were mentioned have a lot of attraction in them because they are objective. And even though the color scales perform well, I do think that there's probably some advantage to using these more objective scoring systems. Perhaps, in terms of standardization of skin pigmentation assessment, perhaps it's more important to say that the full range of skin pigmentation be evaluated. Would that be fair and encompass everyone's opinions, or do you feel that we as a committee need to come down on one thing versus another? Dr. Hennessy, why don't you go at it?
Thanks. So I'd say two things. I'd say that race is a social construct, and that in my mind, it doesn't make sense to evaluate the comparative performance of a medical device based on a social construct. I think it makes more sense to look at skin pigmentation. I think that spectrophotometers are an objective way to measure skin pigmentation. It could be applied to the site where the pulse ox is going to be applied, and it's not a particularly expensive piece of equipment. So, I don't think that's a conclusion, but that's the direction that I would lean in.
Dr. Yarmus.
I am going to just mirror what Dr. Hennessy just said. I think it was perfect. I think we have to hedge a little bit just because of that, the panel expertise, right? We have one dermatologist. If I go to Home Depot and I want to match a color of paint, I can have them do that, right? So I think from a technological standpoint, there are ways to do this that I think are beyond my expertise. But I do think there should be both a subjective and detailed assessment on both ends.
Ms. Edwards.
Hi, yeah. So I agree with Dr. Lowe. And while I agree that race is a social construct, it's one that's that are used to discriminate, and it's caused a lot of disparity in healthcare for African Americans. So that cannot be, I think, made, to be a small issue when it's really a great one.
I think it clearly is. And I think what I would say is if we go with the pigmentation, which is more objective, I think that encompasses the race aspect and it takes care of African Americans. But to the point that was made earlier, folks who identify as African American in terms of skin pigmentation can be very light or can be very dark. But if we objectify it with one of these different measures, the spectrophotometer being one example that I'm unfamiliar with, it's not my area of expertise. I think it takes race a little bit out of the equation, but addresses race at the same time, if that makes sense.
I guess my fear is that historically, the disparity in healthcare is like it never stops. And so when you lump African Americans in with everybody else, we always end up on the short end. And so I guess I have a problem there. But I mean, that's just my perspective. Because this started 30 years ago, right? And here we are today, we're just addressing it. So yeah. So, that's my
I think these studies can be done to address both. I think certainly the part of any study is to identify ethnicity or self-identified race, but at the same time to have the objective measure at the same time. And I think what you'll see is if you address the spectrum of pigmentation, you're going to have great African representation as well as other ethnicities within the group to get these devices validated.
And I guess FDA would just have to make sure they give some pretty strict guidelines as far as the testing, rather than the two darker.
I think that became clear. I think that there needs to be equal representation, because certainly that's where the big problem lies are with folks who have greater pigmentation.
And to hold manufacturers accountable, because I think that's where too it gets a little murky too, because I think in some of the testing it came out that some of the manufacturers didn't even follow that guideline. So yeah.
Right. Yeah. Dr. Collar.
Yeah, I just want to agree. I think right now, most of the literature would suggest that it is race related based on how the studies have been done. So I think we need more studies to see if it is truly race related or if it's pigment. Physiologically, we think it's probably pigment, but we don't know. And I think we need to confirm, I think both should be identified, both race and some measurement of pigment to confirm that that's the case.
I agree. I think a bigger danger is if you say 50% African American and you get 50% very light-skinned African American, then it doesn't address the issue. So I agree with both being addressed. Dr. Connor.
Yeah, you just made the point I was going to make, that is playing the skeptic about device manufacturers. And I think most have patient best interest in mind, but I would worry that, if you need X African Americans, you may recruit, because if it's not in the clinic and if it is in the lab, they can recruit the people who, they know their system may be less biased in. So therefore they're checking the box, but they're not really solving the problem. So I think making sure we have enough people, on, for instance, the Massey Martin scale that are dark skinned. Maybe we also, given this is the United States FDA, also want to make sure that those, darker skin types are represented by enough African Americans, not just, dark Indians or dark, people of other races. That way we make sure that African Americans are included in that to address Miss Edwards' very important concerns. But we're also, like you said, making sure we have enough, dark skinned people so that manufacturers can't game, the evidence they need to bring.
Okay, let's end off with Mr. Branson and I'll try and summarize.
Again, I appreciate Ms. Edwards' viewpoint, and she's the person of color on the group. But I also know currently we are finally doing away with race-based equations for predicting pulmonary function. And we are doing away with race-based equations for renal disease. And in the past, race-based differences were exploited by white supremacists to suggest there's actually a difference between us other than the color of our skin. So I think we have to be careful there. Again, and I still, objective measures are for science, but I do appreciate exactly what she says, that race definitely has to be included.
Okay, thank you. So, Dr. Eidelman, I'll try and summarize this. With regards to question 2, the panel generally believes that yes, there should be standardization of skin pigmentation assessment, which should include an objective measure ideally, but can also include one of these more subjective Fitzpatrick scale. I think it should be easy enough to include both of them. Just it's something else that the companies can do just to see what the correlation is. I think that'll move the field forward a little bit, but definitely identified race should be included as part of the demographics captured, and there should be equal representation across the spectrum of skin pigmentation. In terms of categorization and reporting of skin pigmentation, I think I've kind of alluded to that already. Categorization, ideally based on objective measures, but also can include these visual analog scales to complement that. And the reporting of skin pigmentation can be categorized by all the mechanisms, including the subjective color scales, as well as easily enough to do subgroup analyses based on race if there is the desire to do this. But to the point that Dr. Branson made, the data will exist, but you made an important point in terms of trying to bring the races together in the context of all these things and just differentiate people based on pigmentation. So Dr. Eidelman, I feel like I danced around that a little bit, but did the group and did I summarize this adequately?
Yes, thank you.
Thank you.
Can I add, you did not in your summary say where the colorimetry would be performed. And I do think it's important that it be done at the site of the measurement.
Yes, I agree. I think that is very important. What I didn't also add is, and I'm not sure how far the FDA should go with this, because to the point I'd made earlier, you put it on the finger, don't get a good signal, put it on the earlobe, put it on the forehead. I would encourage the inclusion of multiple sites to fully evaluate the performance characteristics of the devices. All right, let's move on to question number 3. FDA currently recommends assessment of the effectiveness of pulse oximeters using ARMS, which is the root mean square of pooled data pairs, and adherence to the currently recognized ISO 80601-2-61 2017 standard. For this variable, currently pulse oximeters are expected to have accuracy within 1 standard deviation, 66% of the time, and within 2 standard deviations, 95% of the time. Please address the following. Please discuss how accurate pulse oximeters should be for clinical use. In your discussion, please address whether the accuracy varies based on the clinical setting or the levels of oxygen saturation. Well, I think I'll start off the group. I think what we heard is clearly the arms need to be tightened. How much it should be tightened, I don't know. I don't want to throw out a number. I don't know if anyone, statisticians have a better idea of how tight we should recommend to the FDA for the arms to be tightened. We can get to that, but I'll let Dr. O'Connor weigh in at this point.
First, I'm sorry I lost my internet. That's why I was absent for the first question. So
I think that the clinical data suggests strongly that in arms of between 1.5% and 2%, in the range of say 70% to 92 or 94%, would be significantly better clinically than in arms of 3%, which is the current FDA standard. I think the second thing the panel is likely to agree upon is that we need to have data about positive predictive value for each saturation that a pulse ox might report, and a range of 95% confidence interval. We believe the criteria for developing a pulse oximeter should require a sufficiently large population of patients with different pigments. However, the FDA decides, or the manufacturers decide to determine that the 95% confidence intervals for the Caucasian and the African American, or the very darkest skinned of people, will be very similar, both in terms of their accuracy and their precision.
Dr. Kasseri.
Yeah, I'm in agreement with Dr. O'Connor. So I just want to make I think we really need to tighten up in the lower O2 range, O2 sat ranges, especially below 92%. I think the 2% is definitely reasonable. And I also want to kind of highlight, we have to, pulse oximeters in the past were used as monitors. Now we're using them for therapeutic interventions. That's a very different mindset. I am going to start or stop a therapy on you based upon your peripheral O2 saturation. That should be held to a very different standard. And most therapeutic interventions are going to happen in the lower oximeter ranges. So really need to tighten that up in terms of, that 2% sounds fine with me. I mean, the lower the better. I don't know the technology enough to say how low can you go, but we could be California and turn all electric cars in 2033. We could tell manufacturers by X date, we expect your oximeters to be within 0.5 to 1%. But I'm saying really low, because we're making therapeutic interventions, withdrawing therapy and adding therapy based upon this. It's no longer used as a monitor.
So let me try and summarize, unless anyone else wants to weigh in on A, which kind of we've addressed a little bit of B already in terms of folks with different skin pigmentations. Dr. Loeb, I see you have your hand up, and then Dr. Katz.
Dr. Loeb, I know I talk a lot. Sorry about that. I think there is a problem with arms, and that was presented, and it's not the way clinicians think. It was brought up, and I agree with Dr. O'Connor that the problems that we see in the clinical studies are that people have a normal saturation, but they're hypoxemic. That's a big problem. It should be, it's doable, and it should absolutely happen that high saturation, if you have a pulse oximeter with a high saturation, there's virtually no chance that you're going to have a true low SaO2 functional low saturation. So the high levels, it can be and it should be very, very accurate in a subgroup analysis just at those levels. As the levels go down, technologically, unfortunately, I'm not sure that it's achievable that level of accuracy is maintained. So I would be okay with the level of accuracy being somewhat lower at the lower saturations, especially if the curves are going to have to be done to account for disparities in pigmentation. So I would say that I agree that there should be something more clinical in terms of clinical thresholds and making sure that people don't get misclassified at 92% and 85% or something like that. I think we can sort of choose almost whatever number, but something up in the 90s and something down in the 80s.
Thank you. Dr. Katz?
Yeah, I think the discussion mostly deals with 0.2, the levels of SaO2, which I agree with. A1 is the clinical setting, and I think what they're referring to is low perfusion states. So if a company is going to make a claim that the oximeter is accurate in low perfusion states, they should be required to study it under those conditions, which may be very different than providing the FDA with data during wakefulness, awake, under normal conditions.
Thank you. Dr. Lynch?
Just want to comment on what Dr. Loeb just said about him being okay with worse accuracy at lower saturations. I feel like that this entire day we've been focusing on hypoxia as like the pathological state in people that would normally be at 99%, but there's a growing population of congenital heart population where their resting saturation is 80% or 75%. And we also need to know when they have pulmonary illness. So we would need an accurate pulse oximeter for these patients in those percentiles as well. So I would just wanted to point that out, that it's not that everyone needs a more accurate pulse oximeter at the higher percentages.
Dr. Wilson, good point. Thank you. Dr. Wilson. You're muted.
Yes, you're muted.
Sorry. I initially raised my hand to counter the point that Dr. Lowe made about it's okay to have a wider ARMS or whatever the statistical methodology is at the lower saturations. That's where it really matters the most. And currently, I believe all manufacturers can easily have a 2% ARMS across the entire spectrum, 70 to 100%. And if in the future you made it 1.5%, something that they would try and achieve in the next couple of years, that's very doable as well.
Thank you for that insight. Dr. Connor.
Yeah, I think one other metric that I would recommend, because the issue is now we know there is this racial or skin tone discrepancy, that I would recommend, say, a minimum number at 88 or at 92. I would let the clinicians recommend what the right number is, but then show what is the mean bias, for instance, between Massey Martin 2s and Massey Martin 8s or something like that. Because I think the problem is right now you can hit the arms limit because you don't need many dark skinned people. So, the 34% of people you're allowed to have outside the range, or 34% of measurements can just all be in a type of person that leads to racial discrepancies. So now, if we have docs who care about racial differences, and hopefully 100% do, this would enable them to look at the label and say, oh, this one has the smallest bias between light skin and dark skin patients. So I think I totally agree with what others have said, but I think we need a clear, here at 88% saturation, according to an arterial measure, this is the bias between light and dark skin patients. And then we can, comparison shop accordingly.
Thank you. Dr. Kasseri.
Yeah, I just wanted to kind of step off on that, Dr. Connor. I mean, thanks for pointing that. I think that error rate should be in individual skin tone racial groups as well, not just the whole group. And that's calling the company out and saying that you can't just mix the data up and you made the threshold. I want to see the threshold in all the different color tones. I think that should be the expectation.
Right, and maybe do 2 5s and 8s. We don't need, all 10 on the Massey Martin scale, for instance, but right, at least 2 5s and 8s is a minimum. Absolutely, agree.
I see Dr. Edelman has her hand up. Dr. Edelman. Are you muted?
Can you hear me?
Yeah.
Okay. I was just trying to respond to something Dr. Connor said. I just wanted to make sure it's clear that we're trying to get a recommendation from the panel for the minimum acceptable parameters that would be adequate for a new pulse oximeter to go on the US market. So in other words, it is not for the purposes of comparison shopping, but what is minimally good enough. Thank you.
Thank you.
Right. No, and I agree. I mean, if I can address that, but I think showing what the difference is in light and dark skinned people. So maybe it's not comparison shopping, but maybe it's to say this is good enough and I'll buy it, or this is not good enough, and I won't use it in my hospital. So I didn't necessarily mean comparison shop, but it's, I think, the key metric that this whole panel is about. There's a difference in light and dark skinned people. Put that number on the label.
Dr. Lynch.
Sorry, I forgot to lower my hand. Sorry.
Oh, sorry. Okay, Dr. Hennessy.
Thanks. So we've been talking mostly about ARMS, but we haven't talked about its use as a diagnostic test to identify hypoxemia. So should we recommend that, sensitivity, specificity, positive and negative predictive values for identifying SpO2 less than 88% be a requirement? And both overall and in subgroups based on skin pigmentation.
I'm not sure we should go there. 88 is a number that's touted as being this magic threshold, but it's really kind of a spectrum as well in terms of oxygen saturation. Let me see if I can try. There's a lot of information to process, and this is going to be a difficult one to summarize, but let me see if I can try, and I'll give it my best shot. So actually, yes, it should be based on the clinical setting. Healthy volunteers is one thing, but in the hospital setting, or at least simulated hospital setting, I think becomes very important. We heard about the importance and the influence of hyperperfusion. And certainly you heard a lot about the level of oxygen saturation. So there needs to be validation across the SpO2 spectrum from 70 up to 100%. I heard, I think from Dr. Connor, reporting on bias between folks of different skin pigmentation, but what the FDA wants is the minimal requirement to get approved. And what I would say is if the FDA does mandate that the arms gets tightened to, we heard that 2 is achievable, 1.5 is more achievable. As a clinician, I have a tough time contextualizing what this means for my patients in terms of how reliable the oxygen saturation becomes. All I know is that it does become even more reliable. And I don't know how prescriptive we need to be beyond that, but what I would say is that threshold needs to be applied not only 90 and above, but 80 to 90. And I don't know if it's feasible to have the same kind of tight range between 70 and 80. So what I don't know, we know that pulse oxes get more inaccurate as you go lower. What I don't know is, does that make the arms of 1.5 less achievable as you get lower? And I think someone with technical expertise can answer to that. I think that what we should strive for is not the bias between different pigmentation. I think that all pigmentation types should be represented equally. And what I would also kind of guard against is having a label with all these different things, bias, arms, that becomes uninterpretable to the average clinician. So once again, this is a difficult one. I think the FDA has a lot of information and input from us, and hopefully we've addressed at least 3A adequately. I think 3B, we've clearly addressed that there should be equal representation across different skin pigmentation. So that's kind of an easy one. Let me see what 3C says and see if we've addressed that. Please discuss if arms is an appropriate measure of device effectiveness for clinicians and users. If you do not believe arms is appropriate, please discuss alternative methods to assess the accuracy of a pulse oximeter. I heard mention of reporting the performance characteristics, sensitivity, specificity, positive predictive value, etc. That's something that resonates more with clinicians because that's what we're used to reading in clinical trials. But I would think that if you have a tight arms, that can easily be converted into those kind of performance characteristics. But I'm not sure that there's rationale for looking at the sensitivity specificity to predict specific thresholds and cut points like 88%. Dr. O'Connor, do you want to weigh in? Bail me out here.
You had it perfectly. I mean, I think what we should do is for 88%, 89%, 99%, whatever the oximeter is is reporting, the company should have a positive predictive value for that value. Like, how likely is it that it's that number? How likely is it that it's 1 or 2 higher or 1 or 2 lower? And I think they should do that across the entire domain. I think that's
thank you. Maybe we can stick with the recommendations of an ARMS of 1.5 or 2, but then it's up to the companies to translate that to something that's more clinically meaningful as just articulated. Does that seem reasonable to everyone? All right, Dr. Edelman, with regards to question 3, have we addressed this adequately?
Yes, thank you for your thoughtful comments.
Thank you. All right, let's move on to question 4. I believe question 4 is our last question. Current labeling for prescription use pulse oximeters is intended for clinicians, and generally it does not address inaccuracies that may be associated with skin pigmentation. In your deliberations, please discuss labeling modifications to address inaccuracies that may be associated with skin pigmentation. Well, I think I'll weigh in first, and then I'll let the group weigh in after me. I think that what we would hope to strive for is that there is accuracy across skin pigmentation, but certainly if there is any inaccuracy, there needs to be transparency around it. How much inaccuracy is permissible? I'm not sure that there's a right answer, and maybe, to say that the pulse ox needs to perform exactly the same with a saturation of 75% across the spectrum of pigmentation might be unrealistic. My hope is that the ARMS incorporates the accuracy across the spectrum. But I think whatever the inaccuracy is, there needs to be some transparency so that at least people who are using the pulse ox are aware of this as an issue. I'll let the panel weigh in with maybe a different perspective on this, or how better to frame this. Dr. O'Connor.
So once again, I think the criteria should be that the 95% confidence intervals for all of the different identified skin types, skin pigmentation, should be overlapping. They don't need to be identical. They need to be statistically indistinguishable. And you need to have enough people, enough subjects to have really tight confidence intervals. I don't want to see 10 and 10 with confidence intervals that span 25%, which is crazy. I'd like to see confidence intervals in the measurements that are in the range of the ARMS, is the aspiration here, or a little bit smaller, so that they're indistinguishable.
Thank you. Dr. Yilmaz.
Another question to FDA. What and perhaps I should know this, but as of now, what does the label address for inaccuracies for pulse oximetry?
Dr. Edelman, can you address that?
So currently the labeling summarizes the performance, but I'm going to ask Dr. Lee to comment further.
And I guess just for clarity, I think it would be helpful, right? I mean, hopefully it's intuitive, but understanding that, I think, will help frame the recommendations we can give associated.
Yeah, there's also 510K summary and performance, which is a public document, which shows the data upon which the 510K decision was made for all of the prescription use devices. Dr. Lee, is there anything you wanted to add? Okay, I believe he's having issues with his camera.
Can I just answer that question?
Oh, there he is. Sorry, go ahead, James.
Go ahead, Dr. Lee.
Oh, yes. So there are two things. One, we have the regulatory disclosures, where on the 510K websites we would disclose, in the summary for the clearance, and also certainly the labeling, and that's something that we would also highly encourage and have looked at. making sure disclosures about accuracy, particularly in some cases, companies disclose accuracy in what decile. But certainly, I think these comments are welcome, and it will fuel decision-making on the pre-market and post-market side. Thank you.
Thank you. Dr. Connor.
Yeah, I just wanted to clarify or add to something that Dr. O'Connor said about overlapping confidence intervals. So I think we need to be careful because things can be pretty far apart and still have confidence intervals overlap. So I think the meaning of what you're suggesting is 100% spot on. I think executing it that way is probably too liberal. For example, if we measured a whole bunch of people who had arterial oxygen 90, and we saw the mean was 88.1 in light skin and 91.9 in darker skin, both of those would have an arms less than 2, and the confidence intervals would overlap, but they would almost be 4 percentage points apart. So I don't think we're saying that's okay. But I think given other things we've said today, it would actually meet the criteria for okay. So I just wanted to point out that just using overlapping confidence intervals is probably too liberal, that we would want to say, that the bias, that average difference between things that are truly the same, the arterial pressure is the same. The mean difference we see between light and dark skin is sufficiently low, like 2% or like some number that, I would defer to you that is clinically important.
Okay, that's a good way of framing it. Thank you. So coming to 4A, labeling modifications to address inaccuracies that may be associated with skin pigmentation. I think what Dr. Connor just articulated resonated with me in terms of the transparency that should be around it. It's maybe too high a bar to ask that everyone be exactly the same, but that the bias and the bias difference between Caucasian or whites and darker pigmented folks be in the labeling. How much permissible bias it should be really minimal. You mentioned, I think, 2 or 3. And so, but I think the main thing is, as the arms is tightened, make sure that there isn't too much of a discrepancy between folks of different color. All right. Recommendations for the content of labeling for lay users who may use pulse oximeters at home. Anyone? I think we did discuss this. Dr. Hennessy, do you want to weigh in on this?
Sure. I think that for the products that receive little regulatory oversight, for which we don't have much of an idea about accuracy, that the label and the device itself should, accurately portray what's known about the accuracy, even if nothing is known about the accuracy.
Okay, thank you. Mr. O'Brien.
Yes. I don't know what that was about. Yes, Joe O'Brien, patient rep. I clearly would not discuss arms or any other of that technical information within the lay user content. I think what's important here is just well, first, the message is given for these devices that they're not for medical use. That's clear. But knowing that they are being used in a variety of different ways, I think we have to address the fact that what works, it seems, in the patient community is plus or minus. So that, and it is given in various areas I saw on websites, et cetera, that they're being told or communicated already that there's a plus or minus 4% variance. So if you have a 90, it may be 86, or maybe, and some of them use a 6% variance. I think in that process, that's probably a good way to communicate it, to say that there is to be aware that there is overall a plus or minus 4 or 5% variance. And what that means, it could be 5% more, 5% less, and that this variance increases with people of color. So that you should be aware of the variance, and to ensure that you communicate with your physician to follow up if there's any question, something to that effect. And I think, as I said earlier, in this day and age, I think we have to go beyond just the typical labeling. I think it is important to have whether it's on the FDA website or on the company website, but a clear audio visual that explains to them how to use it, what it means, what that variance is, shows the color shades, etc., in a very succinct way that they're used to doing right now. And that's the way they get their information. I think it would behoove us to use that same approach to make sure that the patient population is properly advised.
Okay, thank you. Dr. Kasseri.
Yeah, I'm going to echo that, Mr. Bryan. You know, my thoughts exactly. I think it should be clearly labeled not for medical use. And I would just put it in lay terms. You could have a little package insert there that says not for medical use. Basically, you could have normal oximeter readings and still have a low oxygen level, seek medical advice, something along that line. It has to be, you know, Mr. O'Brien opened up that sheet. Who could read that? It's just lines and lines of minutia. There should be a requirement where there's a small package insert that is there that says not for medical use and some verbiage on there. Why? Real plain English. And I think that should go a long way. And if consumer devices want to reach the threshold to catch up to medical in-hospital use, well, then let them market that and let them get FDA approval for that, and they could win market share if consumers want to use that product.
I think you articulated that very nicely. It just needs to be clear in plain language, big letters, that this isn't a medical device and the reasons why not. So thank you for that. Dr. Collett.
Yeah, just a comment. I do think it's a little tricky. I looked at a couple package inserts and they don't really even, and just from like family members, like who've gotten these, they're like, well, what's normal? So like people don't really know, you know, is 85 okay? So, you know, they don't really know that. They don't know what's normal. So when you say a plus or minus 5%, you know, that's a big spread. I agree 100% it should say that, but I think this whole labeling for lay users is really tricky because, as everybody's iterated, it is being used for medical, but they don't say in the labeling, oh, what's normal, what's abnormal. So I think it's a little challenging for sure.
Thank you. Let me go to Dr. Eidelman. I see that she has a hand up.
Thank you. I just was hoping that the panel in their deliberation in Part B also communicates their thoughts on content of labeling for prescription use for home. In other words, prescription devices, not just OTC, when they are given for home use. Is there something, should there be patient labeling, and what should be in that labeling? Thank you.
Yeah, that's a tough question that I don't know that we addressed at all. But what I would say is that if they get a prescription device to take home, it really becomes the interpretation of the physician who's prescribing it for them that becomes very important. I don't know that the label should say anything other than please refer to your physician's instruction on how best to use this and when to inform him of change or what he or she would regard as significant change. The physician is writing it for a reason that they're concerned. I don't know that a physician would just give it to the patient and say, here, go. There's got to be accompanying prescription instructions, much like, you know, take this medication 1-3 times a day. There has to be some kind of instruction on how often to monitor. And maybe that's what the prescription needs to say. Maybe it's not just provide a pulse ox. And maybe this comes to the pharmacist that, okay, I'm giving the pulse ox, but what are the instructions for use and when do you call? Or when do you go to the ED? I don't know if anyone else wants, I see a lot of good hands here. Mr. O'Brien, I'll come back to you. Dr. Klein, go ahead.
Sure. I actually was going to comment on the OTC portion. I totally agree with the simple messaging. I am a little worried since we don't know exactly how well these perform of really giving a, you know, within 5% up or down. But then also, you know, that people don't know what these mean, but also when we think about some populations that are not normally saturated at baseline. So one person's normal is not another person's normal. So just getting into that consideration as well. So thinking about the cyanotic heart disease children specifically.
I think that's where doctors' instruction become important, not just call if your SAT's 80%, because they might be walking around with SATs of 80%. Dr. Yarmus.
Thanks, Lonnie Yarmus. I have two thoughts on one of each group, I guess. The first is for the over-the-counter aspect. I think, again, I'll advocate for, I think there needs to be some communication directly to physicians about this, because I think a lot of that is where this lies, right? Better understanding from physician groups that these are proposed to not be as accurate and should not be used under routine care for patient monitoring, which then gets to the second for the prescribed devices. Are we working under the assumption that because those devices are FDA cleared, they are of the same standard as those used in the hospital?
That's my assumption.
Okay, then if that's the assumption we're going with, then I totally agree with what you said, Dr. Nathan, that this should fall under the purview of the prescribing physician.
Thank you. Dr. Lum.
Hi, I would agree with what you said, Dr. Nathan, about for the prescription products, that's really the purview of the physician. For the OTC, I agree with this general sentiment, but I can't say I completely agree with what's been said before regarding it should just say not for medical use, because first of all, that's a peculiar thing since we've already said that these are being used for medical use, since no one's really using these as a toy or as a form of entertainment. To the extent that they're not to be used by physicians in lieu of prescription products, that's certainly true, but that's not a message for the general public. I mean, the general public are not physicians and they don't really even know what medical use means. So since these devices are going to continue to be used by the lay public as a screening device, essentially, as a home screening device, I think we should give more detailed instructions on what they're supposed to do with them. I understand we don't know exactly what their accuracy is, but I would suggest that FDA might want to consider a labeling change so they can find out, so they have then the purview to find out what the accuracy is and potentially to create some benchmarks, which even if they're not able to be implemented in the very short term, are reasonable, do not super increase the cost of the product, but are some benchmarks that might be implemented further downstream. But in the meantime, would allow the FDA the ability to investigate these products and give consumers some guidance as to how to use them and have some power to actually, at the very least, compel the manufacturers to convey that simplified and expanded labeling to patients who will in fact be using these to try to save their own lives.
Ms. Edwards, do you want to weigh in?
Yeah, I was just thinking as a consumer, simple language, I was thinking that to address it, you could say that, put in there that the levels of melanin in the skin may cause discrepancies or inaccuracies in the measurements. Or it may be too simple, but that's
I think that there's a lot that can go in a label, and maybe that's something that is a quicker fix, at least for now, and certainly for the consumer devices that there's less regulatory requirements around. There should be some kind of warning around that, I would think, to the point of the consumers have it, and I still agree that the labeling needs to be very simple. And you could nuance it, not for medical use, not for medical decision making. So, it's kind of a warning: if your SATs go down, don't make a decision, but you might want to act on it and call your doctor, and it needs validation, but you're not going to make a big decision around it. So I'm not sure if there's a middle road around this, but I think the warning clearly needs to be there both about you can't rely on this. It might guide you a little bit, not only because it's inaccurate in everyone, but especially in patients who have increased melanin content. So getting back to B, Dr. Loeb, bail me out.
Well, in thinking about both of these labeling questions, I think about pharmaceuticals. And first of all, if I get vitamins or some other a pill that you take that's not regulated by the FDA, it says on it, not regulated by the FDA. And I think it's appropriate for things that devices that FDA does not check on, that it says the accuracy of this device has not been verified by the FDA. Basically, in addition to, and it's not for medical use because its accuracy has not been evaluated. I also think that when I get a prescription, I go to the pharmacy, I get a thing, and they give me a whole big sheet of things that are mostly things that your doctor was supposed to tell you about side effects and how to take it, et cetera. And I think that all pulse oximeters come with labeling that says it's affected by motion, it's affected by dyshemoglobinemias, all this sort of stuff. And I think the layperson should know these are the things that affect its accuracy, including pigmentation. And lastly, I was really surprised that the FDA presented in the very first session what labeling is on pulse oximeters, and I did not know. It looks like it's up to the company what they put in about what affects their pulse oximeters, but the same things affect all pulse oximeters pretty much. So I don't know why the FDA doesn't have a list of all these things, dyshemoglobinemias and certain nail polishes and motion and pigmentation. All of these should be, I would think the FDA would have this list, and the companies, it'd be incumbent on the companies to say, no, ours is not affected by that. Otherwise, you got to put it in there. It's affected by that.
I'm trying to think of a simple way to help the FDA in terms of the labeling. Now, with the consumer devices, do they go through the FDA at any point? It's just a different threshold or standard that they're held against? Because maybe a simple way is for the label to say not approved by the FDA for medical use or something like that. And I think keeping the warning simple that people can see and it's very evident and it's not buried in the background, I think is effective. It should be a simple sentence, something like that or something else. But to that point of that 19-page thing and, all these caveats. And then, but I think that it needs to be out there that people can see a message like that. Anyone want to endorse that recommendation? Very simple, not approved by the FDA for medical use.
Sounds great.
All right. Dr. Edelman, with regards to question 4, did we address this adequately?
Yes. Thank you
very much. I'd now like to ask our 3 representatives, Beverly Edwards, our consumer representative, Dr. William Wilson, our industry representative, and Joseph O'Brien, our patient representative, if they have any additional comments. Ms. Edwards, you first.
Okay. No, I thank you for inviting me to be on this panel, and I do think this is something that's of we need to discuss and in the discrepancies and the disparities in oximeters and just to the FDA. It really happens, and it's not just oximeters, it's across the board in other areas of devices. Again, I thank you, and I think I've made enough comments today.
Thank you for that. But I think you bring up a point that came up in the morning discussions by one of the speakers, is that it's not just pulse oximetry, it might be other devices as well. And perhaps one of the recommendations is that the FDA should be proactive and look at these other devices where skin coloration may play a role in inaccuracy. So thank you for that. Dr. Wilson, do you have any comments?
First, just like to thank the panel for including me and during all of my many comments as well. But really, my parting words would be that the public is best served when we ensure the quality of the devices by raising the bar. That doesn't unfairly bias any particular company. Everybody, if they're they want to do the work, is going to now provide devices that are going to more accurately measure pulse oximetry for our patients. So that's the goal, and I really support the discussion that we've had here.
Thank you very much. Mr. O'Brien, any comments?
Yes, as the others, I would thank both the panel members and the FDA for today in addressing the issue. Over the last several years, the risk associated with these devices has been redefined. And it is clear from the evidence, et cetera, that if we don't redefine the accuracy, then we'll continue to potentially harm all populations, and in particularly certain populations that are in the Black community and the darker skin. And we've got to start eliminating the disparity. And I think that this type of dialogue and these type of days are what's absolutely essential to begin. And I compliment the FDA. And I once again, after another panel, say I'm certainly glad I'm not them.
Thank you very much. I'll now turn the discussion over to Dr. Adelman from the FDA for summary remarks before we adjourn the meeting.
Thank you. I want to thank all of our invited speakers, and a special thank you to all of the open public hearing speakers. We truly wanted to hear opinion from everybody, and I'm really excited that so many people have signed up and have taken the time to come and share your valuable input with us. I also want to thank all of you, the panel members, for very thoughtful and thorough deliberations during this very long day. We really appreciate your commitment to the public health. And last but certainly not least, I want to thank a very extensive FDA team that made today possible. I now pronounce this panel meeting complete. Thank you.
Thank you. I'd like to thank the panel, the FDA, the invited speakers, and all of the open public hearing speakers for their contribution to today's panel meeting. This meeting of the Anesthesiology and Respiratory Devices panel is now adjourned. Have a pleasant evening, everyone. Thank you all very much.